Good morning. My name is Lorraine Stiehl. I'm the Chief Executive Officer of the American Liver Foundation. It is an honor for me to welcome all of you to the 31st anniversary of the annual Irwin M. Arias Symposium, Bridging Basic Science and Liver Disease. More than 45 years ago, a small group of physicians from AASLD recognized the devastating impact of liver disease on patients. Together, they created the American Liver Foundation, the nation's leading nonprofit patient organization promoting liver health and disease prevention. Today, we remain steadfast in our mission to promote education, support services, advocacy, and research for the prevention, treatment, and cure of liver disease. It was in the spirit of fostering community, advancing medicine for liver patients, and highlighting the power of basic science that the ALF voted over 30 years ago to hold an annual symposium named for Dr. Irwin Arias. This program honors Dr. Arias, a founder of the American Liver Foundation, who is a longtime board member of both our New England division and the ALF National Board of Directors. Dr. Arias has contributed substantially to the mission of the American Liver Foundation, particularly by supporting young PhD and physician scientists interested in the pursuit of liver disease. In the early 1990s, around the same time the symposium was created, the American Liver Foundation established the Irwin M. Arias Postdoctoral Research Fellowship Awards. Since 2005, 15 postdoctoral awards have been given for this endowment. Later today, you'll be hearing from Dr. Arias, not only a visionary leader for the American Liver Foundation, but he's also a personal mentor to me as CEO of the American Liver Foundation. I would also like to recognize the extraordinary leadership of 3 research visionaries, all from the Boston area, who made this program possible. Dr. Sangeeta Bhatia, Dr. Wolfram Gosling, and Dr. Heather Fleming. We are so grateful to you for the countless hours you spent to make the vision of the Arias Symposium a reality. All of you who are attending today's program are so important to over 100 million Americans battling liver disease. Through your own ongoing liver research and this annual event, we can bridge the advances of basic science with better diagnosis, treatment, and prevention of liver disease for liver patients and their loved ones and caregivers. Today, I am very excited to share great news. We have an important announcement to share about research at the American Liver Foundation. We will be taking our basic and translational research to a much higher level than in the recent past. We plan to direct significant research to fund early career liver researchers, plus provide support for research training programs. Expect to hear so much more about this exciting program in 2023. Again, thanks to all of you for all you do to fuel patient medical advances made possible through your heroic basic translational and clinical research. All of us at the American Liver Foundation are so proud to partner with you. So I've now been asked to share a few housekeeping announcements. This program is being delivered on the Zoom webinar platform and will be recorded. During today's presentations, if you have a question, please hover your mouse over the bottom of your screen and click on Q&A. Then simply type your question into the Q&A box. Again, please note this program is being recorded and will be available on the American Liver Foundation's YouTube channel for future viewing. Now, I know some of you may have a challenge getting on. We started to get alerted. Please contact Megan Glynn, mglynn, Glynn, at liverfoundation.org. She will be happy to help you. So lastly, we have so many extraordinary speakers today sharing the latest and greatest in liver research. Please spread the word to those you know who would be interested in joining us today, or have them watch the program on YouTube channel. Now it's my distinct pleasure to introduce Dr. Sangeeta Bhatia of MIT, one of our amazing organizers of our program. Thank you, Sangeeta.
Hello, can you hear me?
We can. Thank you, Sangeeta.
Great, good morning. I know there are some technical difficulties, so we're just going to keep on going with our introductory remarks. I know a new link is getting hopefully emailed out to everyone, so I'm just going to keep right on going. And if I'm not done, if everyone's not online in 5 minutes, then Wolfram is going to come on and we're going to have an interesting liver conversation. So, hi, I'm Sangeeta Bhatia. I'm a liver bioengineer based at MIT, and I'm so thrilled for the day that's ahead of us. Together with my co-organizers, Wolfram Gosling and Heather Fleming, I really want to welcome everyone to what promises to be a really thrilling day of science. The 31st annual Irwin M. Arias Symposium on Bridging Basic Science and Liver Disease. We want to thank the American Liver Foundation with Lorraine Stiehl and her entire team, our sponsors, and of course, our inspiration, Win Arias, who you'll hear from as the day unfolds. Many of you know, and you heard from Lorraine, that this meeting has a rich history. In the past, there have been many notable presenters from within and outside the liver field. And this meeting has been a place where collaborations and friendships and discoveries and careers have been touched by what happened on this day. So a few years ago, when pre-pandemic, when Win passed the baton to Wolfram and I, we were very happy and honored to accept really the privilege of co-organizing the meeting. And I will say the single best idea we've had was to recruit Heather Fleming as our co-organizer. She has been the architect and driving force behind what you'll experience today, and we're really very grateful for her heroic efforts. As the three of us have evolved this meeting, we have sought to really stay true to the concept of bridge building between basic science and medicine. Really believe that that is where the magic of new ideas, discoveries, and inventions for our patients is going to happen. And we've also sought to include industry colleagues, all with the collective goal to impact patient lives. The pandemic has taught us that we can really leverage this digital format, I hope, as long as this link works very soon, and we can convene a broader conversation so that we have sought out speakers around the globe, and we've been very intentional in collecting them as well as deliberately diverse voices in what you'll see today. So the way the meeting will go is we'll have 4 sessions of science. There'll be 30-minute live talks. as well as what we call microtalks. These are 2-slide talks that were selected from submitted abstracts and really provide an opportunity for the next generation of liver enthusiasts to share their science and gain some visibility. Finally, in the spirit of discovery, we are always trying experiments. This year, our experiment is a lunchtime roundtable entitled Meet Me on the Bridge. And the moderator for this session is Nicola Amambakis. She's the editor-in-chief of Med, which is the flagship medical journal from Cell Press. And Wynn and several others of us will participate. And as I said, as an experiment, the idea is that while we love the ability to involve more people in this digital format, we all miss the personal human interactions that happen at conferences. We miss that sort of cocktail chatter, a chance to bump into a colleague or to meet a new one, to discuss, learn, and wonder together about the topics of the day. So our lunchtime roundtable is really an attempt at recreating that spirit. There will not be cocktails, but you can channel them if you like. And also we want to model for our trainees how these kinds of interactions can lead to new ideas. So with all that said, please sit back and enjoy the day. I'm going to make sure that we are on track. I think the panel numbers are hopefully increasing. And I'm wondering if I should get started, or Wolfram, if you want to join me on screen and we could talk a little bit more. But maybe what I'll do is introduce the first session chair, and together she and I can talk a little bit until the more participants are in the room, and then we can invite our first. Welcome, Wolfram.
Hey, Sangeeta, and welcome everyone. I'm getting feedback that I think the new links that are being sent out are working. So hopefully you and I, Sangeeta, don't have to have a one-on-one conversation for too long. But since I'm on, I also wanted to welcome everyone, and I'm truly excited about the spectacular program we have. And hopefully we can. I see the numbers rising here on my screen, so I think we should be almost back on track as we approach 9:15.
Great. Well, good morning to you. Maybe we could pre-conference chatter. What do you think?
I think that is perfect.
So I know that there's a lot of trainees in the crowd, and maybe we could just tell you the story of Wolfram and I met, I think some time ago. We've both presented at the Arias meeting. This is Wolfram studies zebrafish, which and liver regeneration in zebrafish, amongst other things. And I study liver regeneration in the human system, amongst other things. So we've always shared that love of the liver together. This is a meeting where over the years, I was really privileged to start a collaboration with Charlie Rice on hepatitis C back in the day. That was sort of my most, I think, notable Arias life-changing event. I don't know, Wolfram, if you have one to share.
Oh yeah, I have a story to share. I was a very young, a recently arrived a graduate student at Harvard, and in 1993 was at my first Arias Symposium. That was number 3, and it was in a big hotel ballroom, and I remember Tom Starzl giving one of the lectures, and I was truly transfixed. And I think from that day on, I have not let go of the liver. And, that's almost 30 years ago, which dates me, but also, shows the storied history and impact of this symposium, and how it both, I think, brings people together, leads to collaborations, as you highlight, Sangeeta, ours included, and inspires people and inspires trainees. And I think all of that, if we think what we need to do in the future, we need to do better work and more work. And I think that's what we do here. But also we need to make sure that other people and younger people and investigators in the future do this. And so if we can inspire people to stay in this field, join our field, work on liver biology and disease. I think that is also an important aspect of what we do here. And that bridge is not bridging, scientific fields, but is the bridge into the future. And that's important too.
Yeah, I totally agree. And I think, in this day and age where I think it can be challenging to be a physician scientist, right? I think a really important vehicle for bridge building is going to have to be that physicians and scientists are talking to each other, right? And and sharing observations and clinical challenges and inspiring new ideas. So hopefully the trainees on the line will see that this is a really exciting area to be in. I think I can think of nothing better than to study the liver your whole career.
I totally agree with you. And I think from the days we started and the challenges we have faced in the past, I think I started in the field when hepatitis C had no name. It was non-A, non-B, to now we have a cure, obviously. It shows the tremendous progress that we can make when we bring people together. But at the same time, when we think about alcoholic, non-alcoholic fatty liver disease, metabolic diseases in general, we also see the tremendous challenges that we are facing that we currently have no answers for, that affects people's lives every day. And we need to really bring our best every day and work together to find solutions and to find answers. And as you say, Sangeeta, I think for me, I can't come up with a better way to spend my time than studying the liver either. And I hope that we can pass that excitement and also impact of working in this field onto others, and hopefully out of this day and these interactions that we have here online, and hopefully again soon in person with real cocktails in hand, that we can do even better science and do it faster.
Well, great. I think that I see the numbers rising. I know that not everyone is online yet, but I also want to be mindful that our speakers have time slots during the day. So we are recording and we should get started. And I expect in the next 5 or 10 minutes how you can save your most interesting data for the end of your talk. And we should have a good audience for you.
Yeah, maybe I have. My lab keeps texting me because they can't get in. And so I have one question from the audience by text, and maybe we can answer this question and then you go ahead. And that is, how do you go about coordinating and setting up a meeting like this and getting such wonderful speakers? And let me just start with saying what Sangeeta had said before. Heather Fleming is an absolute rock star. And neither Sangeeta and I would be here on screen without her. But I also think when we think about our truly terrific panel of speakers today, I think we draw on both personal connections, we draw on friends and colleagues, but this is not just a friends of Sangeeta and Wolfram gathering. I think we also pay tremendous attention to representation and diversity and diversity both in the fields of research and study, but also when Sangeeta and I started this symposium, we made a pact and an agreement that we would have complete gender balance between male and female speakers. And actually, for all these years, we have maintained that. And you can, if you want to do the numbers on today's program, I challenge you, please do. This is not a manual. This is a true scientifically diverse, balanced panel. And I think that also makes for the attraction of our group and our events. And I think that is another way we can, guide into the future.
Yeah, I think that's well said. And it's very much by design. I mean, just to follow up, I think, we think about this conference all year long. So when we see an amazing talk or we read an amazing paper, it gets on our list of, oh, this would be great for Arias. And so I think it's very much something that emerges from group, a little mini community amongst ourselves, and sort of constant brainstorming and intentionality about who we want to include and how. We would love your ideas, other than getting the Zoom right, which we plan to do. we would love your ideas about other little experiments. The cocktail chatter idea actually came from a podcast that I listened to. There are three amazing, thinkers, and they just chatter. And we thought, oh, that's what it's like when you're at a conference, when you're at the bar after the meeting. You say, what did you think of that talk? Or what do you think of this? So we would welcome other ideas on experiments that you're seeing elsewhere.
Yeah, and also I think we today as well, we have ways for our trainees to and junior faculty in the field to be engaged, both as session chairs and with microtalks. And it would be great to get feedback on that too, because clearly we want to make sure that that people feel included, feel engaged, feel interactive, and that we do this particularly on Zoom as well, and as satisfying and gratifying as we possibly can.
So I'm getting the thumbs up from that. Maybe we should get started. Is your lab in?
I think people are in, and I look at the numbers, how they're rising. I'm not sure whether they are, are on our own panel link or whatever link they're on, but yeah, they're in, says Scott, one of my graduate students.
Good.
So at least we can view this as an extended lab meeting with a fantastic participation. But I hope that we had almost 450 registrants as of last night. I hope that that number rises quickly.
Yeah, good. Well, I think that you and I are not intended to ever be stand-up comedians, but hopefully it's been nice to start the morning with you, and it's good to see you. And I think what I'll do now is introduce the first session chair and the first session, which is Eugenia Uceanya. Welcome.
Thank you. So I will begin by introducing Dr. Hao Zhu. Dr. Zhu received his bachelor's degree in biology from Duke University and earned his MD at Harvard Medical School and Massachusetts Institute of Technology. He received training in internal medicine at the University of California, San Francisco, and in medical oncology at the Dana-Farber Cancer Institute. In 2012, he joined the faculty of UT Southwestern and the Children's Medical Center Research Institute, CRI, at UT Southwestern. He's also an attending physician in the multidisciplinary liver cancer clinic at Parkland Memorial Hospital. Zhu Laboratory is interested in understanding the relationship between tissue injury, regeneration, and cancer. And without further ado, I'll introduce Dr. Zhu and his symposium topic, Clone Wars, Evolution of Adaptation and Malignancy in the Liver.
All right, well, thank you, Eugenia. This is a very nice introduction. I would like to express my gratitude to Wolfram, Sangeeta, and Heather, as well as the American Liver Foundation for putting this fantastic symposium together. It's truly an honor to be a part of this and to celebrate Erwin Arias's life and contributions. And I'm also really excited to hear what the other speakers are going to show. So here are my conflicts. I think it goes without saying that the liver is an amazing regenerative organ, and it's right up there with the Hall of Famers in terms of regeneration. We've known this for a long, long time, but yet there's still mysteries about how the liver regenerates from a cellular and mechanistic basis. So my lab does devote a lot of its efforts towards understanding how this occurs. And we are extremely interested in trying to figure this out. However, we're also trying to leverage regeneration as a sort of a platform for gene discovery to see if we can find, using the proliferative nature of the liver, see if we can find novel genes in the context of cancer, regenerative medicine, as well as metabolic liver disease. So, you know, with even though the liver regenerates really well, with decades of injury from viruses or monogenic genetic disorders, alcohol or fatty liver disease, this does eventually lead to a condition called cirrhosis, which, as you can see here, is not a pretty situation and does lead to very severe organ dysfunction. This is accompanied by a whole host of clinical problems like high bilirubin, fluid overload with ascites, bleeding from esophageal varices, and brain dysfunction from toxic elements that aren't metabolized. And these are very difficult to treat in the clinic. We don't have very strong mechanism-based therapeutics of these kinds of problems. And as if that wasn't enough, at the end of cirrhosis, patients often get hepatocellular carcinoma, which is the most common form of liver cancer in the cirrhotic liver. And I'll say it's a very difficult to treat cancer because it's an aggressive tumor in the context of a non-functioning organ. So it's challenging to sort of deal with this cancer. And so as you can see, there's a lot of pressing clinical issues, and the field is grappling with how do we understand the contributors, the drivers, the adaptations, even the passenger events that are occurring in this wound healing situation. So with, and I'll tell you about some of these steps, with injury, you do have cell death and then tissue damage that leads to this sort of compensatory regeneration or proliferation. And I think what we're appreciating in recent years is that this leads to somatic genetic diversification or mutagenesis across the liver. And that there's this evolutionary process where individual clones are trying to figure out how to deal with this damaged liver. And some of these clones are adapting and some are causing the disease and some are doing nothing. But there's all kinds of things going on a genetic level and an evolutionary level here. And not only are there cell autonomous things happening, there are all these non-cell autonomous factors like inflammation, fibrosis, and then this culminates ultimately into cirrhosis, which is this sort of highly nodular end-stage liver damage that leads to HCC. So as you can see, there's tons of issues going on. There's a lot of moving parts, and a real challenge in our field is how do we grapple with the variables that are leading to this damaged organ and why is there organ dysfunction? I think a key insight is somatic mosaicism, because I think this gives us a genetic handle on how to kind of understand the things that are driving and adapting in disease. And just to take a step back, somatic mosaicism is we now understand that this is a common scenario in many of our tissues. And I'm highlighting the esophagus and the skin here. This is work from Inigo Martin-Carrena and Peter Campbell's groups at the Sanger, showing a high density of mutations in the skin here on the left and in the esophagus here on the right. These mutations seem to increase with age, as well as tissue damage, with smoking and drinking, as you might expect. And we sort of contributed to this field by trying to understand the somatic mosaicism that occurs in the cirrhotic liver. So a number of years ago, we sequenced, using whole exome sequencing, cirrhotic liver tissues that were obtained from surgical samples from HCC liver resections. And naively, we just took chunks of liver and did a whole exome sequencing, later realizing that we would need to go much deeper with our sequencing. So we decided to use targeted sequencing panels to look at approximately 150 genes and did much deeper sequencing into these tissues. And I have to say that these kinds of efforts represent the bridge that we're talking about at this meeting, where we have to integrate the efforts and the expertise of hepatologists, surgeons, bioinformaticists, as well as biologists in my lab. The upshot of all this is that we found many mutations, and that as you got more and more liver damage, you had more and more mutations. And what was interesting is that a number of these were involved in HCC or liver cancer formation, but a bunch of these mutations had nothing to do with liver cancer as far as we could tell. This suggests that there's a whole world of events that is occurring that is independent of cancer and that could be interesting from a functional standpoint and potentially needs exploration. So in the last couple of years, we've also repeated these studies, these ultra deep sequencing studies in additional patient samples and found that there's similar patterns of mutations occurring. So this is a highly reproducible set of findings. And notably, we found that PKD1 was one of the most recurrently mutated genes in both of our data sets. I think a contribution that we made to the field was to try to understand whether or not these mutations had some functional relevance and which ones might be more or less relevant. So without going into the details, because this is published work, Joyce Jha, a graduate student in my lab, devised a series of CRISPR screens in the liver in the setting of a hereditary tyrosinemia mouse model, whereby she could introduce genetic mutations and look at clonal expansion from a regenerative standpoint and ask which genes or mutations were causing clones to expand faster. And the take-home message here is that there are many genes that could lead to clonal expansion and improve regeneration. And some of these, such as ARID1A and PKD1, are also at the top of the recurrently mutated gene list. So there's some correlation with the frequency of mutation and also their functionality in a regenerative sense. So I've told you a story about proliferation and regeneration in the liver as it relates to mutations. So we were really excited when Peter Campbell's group at the Sanger had done their sequencing in NASH livers, and they found that there were recurrent mutations in metabolic genes involved in insulin signaling or lipogenesis that were recurrently mutated in different patients and even in different parts of the same liver with different mutations in the same gene. This strongly suggested convergent evolution between different parts of the liver, seeking to select for mutations that are driving an adaptation to fatty liver disease. And notably, most of these are not involved in liver cancer. So this concept really captured our imagination in combination with our own work. And so, but still, there are sort of fundamental questions that need to be addressed in this area. We think that the current state of the field is quite descriptive. So something that we want to try to do is to see if we can endow some functional relevance to these human genetic findings. And another key question is, if you have these tiny little clones that are mutated and they're adaptive within that little clone, does that have any benefit to the tissue or the organism? And also, if there's adaptation here, could this be used as a framework for identifying novel therapeutic strategies for liver disease? So a fantastic postdoc in my lab is trying to address these questions, and I'll talk to you about some of the work that he's done to address them. So a first step that we needed to answer was, can we take a gene with a really strong effect on fatty liver disease or metabolic liver disease? And what does that effect have on the clonal survival or expansion of a clone within a liver? So we picked a well-understood gene, or at least a well-known gene, called MBOAT7. This is a membrane-bound lipid synthetic enzyme that has something to do with phospholipid synthesis. This is identified by GWAS studies to be potently involved in human NASH, and it is well known by several groups, including Matt Mitchie's group at UT Southwestern, that when you delete MBOAT7, this does lead to fatty liver disease, and we have confirmed this in a liver-wide fashion in Western diet-fed livers. Now, we know that this is true, but the real question is, what happens when MBOAT7 deficient clones are surviving side by side with wild-type clones of the liver? What is their fate? And so to do this, Zishi took Rosa tomato reporter mice, Rosa lox tomato reporter mice, either wild-type or MBOAT7 floxed mice, and then gave low doses of AAV-CRE to set up this mosaic situation whereby you could either have wild type or MBOT7 deficient clones, and then trace these over time, over long time periods with either normal diets or Western diets, and then collected the liver and evaluated their clone abundance. So first, I'm going to just show you the data from wild type livers. So you make tomato positive clones in a wild type liver, and these tomato positive clones are wild type, and you feed them chow or Western diet. And interestingly, there's not much of a change in the clonal abundance. And that kind of makes sense because the red clones, the tomato clones and the non-tomato clones are both wild type. So there's no competitive difference. However, if you take MBOT7 deficient clones and you look at them under chow conditions, not really much of a difference here, but clearly when you put them on Western diets, these MBOT7 deficient fatty liver clones begin to die in a pretty dramatic fashion. So you might say, and we agree, that this is an obvious result. This is showing that lipotoxicity is real and causes cell death and disappearance of clones. But I think there was still an important result to get in order to establish the framework and the foundation for this concept that we can measure metabolic fitness using clone size and clone abundance over time. And this is the kind of idea that we want to pursue is if a clone disappears or if it expands, this could have a major effect on metabolic liver disease. So we wanted to do this not on a one-by-one fashion, but instead in a pooled fashion. We want to study many genes at once because each clone could represent a different genetic subtype. And so Zishi devised an approach called mosaics, or a method of somatic AAV transposon in vivo clonal screening. And that's a long name for essentially AAV-based CRISPR screening. And as you know, AAVs do not integrate in the genome. So we needed to engineer a system where the guides could be integrated into the genome with a Sleeping Beauty transposon integration sequences, as well as the Sleeping Beauty transposase within it. So it's really an auto-integrating AAV. It goes in the genome so that we can later measure the guide strand abundance to figure out which genes had more or less clonal expansion. And the general idea is to give these AAVs to Cas9 expressing mice that are DOX-inducible, generate somatic mosaicism, give them normal chow diets, and then evaluate their clone abundance over time. And then to do the same thing, but under disease conditions involving NASH diets like Western diet and high sugar water. If clones expand or contract specifically in this disease setting, then it tells us that gene has something to do with that disease. Either it could be therapeutically relevant or protective or maladaptive, who knows. One thing I'll say is that this system can be substituted for any liver disease model where you can add alcohol or you can add biliary injury. So it's a very sort of flexible system. This is to show you how the system works when you give different doses of AAV. We can delete PTEN either at low levels or at high levels. We can activate tomato at low or high levels. And this will come in handy later on when we're trying to validate the genetics of the system. So we have the system in hand. So what happens when we integrate human somatic mutations that are involved in NASH? What happens to their clonal fitness? So we picked about 63 genes that were bona fide NASH-related genes, some of which are germline, discovered through germline studies, somatic studies, or bona fide therapeutic targets. This puts the screening system or the somatic mosaicism to the test. Essentially, we do what I've said, turn on Cas9, give AAV, turn off Cas9, and then put them on 6 months of NASH-inducing diets. You get these fatty livers or normal livers, and then grind up the livers to measure the guide strand abundance to figure out the clonal dynamics. The results were quite interesting. What we found is that most genes had no effect on clonal abundance, but a subset of genes did have an important impact. So when you deleted, it seemed like when we deleted genes that were involved in lipogenesis, triglyceride synthesis, or insulin signaling, that they would promote clonal expansions. And this kind of made sense to us because if you protect a cell from getting fat, then it doesn't die in the setting of a fatty liver disease. Notably, DGAT2 and GPAM are triglyceride synthesis genes, SRABP and PPAR transcription factors in de novo lipogenesis, and some other important genes in NASH. A key finding was that ACVR2A and GPAM were the highest in our screening list here, and they were also identified in the human studies from Peter Campbell's sequencing efforts. So this told us that the mouse and the human were showing similar patterns of mutant gene selection. We validated these results by doing a mini screen. Instead of with 63 genes, we only checked 4 genes here, our positive controls, with guides that we know to work. And essentially what we find is that there's selective clonal expansion of the genes that we saw higher in the screen, but no expansion of control guides that don't target anything. So we have confidence in the screening results. Now we wanted to sort of focus on GPAM as one of the key findings, both the human and the mouse. And so we repeated the lineage tracing studies for GPAM, and for wild type clones, after 6 months of chow or Western diets, you see a little bit of a decline in the tomato positive clones. However, if you delete GPAM under chow conditions, not much happens, but notably under Western diet conditions, these GPAM deficient clones begin to expand modestly. And so I think this is a satisfying result because essentially this is a visual lineage trace of what the human somatic sequencing studies are showing us, that GPAM clones expand. And this is quantified here, and I won't go through this. So having shown you that this is a way in which we can identify adaptive clones, can we use this to systematically identify adaptive genetic changes in liver disease? And so we partnered with Yujin Hoshida's lab. So Yujin Hoshida is a well-known investigator in cirrhosis genomics, and he's looked at, he's taken NAFLD patients and HCV cirrhosis patient RNA sequencing data and gotten the differentially expressed genes, and then looked for transcription factors or epigenetic factors and further overlaid them with clinical data such as survival or histologic outcomes, and then narrowed down a list for us with 472 candidate NAFLD genes or candidate liver disease genes. So we divided this into a transcription factor and epifactor screen, and again found outliers whereby when genes are deleted, clones expand. The cool thing here is that most of these genes, we had no idea what they were doing in the liver. And so that gets us really exciting. However, there are some genes in here that were known to be protective against fatty liver disease. So that's also good. So the question is, we've got the screening approach. We can identify clonal expanded hits. Then how do you understand the phenotypes and the mechanisms related to these genes? So an advantage we have now is that where you used to have to delete genes one by one using conditional knockout mice, we can use our AAV Cas9 system to delete genes in a saturating fashion, in a much more efficient fashion. Then you can use your traditional studies to figure out if they have anything to do with NASH or HCC, do genomic studies, et cetera, et cetera. So the system that we set up is to take our mice, give a single AAV at saturating doses, give 3 months of Western diet, and then evaluate their phenotype after this point. We show that you can get pretty good deletion, about 75% deletion in several genes that we checked. So it's a pretty effective knockout method. Now, the cool thing is that we can now delete, we can examine 24 individual genes and compare them side by side to rank order the effect of these genes on liver disease. So here I'm showing you liver to body weight ratio after 3 months of Western diet with lower liver weight to body weight, suggesting less fat accumulation. And so our positive control genes sort of show us this. IRS1, SRABP, DGAT, all show protection against fat accumulation in the liver. And we see that a lot of these hits also do that but not to the same degree. However, we do have several outliers that we're now focusing on in our studies: TBX3, BCL6, and SMID2. These features also correlate with ALT, which is a measure of liver damage, plasma ALT, and also somewhat match with triglyceride content within the liver, as well as cholesterol content. And I won't go into the details here. Histologically, our top hits do seem to protect against steatosis as evaluated by histology here. And for one of our hits, SMID2, SMID2 is a protein methyltransferase that has histone and non-histone targets. And fortunately for us, this enzyme has an inhibitor called AZ505. And we wanted to use this as an orthogonal method to see if some of our genetic findings held true. So we gave this in the context of 2 months of Western diet. And indeed, we found that AZ505 could protect against fat accumulation as measured by liver weight to body weight ratios, as well as by histology. Actually, the effects here were almost more potent than our genetic effects. And these were seen in the liver weight to body weight, ALT, and liver triglycerides. So this shows us that this is a system that can identify bona fide therapeutic targets for fatty liver disease. Now, you're going to ask me, well, does this have any relevance to late-stage NASH features such as fibrosis, inflammation? And by virtue of selecting these genes to be correlated with histologic features of human liver disease, indeed, there was some correlation with these features, but we don't yet know the directionality of these associations. We've now examined some collagen-associated genes with many of our knockdown models, and they do seem to be reduced. But this is at a relatively early stage of liver disease, and we need to extend these studies out to 9 months or so. And we're beginning to do that, where we can knock out a gene and then give Western diets for 9 months or so. And we still see that TBX3 and TBX3 plus SMID2 have protection against liver to body weight accumulation, suggesting that this could be relevant to late stage NASH as well. We don't yet understand the mechanisms, but we have a working hypothesis. SMID2 has many methylation targets, including beta-catenin and STAT3, which are well known to be involved in fatty liver disease. TBX3 is a transcription factor that regulates lipogenesis genes. So we're currently evaluating whether or not the ablation of these genes limits the ability for beta-catenin and STAT3 to drive fatty liver disease in its many manifestations. Okay, so the upshot is that I think what's really cool is that our human livers are essentially performing CRISPR screens of their own, especially in the context of long-term liver disease. And when we sequence a human liver, we're essentially getting the results of those screens. And I think the mutations that we're going to find are going to have something to do with the important biology of the liver. And not all of them are going to be relevant to cancer. Some of them are potentially going to be therapeutic targets. So I'll just quickly highlight our conclusions. I think we've functionally validated some of the somatic genetic discoveries in liver disease. We think that clonal expansions can benefit tissue and organismal health. And we think that in vivo screening plus human genetics can identify genes for metabolic disease. I think a key point here is that we've focused for a long time on germline genetics. And the germline genetics has yielded amazing pathways. But I think the low-hanging fruit from that is gone. And so we need to begin to think about somatic genetics as another source for human disease genes. All right, I'll thank the people who did the work. Zixi Wang really did a lot of the work that I showed you in the talk. Min did much of the work, and Min and Joyce did much of the work for the original cirrhosis sequencing effort. And Lynn has contributed as the lab manager. And I'll thank my many funding sources as well. Thank you, and I'll stop there.
Thank you so much for that really impressive talk. Now I'd like to turn it over to the audience. I wonder if there are any questions.
Dr. Zhu,
Yes.
what is the role of energy, let's call it energy metabolism or energy regulation, in the various patterns of gene expression and the metabolic consequences that you find? Are there any modifications, for example, in the AMPK, LKB1 pathway? And are there mitochondrial regulatory things, perhaps involving the Krebs cycle, that underlie these genetic changes?
Yeah, we haven't, this sort of early days, we haven't gone into specific metabolic mechanisms. I think the net result of what we're finding, I believe, is that by any means necessary, there are ways in which clones are protecting themselves from an overaccumulation of toxic lipids. And even though the organism is upsetting the energy balance, individual clones can try to counteract that disparate energy balance to try to survive in this environment. I think that's the upshot. But there's going to be numerous storylines about how any particular pathways are acting in these individual clones. Thank you for your question.
And I would just like to remind the audience that if you have any questions, you can feel free to enter them into the Q&A box and I will read them out loud.
Hal, is this, am I unmuted? I think.
Yes, yes.
Oh, it's
Hi,
David
David.
Austin. How are you? Good. Terrific. I have a question for you. There is a notion that's developed that the capacity to make triglycerides would be expected to be protective from fatty acid toxicity, but your GPAM results maybe suggest that that's not really true, or at least under this setting. So I just wondered if you can comment on that, and then maybe have you manipulated any lipid droplet proteins to see if they've, confer survival or advantage or disadvantage?
Yeah, right. There's some idea that maybe certain triglycerides are benign or not, or even beneficial to hepatocytes. And I don't know why that, I don't know whether or not these results are really saying that that's not true. I would just say that it's clear from, the germline studies are also identifying GPAM as a regulator of long-term NASH. And so, potentially that, when you have triglyceride overaccumulation, it's not, that lipid species is not purely protective. There's other things that are happening that are counteracting that positive effect. So that could be one explanation.
Yeah, no, your system is so great because it pits, acutely pits one kind of versus the other.
I think it's nice because you can have a lot of internal controls for the same environment. And so you're really getting a fine-grained competitive understanding of which clones are benefiting and which aren't.
Yeah, terrific. Thanks.
Thank you.
We also got a couple of questions in the Q&A box. First is from Sean Guang. Hi, Hal. Great talk. I see FOXO1 on your final slide, but it's a gain of function mutation in the human samples, I believe. How did you test this in your mouse models?
Yeah, that's a great question. So the FOXO1 mutations are gain of function from Peter Campbell's group, and so the prediction from our models is that when you lose FOXO1, you get decline in clone health, and that's exactly what we see. So FOXO1 is when you delete FOXO1, those clones are rapidly removed from the population. So it's exactly opposite of the gain of function mutations. And that's why I think that on some level we validated that finding.
Okay, and I think we're just going to get to a couple more so that we can move on to the next talk.
Yes.
But this says from Jesse Kirkpatrick, super impressive work. Do you have evidence that clones that are protective and therefore clonally selected in chronic liver disease may be predisposed to cancer?
Yeah, that's a key question, right? That's the $1 million question. Why? Yeah, I have a question. When you look at a cirrhotic liver, it's got millions of nodules. Why are we only getting 1 or 2 cancers? It's almost as remarkable for the fact that it isn't becoming cancerous faster. And so I think there's, some of these adaptive clones are definitely not becoming cancer, because when you look at the mutations that are dominant within these clones, they never show up in HCC, or they show up at a lower rate. So I think there's a million different storylines for how this is going to go. A lot of things will contribute a little bit to cancer. A lot of things will block cancer a little bit. So any story you want to tell, you're going to find some genetics to kind of bolster that, I think. Sorry.
Gina, you're muted.
I'm sorry. Okay. This is a final question for this session from Anna Salkani. Did the patient sample with HCC have any history of previous injuries in early life?
Not sure about that. So when we look at our sequencing, we use the non-malignant adjacent tissue samples, and of course they have hepatitis C and they have NASH and alcohol. So they have a long history of injury, which has led to the mutagenesis. Right next door, there's an HCC, but it's fairly far away. So there's certainly a long trajectory of injury, and the more injury you have, the more mutations you'll see.
Thank you so much, Dr. Zhu. Again,
Thank you.
We'll talk, and then we'll move on to the next session. So I would like to introduce Dr. Bao-Liang Song. He received his BS degree from Nanjing University in 1997 and a PhD diploma from the Chinese Academy of Sciences in 2002. He was a postdoctoral fellow in the Brown Goldstein Lab in UT Southwestern Medical Center from 2002 to 2005. In 2005, he moved to Shanghai Institute of Biological Sciences at Chinese Academy of Sciences as a principal investigator. Since 2014, he worked as a professor and the Dean of College of Life Sciences in Wuhan University. He was elected to the Chinese Academy of Sciences in 2021. His topic for the symposium today is inhibition of ASGR1, decreased lipid levels by promoting cholesterol excretion. And now.
So I think we're working on getting the volume working, right? We're all not hearing anything, but we're seeing.
Correct. Yep.
Okay, so I think.
Okay, here we go.
It's my great honor to attend this wonderful meeting. Next, I will talk about our recent work on cholesterol excretion. Shown here is the structure of cholesterol. Cholesterol is a hydrophobic molecule. It regulates membrane function by forming membrane microdomains with sphingolipid and other proteins. Cholesterol is a precursor for bile acids and steroid hormones. Cholesterol can covalently modify hedgehog and smoothen the proteins, therefore regulate embryonic development. We know that high level of cholesterol is a risk factor for cardiovascular disease, NASH, and diabetes. There are about 330 million CVD patients, 200 million fatty liver patients, and about 130 million diabetes in China. Recent studies have shown that cholesterol accumulates in liver of NASH, in liver of NASH animals and patients. You can see the cholesterol crystal in the liver samples of NASH patients. We also find that cholesterol crystals can promote NASH. In normal mouse liver, no cholesterol crystal was observed. Nuffield liver showed some cholesterol crystal that was indicated in red, and NASH liver displayed a large amount of cholesterol crystals that was surrounded by Kupffer cells. The cholesterol crystals can recruit immune cells and increase inflammation and fibrosis. Inhibiting cholesterol biosynthesis reduced the cholesterol crystals and showed a therapeutic effect on NASH. A human adult excretes about 1 gram of cholesterol through feces per day. He also obtains such amount of cholesterol from dietary absorption or de novo biosynthesis. And the liver is the major organ for cholesterol metabolism. Currently, there are 4 classes of cholesterol-lowering drugs. And the first one is statin that inhibits HNO reductase and then upregulates LDL receptor. Bempedoic acid inhibits ACL and then upregulates LDL receptor. PCSK9 inhibitors can stabilize the LDL receptor, and ezetimibe blocks the NPC1L1 mediated intestinal class absorption. And the first 3 drugs increase cholesterol uptake by hepatocytes and may worsen NASH. Ezetimibe can only decrease cholesterol by at most 20%, and no drug directly promote cholesterol excretion. So although PCSK9 inhibitors can dramatically reduce cholesterol level in blood, they promote liver and other organs, such as heart, to take up lipoproteins. Some recent studies have reported that PCSK9 deficiency increases NASH in animal and humans, and the PCSK9 deficient mice increase heart failure because the heart also take too many blood lipids. And we should pay attention to this possible side effect of the PCSK9 inhibitors. As we know, among different glucose-lowering drugs, SGLT2 inhibitor promote kidney to remove glucose through urine and displays multiple beneficial effects on cardiovascular system and renal system. The reason is that glucose is completely removed out of the body. So can we use the same strategy to lower cholesterol? This idea is very important because cholesterol is much harder to be degraded by mammalian cells comparing with glucose. Luckily, the cholesterol export machineries, ABCG5 and ABCG8, have been identified. ABCG5 and G8 from functional heterodimers and located on the apical membrane of hepatocytes and intestinal enterocytes. ABCG5 and G8 transport cholesterol and plant sterols to bile and intestinal lumen. Then cholesterol is removed through feces. These two genes were first found in 2000 by Hannah Hobbs' lab. Mutation of ABCG5 or G8 cause cytosterolemia because they also pump plant sterols besides cholesterol. The cytosterolemia patients show much higher plant sterols and cholesterol in their blood. They have early onset cardiovascular disease. ABCG5 and G8 genes located in the same locus of chromatin and share the same promoter region. They are transcriptionally activated by LXR. Transgenic overexpression ABCG5 and G8 lowers the lipid levels in blood and liver because the cholesterol is excreted to bile and feces. The LXR agonist T compound can also upregulate ABCG5 and G8. The total cholesterol level in liver was reduced. However, the triglyceride level in liver and blood were dramatically increased because LXR also activates SIBP1C and then fatty acid biosynthesis was increased. Thus, the pan-LXR agonists have been abandoned by pharmaceutical companies. In 2016, New England Journal of Medicine reported a paper showing that loss of function and mutations of ASGR1 were strongly associated with a lower cholesterol level and a lower risk of coronary artery disease in Finland populations. And this association has been validated in other populations. So what's ASGR1? ASGR1 is the major subunit of acetylglycoprotein receptor. Most of blood proteins contain sialic acids at the terminal of its glycans. Removal of sialic acid exposes galactosis and accelerates the protein turnover because ASGR1 can bind to galactose with high affinity and then internalize the glycoprotein, deliver them to lysosomes for degradation. ASGR1 is highly expressed in hepatocytes. At first, people thought ASGR1 affects the cholesterol level through controlling the LDL receptor protein turnover because LDL receptor is a glycoprotein. However, our results show that ASGR1 does not directly affect LXR protein level or LDL receptor clearance or the HNE co-reductive degradation or SIBP cleavage. So what's the mechanism? We then performed an RNA-seq analysis on wild-type and ASGR1 knockdown cells. Surprisingly, the mRNAs of ABCG5, ABCG8, and ABCA1 were dramatically increased. These genes are all LXR targets. Western blot showed that the protein level of LXR alpha was elevated in the ASGR1 knockout cells, and LXR alpha is the major isoform of LXR in liver. ASGR1 is highly expressed in liver. Knockout of ASGR1 reduced the lipid content in liver, and the total cholesterol level in serum, triglyceride in serum were all reduced in the ASGR1 deficient mice. In ASGR1 knockout mouse liver, the protein levels of LXR alpha, ABCG8, ABCA1, CYP7A1 were increased. SRABP was decreased and FASN was decreased. This is very interesting. I will come back later. So consistent with the elevated expression of ABCG5 and G8, the volume of gallbladder was increased by almost 50% in ASGR1 KO mice. Biliary cholesterol concentration was increased by 50% too, and the total biliary cholesterol was increased by about one-fold in the homozygous ASGR1 knockout mice. The fecal cholesterol was increased by about 50%. The phenotype was so dramatic that you can see the bile became clouded due to higher biliary cholesterol concentration. Can we use this mechanism to treat metabolic disease? We knock down ASGR1 by shRNA in LDL receptor knockout mice. The blood cholesterol and triglyceride were decreased, and the biliary cholesterol was increased in the ASGR1 knockdown mice. ASGR1 knockdown dramatically prevented atherosclerosis. These experiments were done in the LDL receptor knockout mice, further confirming that the lipid lowering effects of ASGR1 is independent of LDL receptor. Next, we generated an anti-ASGR1 neutralizing antibody, 4B9, and the 4B9 injected mice showed a higher LXR alpha, phospho-ACC indicating activation of AMPK, higher ABCG8, and higher CYP7A1. The expression of NSRABP was decreased. And in ASGR1 knockout mice, the 4B9 antibody had no effect, confirming they are on target. 4B9 dramatically decreased total cholesterol level in serum by about 50%, decreased the triglyceride level, or increased the total biliary cholesterol in the treated wild-type animal, and increased the fecal cholesterol. 4B9 have no effect on knockout mice. Notably, the neutralizing antibody 4B9 displays synergistic lipid lowering effect with atorvastatin or ezetimibe because they work on different cholesterol metabolic pathways. 4B9 promote ABCG5G8 to excrete cholesterol, and statin inhibit cholesterol biosynthesis. Ezetimibe block MPC1L1 mediated intestinal cholesterol absorption. So why is LXR activated but SRABP inhibited in ASGR1 deficient mice? The mechanism is a little bit complex, and so I show this model in the right panel. ASGR1 binds blood glycoproteins and internalize them or deliver them to lysosome for degradation. Then after degradation, the glycoprotein can release amino acids. The amino acids in lysosome can activate mTOR1 and then inhibit AMPK. In the ASGR1 deficient or knockdown or knockout cells, the glycoprotein internalization was decreased. So the lysosomal amino acids was decreased. Therefore, AMPK was activated and mTOR was inhibited, which caused the decrease of BRCA1 and BARD1. And these two proteins form a E3 complex that can degrade IOXR, which has been reported by others. So finally, in the deficiency of ASGR1, IOXR protein level increased, and the target gene ABCG5 and G8 and ABCA1 are increased. So cholesterol excretion and to Bile is increased, and the HDL cholesterol concentration was also increased, which is beneficial too. And AMPK activation can block SIBP, which has been confirmed by, has been reported by in many studies, and therefore the lipogenesis was decreased. So I here show you some key results. First, the lysosomal amino acids was increased when the wild type cells were treated with acetylphytolin A, a ligand of ASGR1. But the ASGR1 deficient cells were insensitive to this treatment. Acetylphytolin A activated mTOR1 in wild type cells. That depends on the lysosomal amino acid transporter SLC38A9. And BRCA1 and BRD1 are E3 complex, can ubiquitinate LXR. Acetylphytolin A treatment can increase the protein level of BRCA1 and BRD1, and therefore decrease LXR alpha. Knockdown BRCA1 or BRD1 and prevented glycoprotein induced LXR degradation. On the other hand, ASGR1 deficiency decreased the BRCA1 and BRD1 proteins, and then increased the LXR alpha protein. The phospho-ACC was increased, indicating AMPK was activated, and thus the SIBP was decreased. So here I summarize our work. In addition to micropinocytosis and autophagy, we found that the ASGR1 mediated acetylglycoprotein uptake and lysosomal degradation is a major source of lysosomal amino acids that can regulate mTOR1 and AMPK in liver. Inhibiting ASGR1 activates AMPK and block mTOR, causing the LXR elevation and SIBP inhibition. So inhibiting ASGR1 and stimulate cholesterol excretion and to bile, and then cholesterol is removed through feces. And ASGR1 inhibitors display synergistic lipid lowering effects with statin or ezetimibe. And this study suggests a new way to liver specifically activate AMPK and increase specific LXR target genes. AMPK has been considered to be a drug target for a very long time. However, a non-selective activation of AMPK can be problematic. Shown left is a paper from our group. Together with Dr. Yan, and we found a cardiac hypertrophy patient family and identified the disease-causing mutation in PRKD2 gene. This gene encodes for gamma-2 subunit of AMPK, and the mutation causes constitutive activation of AMPK and results glycogen accumulation in myocytes. The patient develops cardiac hypertrophy and die early in teenage. So very similarly, Merck has developed a potent AMPK agonist, which improves glucose metabolism in mice and monkey. However, it causes cardiac hypertrophy due to activating cardiac AMPK. So it's impossible to use pan-AMPK activator to treat metabolic diseases. And our finding on ASGR1 provides a new way to liver specifically activate AMPK and increase the specific IOXR target genes. This story was done by a talented student, Wang Jiqun, with the help from other members. And I thank our collaborators and the funding agents. I will stop here and I'd like to answer any questions. Thank you.
Thank you so much, Dr. Song, for that wonderful talk. Even though that was a recorded presentation, Dr. Song is here to answer questions live, so you can send your questions into the Q&A box. Because we're running behind schedule, we may only get to a couple of them, but you can feel free to send your questions in, and the speakers are actually going to try and answer them as their schedule permits.
May I ask a question? If I recall correctly, there is something like about 4 or 5 million molecules of this receptor on the sinusoidal surface of every hepatocyte. And it allegedly, when it was first discovered and then later, is believed to be the way in which decylated proteins, particularly coagulation proteins, are regulated in their disappearance from blood. So I was surprised to see in your slide, you indicate that knockouts have no phenotype, but I presume that's a physical phenotype. But what happens to all those decylated glycoproteins and the coagulation factors when you inhibit the receptor?
Yeah, so that's a very good question. Yeah, as you mentioned, the ASGR1 was first identified to remove some glycoprotein in blood. However, the ASGR1 and knockout animal showed a normal turnover of endogenous deacyloglycoproteins, but the injected exogenous glycoprotein turnover was impaired. And it was believed that some other glycoprotein receptor can compensate the function of ASGR1 in mice. Therefore, the endogenous blood glycoprotein turnover was seems normal. And what we found is actually the ASGR1 deficient hepatocyte is in a relative starvation state. So the glycoprotein internalization was decreased, and the lysosomal amino acids level was low. So the AMPK can be activated. So
Very interesting.
Thank you.
And Our final question before we move on to the next talk is, do other endocytic cargoes also change lysosomal amino acid content to alter mTOR and AMPK? What is special about the ASGR1 ligands?
Well, ASGR1 has many ligands, so the ligands are not very specific. And ASGR1 is highly expressed in hepatocyte, so that can cause liver specific activation of the pathway.
Awesome. Thank you so much, Dr. Song, for such a wonderful talk. And we will go on to the next presentation.
Thank you.
So it is my pleasure to introduce Dr. Heather Francis. The goal of Dr. Francis' laboratory is to investigate the synergistic role that cholangiocytes and mast cells play during cholestatic liver injury, cholangiocarcinoma, and non-alcoholic fatty liver disease. Her lab is currently funded by both the NIH and the VA, and these studies examine the link between cholangiocytes, mast cells, and hepatic stellate cells during liver disease, focusing primarily on primary sclerosing cholangitis and non-alcoholic fatty liver disease. Her expertise also extends to cholangiocyte pathophysiology and paracrine cellular signaling, including crosstalk with hepatic stellate cells and Kupffer cells. Her topic for today is regulation of chronic liver disease and cholangiopathies by mast cells. So thank you, Dr. Heather Francis.
Thank you for that introduction. And I want to start by saying that I'm really very honored to be here. I want to thank the American Liver Foundation, Wolfram, Sangeeta, Heather Fleming, and all of those involved that were able to get me to this place. Because for me, this is quite an honor to be part of this very prestigious event. And so today I am going to share sort of like a little bit of an overview of some published and unpublished data that we've been working on in my lab about over the last 10 years or so. And I'll start with just a little outline, some background. I want to talk to you about how we've demonstrated that mast cells are very prominent in promoting cholestatic liver injury, that we can block mast cell signaling and also histamine itself to demonstrate some therapeutics. And then I was going to follow this, since this is such a timely topic with the prevalence of fatty liver growing right now, I want to also discuss some of the data we have on that. So my lab primarily focuses on cholangiopathies, and the main focus of my work is primary sclerosing cholangitis, or PBC, but we're also doing some projects on PBC, and I'll show you some of that data as well. We know that these two cholangiopathies can both progress into liver cirrhosis. The PSC targets primarily the large bile ducts, where PBC seems to really implicate the small bile ducts during the disease progression. PSC patients typically have a high rate of inflammatory bowel disease, or IBD, and this of course then contributes to poor quality of life. Within PSC, we find biliary fibrosis, or onion skin fibrosis. This is typically male dominant, where PBC is predominantly found in females. So like I said, my lab has been studying the role of mast cells in liver diseases for the last 10 years or so. And we know that these are circulating immune cells that can migrate to different tissues and organs through multiple signaling pathways. And once they're degranulated and open up, mast cells then release just a myriad of mediators that can be preformed or newly formed. And one of the prime mediators that my lab is interested is in histamine, which is formed by the conversion of the ends of the amino acid histidine by L-histidine decarboxylase, or HDC. So we've shown that mast cell number really increases and is enhanced during human liver disease. And so when you look at a controlled human liver, you might find 1 or 2 resident hepatic mast cells lying in close proximity to bile ducts. However, when we look in patients with PSC or PSC IBD or PBC, we do find this increased infiltration of mast cells that seem to still surround primarily the bile ducts in all of these diseases. And in fact, when we co-stain for CK19 and tryptase, which is a marker of hepatic mast cells, we can see a nice infiltration here in our human PSC when we compare these to the controls, suggesting that there's some sort of initiation or event that occurs that drives mast cells to want to enter the liver during damage. And what we've primarily demonstrated is the role that they play there is quite inflammatory. We've also looked in models of cholestatic liver injury in both rats and mice. And in an early study we did, we wanted to try to understand at what time point mast cells might be migrating into the liver. So we took rats and we performed bile duct ligation, and we sacrificed them from 6 hours all the way out to day 14. And if you look around day 3, we start to see a little bit of hyperplasia or ductal reaction, where we're starting to see more and more bile ducts being formed, presumably part of the reparative process that occurs during bile duct ligation. The problem is that also during this time, we start to see this influx of the mast cells that are marked here by these red arrows. And this just continues out and peaks around day 7, but then continues out even through day 14, when sometimes after a long bile duct ligation, you even start to see reversal where the liver is able to repair itself. So we're unsure about the events that are happening out this far. And again, we were able to show that in a rat, the mast cells do infiltrate and they stay close by bile ducts, shown here by immunohistochemistry and fluorescence. So we started using a mouse model. These are KIT SASH mutated mice. And so they present without any mast cells. And so we're able to use these mice and manipulate them in the lab to be able to understand if the depletion of mast cells actually can reverse disease phenotypes. And in fact, we found in an early paper when we did bile duct ligation in these mice, of course, in the wild type, we have an increase in hepatic fibrosis and we have an increase in ductal reaction. But when we did the same surgery in the KIT mice that were lacking mast cells, we were able to show histamine secretion was significantly lower, fibrosis was down, as was ductal reaction. And all of this demonstrates to us that mast cells do in fact promote biliary damage and hepatic fibrosis. So in this first part, I just want to talk about mast cell injection. What happens if we put mast cells back into mice that typically either don't have any or only have a few? And so using either wild type mice or the KIT SASH mutated mouse lacking mast cells, if we take labeled mouse mast cells, and these are fetal hepatic derived mouse cells, and so we don't have any issues putting these back into mice, and we inject them via tail vein just one time. And after 3 days, by fluorescence, we're able to find these labeled mast cells that are marked by the yellow arrows, and you can probably see better here in the zoom box. And again, we find them in close proximity to these bile ducts that are stained in fluorescent green. Well, what happens to the phenotype of these mice when we do this? So we published a couple of papers where we've demonstrated that if we inject wild type or KIT with the mast cells, this is one injection sacrificed 3 days later, we're able to increase serum ALP levels both significantly, and we are able to also promote ductal reaction in these mice. And again, we see a significant increase in both of our models here. Looking further, we looked at also some inflammation, and we see here that there's an increase in the number of F480 positive cells, so the macrophages within the liver, either in our wild type or in our KIT mouse injected with mast cells. And we also see changes in some of the interleukins that are very important in inflammatory processes, including IL-1 beta and IL-33. And both of these factors are significantly upregulated after just one injection of mast cells via tail vein. We see an increase in hepatic fibrosis, and we also see an impact on hepatic stellate cells in that we're able to activate a population of these shown here by Desmond staining that's increased following injection of mast cells. Biliary senescence is also a key regulator and a marker of cholangiopathies, both PSC and PBC. And again, in our mice that we inject with mast cells, we're actually able to also promote biliary senescence in this population of bile ducts. So what about inflammatory bowel disease? So we talked about this a minute ago, that PSC patients do present, a large portion of them present with IBD. And in a study that we published last year, we were also able to show that patients with PSC and PSC IBD have an increase in their serum levels of not only histamine, but also of FGF19, which we know is an important player in regulating bile acid, both intrahepatic and colohepatic shunting within the liver. And in this paper specifically, we were interested in FXR. And so we were able to show that these patients do have an increase in both of these factors. But what happened when we injected mast cells into these mice? This was the first time that we'd looked in the intestine. And what we found, in fact, was that both FXR and FGF were increased in the ileum of KIT mice after a single injection of mast cells. And we were also able to increase some of the inflammatory mediators, such as stem cell factor, IL-1 beta again, TGF beta, and CCL3. And so this was really interesting to us that we're able to not only implicate the liver when we inject mast cells via tailbane, but they're also having an effect on the intestine. And this could also be a new model where we might be able to study intestinal inflammation and the role that mast cells play in that. We also looked at some tight junction proteins and stained for ZO1. And if you look here in our wild type mice, we have a nice pattern of ZO1 expression. But when we injected the wild type mice with mast cells via tailbane, we do see a disruption in the expression of ZO1. So this is suggestive that we might be also interrupting tight junctions and then impacting gut-liver crosstalk. And these are some studies that we're currently doing in my lab. So we wanted to take this a little bit further and think about the mediators that are inside of mast cells when we inject them into the mice. And the thought was, what happens if we sort of manipulate some of them? And so we used a number of mediators to try to see what implications we would have. So we used an FXR inhibitor, we've used an NF-kappa beta inhibitor, we've used a TGF beta receptor inhibitor, and an H2 histamine receptor inhibitor. And in fact, when we treat these cells prior to injection using different mice this time, not only the KIT and the wild type, but also an HDC knockout mouse, we're able to show that if we inject with the mast cells, as I showed you, we do have an increase in inflammation. But when we blocked FXR signaling within mast cells using glucosterone, you can see here that there's actually a pretty dramatic decrease in our F480 staining. If we knock out TGF beta or inhibit TGF beta, we can decrease the mast cell induced fibrosis and ductal reaction. And in new studies using the caffeic acid drug, which is an NF-kappa B inhibitor, we're able to also decrease hepatic stellate cell activation. So these studies demonstrate that we can manipulate mast cells prior to putting them into the mice to study this pseudo model of cholestasis, and allows us to understand what mediators might be involved in mast cell damage. And then we also looked again in the intestine in this same study that we published last year, where we treated these mast cells with FXR inhibitor. And even in the intestine, we were able to decrease FXR and FGF immunoreactivity, but also the intestinal inflammation that we drove when we injected the mast cells. So all of our markers were downregulated when we injected these KIT mice with mast cells that were lacking FXR. So in this first part, what I hope I showed you is that the single injection of mast cells can really promote cholestatic liver injury, but also intestinal inflammation. And if we block or mediate mast cell mediators like TGF beta or the histamine, the H2 receptor, we can reverse PSC phenotypes, and we can also impact intestinal FXR, FGF signaling. In that paper, we looked very closely at bile acids as well. And so our thought is here that mast cell injection and manipulation would be a really unique method for us to be able to study cholestatic phenotypes. So in the next part, I want to talk about how we can target mast cells, histamine signaling in both PSC and PBC models. We did some early studies where we used MDR2 knockout mouse, which is a very prominent model of PSC that is widely used. And we use this old drug, cromolyn sodium, which is an asthma drug that is typically used by inhaling to reduce inflammation within the lungs. And so we thought, well, what would happen if we put this into our mice, since it's supposed to be a mast cell stabilizer? So these studies were done by mini pump into the MDR2 mice for up to a week. And what we find is a very dramatic decrease in hepatic fibrosis, shown here by fast green serous red staining, also by Mason's trichrome. And of course, we also decreased ductal reaction, shown by CK19 staining, insinuating that this drug may have a new purpose for PSC patients. Also, interestingly, looking at physiology a little bit, we looked at some bile flow, and when we treated these mice with cromolyn, they did have a significant decrease in bile flow when we compared these back to our MDR. The bicarbonate levels were also down, and we saw changes in serum and total liver total bile acids when we treated with cromolyn sodium. So this suggests to us that mast cells and mast cell mediators like histamine not only can impact the pathology of PSC phenotypes, but also potentially some of this physiology that's involved in driving PSC progression. Next, we did a study where we looked at using just normal antihistamines. And so we know that everybody takes a lot of antihistamines, sometimes in the morning for allergies, sometimes late in the evening. If you ate something you shouldn't have, drank a little bit too much, you might take an H2 blocker. So we took, again, and in this study actually too, we also looked at cholangiocarcinoma. So first we wanted to see if these receptors are upregulated. So in both human PSC and in human CCA, we do see an upregulation of H1 and H2 receptors. And we took our MDR mice again, and we treated them with an H1 or an H2 receptor antagonist. In this study, we actually treated for a month by mini pump. And we were able to show, again, a nice decrease in ductal reaction, a very nice decrease in hepatic fibrosis as well. What was also interesting to us is we used a CCA model of nude mice xenograft tumors. And when we implanted these tumors into the hind flanks of mice, you can see here that the vehicle, really took off, and there was a strong tumorigenesis here. But when we treated mice every other day with our antagonist, these tumors really never had a chance to really take off. And so we really saw an ablation here of tumor growth, and also among other phenotypes, but just demonstrating here, angiogenesis was also significantly reduced in our mice after treatment with H1 and H2, again, representing a potential therapeutic option for patients with PSC and PSC-CCA. So what about PBC? These are some studies that we've done that are unpublished at the time, done by Lindsay Kennedy, who was a postdoc in my lab and now is an assistant research professor at IU. And she took a PBC model, which is the dominant negative TGF-beta receptor 2 at 34 weeks, which represents a late stage of PBC. And she treated these mice with cromolyn sodium to try to understand these phenotypes as well. And in fact, we did find in data I didn't show here that there's an influx of hepatic mast cells that come into the PBC mice. And when she treated them with cromolyn, we have a decrease in hepatic damage. We have a decrease in histamine levels. Really, more importantly, though, she found a decrease in serum AMA levels. So again, demonstrating that this drug might have a very positive impact on this patient population that present with these high serum AMAs. We also found a decrease in hepatic fibrosis and HSC activation, and she found a decrease in serum TGF-beta1 levels that were significantly increased in her 34-week-old mice. So in this part, what I've shown you is that we've been able to show that if we can inhibit mast cell mediators like cromolyn or histamine using antihistamines, we can ameliorate some of these PBC and PSC phenotypes that we see. And this really offers a novel therapeutic option using old approved drugs that are readily available. One problem with the cromolyn sodium is it is an inhaled drug. And so this one is a little more challenging in our mice since we did do this by mini pump. However, we are working with a company now to develop small molecule inhibitors that might act like chromalin sodium to be able to reduce some of these mast cell mediators. So in the last part, I do want to just touch a little bit on fatty liver disease. Even though my title was all about cholangiopathies, my lab does have an interest in studying fatty liver disease. And we showed years ago that in NAFLD, NASH, and in an end-stage livers serum, we were able to show an increase significant in the serum histamine levels in these patients. And there was also an increase in the enzyme and in all of the receptors. And these were all upregulated in the NASH patients when we compared these to the control. But what about human patients? In fact, taking NAFLD or NASH patients, we're able to show again by tryptase staining that there are a large number of hepatic mast cells that infiltrate these livers. Now, these are going to be later stage explanted livers, but if you look in the NASH patients, you can really see a very predominant infiltration of hepatic mast cells. We saw an upregulation in the NASH patients of all of the mast cell markers that we looked at, including c-KIT, chymase, and tryptase. In our mouse model, we took mice starting at 4 weeks. We fed them Western diet for 16, sacrificing at 20. And again, we see an infiltration of mast cells. And more importantly, their serum histamine levels are quite increased in the mice that have been fed this Western diet. In this study, we also then decided to take the KIT mice. So these are the mast cell deficient mice, and we fed them as well with the Western diet, which is high cholesterol, high fructose corn syrup equivalent, and saturated fat. And really, we found very, very dramatic effects. And so if we look at some of the phenotypes of NAFLD, we see a just almost complete reversal of hepatic fibrosis. We see a large decrease in inflammatory cells, the macrophages specifically. And most importantly, we found in these mice that were fed Western diet lacking mast cells that the steatosis was almost completely ablated as well. This was a really big study, again, from Lindsay in my lab, where she was able to demonstrate that when we also re-injected these mice with mast cells, we were able to further drive the steatosis back to the normal level, again, suggesting this prominent role for mast cells in this disease progression. So what about chromalin sodium? We went back to that drug using methionine choline deficient diet as another model of NASH. We treated these mice for 2 weeks during that last feeding. And I think we showed pretty nicely here, we have the MCD steatosis shown by H&E and oil red O. And when these mice were treated with chromalin sodium, we do see a reduction in this. This is work being done right now by my graduate student, Deb Jody, who is also doing this, repeating these experiments in high-fat diet mice, Western diet, to see if we see similar effects. But in our chromalin MCD, we do see a nice reduction in hepatic fibrosis that's significant. And of course, ductular reaction is also decreased when these mice were treated with chromalin sodium. Well, what about alcohol? So in a very newly started project in my lab that was just funded by AAA with an R21, we know that alcohol exposure can actually affect mast cell physiology very directly. So if you put alcohol or ethanol on mast cells in culture, you can induce activation and differentiation, degranulation, they can migrate, et cetera. So there's really no question that mast cells play a role in this inflammatory process in alcohol and contribute to different factors in this microenvironment. And in fact, when we took end-stage explanted ALD samples and stained them again for tryptase, you can see here again in the pink that there are just a number of hepatic mast cells that have infiltrated, and we do see an increase in the serum histamine levels in patients with ALD. So we took some mice again, the wild type or the mast cell deficient mouse, and we did the Bin-Gal model for 8 weeks total, but we also did a more acute model where we do the 10-day ethanol plus just one binge. And if we look in those mice that were fed ethanol for 10 days, we do see again, the mast cells are prominently found in close proximity to bile ducts. And we find serum histamine goes up in our mice that were fed the chronic binge ethanol diet. In our mice that are mast cell deficient, we do see a reduction in steatosis and in inflammation. These are preliminary studies, and so we're doing more, and we're quantifying these to ensure that what we're seeing is statistically significant. But just eyeballing these, it does look quite dramatic. How is this happening? In this project, we think that this is really being driven by cholangiocytes that begin to express P16 when they're damaged and secrete senescence-associated secretory phenotypes like TGF-beta and stem cell factor, which happens to also be something that mast cells will migrate towards. In fact, we've shown before that if we knock out that in human PSC, there's a significant increase in stem cell factor expression and in the serum. So in our mice, in our human samples, we looked at stem cell factor staining, and we do see an upregulation of this in ALD patients when we compare them to the humans. But really what was interesting is that if we look at cholangiocyte expression, stem cell factor is significantly increased, where in hepatocytes, it's not increased. So this suggests to us that what we might have is a stem cell factor cholangiocyte specific event drawing c-KIT positive mast cells into the microenvironment to increase ductal reaction, steatosis, and all of these phenotypes. So in summary of this last part, what I've shown you is that mast cells are upregulated in fatty liver disease. NAFLD phenotypes were prevented in mast cell deficient mice that were fed Western diet. If we use the drug chromalin sodium in MCD mice, we're able to also reduce those phenotypes. And in our mast cell deficient mice that were treated with ethanol, we see a reversal of these phenotypes that are associated with alcohol-induced liver damage. So just an overall summary, and this was a review that we published last year in Hepatology. Mast cells really are playing a central role in a number of diseases. And I talked to you mostly about ALD, NAFLD, PBC, and PSC, and a little bit of our CCA work. We're starting to do some HCC work. We're working on a paper now. But what we found over the years is that histamine specifically, and then other mediators are key regulators of all of these different types of hepatic damage. And if we use different therapeutics, including approved drugs or small molecules, we are able to ameliorate some of these diseases, demonstrating potential new uses for these old drugs that we maybe haven't thought about. So in closing, I want to thank all the members of my lab, including Vic Meadows, who did a lot of the work on the FXR that I showed you, who was a graduate student and is now at Rutgers. I have to thank Franco Alpini, Dr. Exer, Dr. Kennedy, also for all of their assistance. Last week, I can't think of any other way to follow this that could be any better, because most of us just came home from AASLD, where we had a fantastic meeting in person, getting to see everyone. And one of the highlights was getting to meet Dr. Arias at the ALF awards presentation. And so with that, I think I'm finished, and I'm happy to take any questions.
Thank you, Dr. Francis, for that really impressive talk. We have some questions and comments in the QA section. This is from Sura Bidinger. I have very much enjoyed following your work over the years and the important contributions to understanding lipid metabolism. It was a pleasure to hear you talk. I think that was just a comment. From Wending, a question. Do you think histamine also affects ductal reactions in alcohol liver disease?
Yeah, I do think that this is a really good question. And, we do see that increase in histamine levels in serum from patients with ALD, but we haven't actually manipulated the histamine yet in models of ALD. But I won't be surprised to see that if we give, even if we gave histamine to wild type mice fed ethanol, I do think that we're going to see an increase in ductal reaction. Similarly, we can take our HDC knockout mice and feed them ethanol. And I would assume that the damage there would be reversed. Whether it's a direct effect or not, I'm unsure. We have not treated we've treated cells in culture with ethanol, but we haven't looked at histamine levels, and we haven't treated cells with histamine to look to see if we can induce ALD phenotypes. But it's an excellent question.
Thank you. I think
That's a question. So it is very exciting work. I have a question for you. So if I recall correctly, mast cells secrete enormous amounts of polyamines, serotonin, in addition to histamine.
Yes.
Do these other compounds have anything to do with the processes that you've been describing?
Yeah, I mean, that's an excellent question, Dr. Arias. I get this a lot because I've had a little bit of tunnel vision only looking at histamine over the years. And I think that you're exactly right, especially factors like serotonin and other growth factors too, VEGF, FGF. And what we see when we inject mast cells per se, and we knock things out inside of them, is we still manipulate histamine levels. We're still able to show in mice that the histamine levels are changed. So whether or not it's histamine only that's altering things, probably not. But I think that some of these other factors, the big implication on, let's say, inflammation that might be driven by histamine is obviously impacted. And, we've started other studies now where we're looking at interleukin 33. We know mast cells make interleukin 33. So we're doing studies where we're knocking that out to see if we can also ablate some of these effects.
Great.
Thank you for the question.
Okay, thank you so much for a wonderful talk, and we're going to go to our next presentation. I'd like to introduce Joanna Krawcyzk. She is a graduate from the University of Pennsylvania with a bachelor's degree in biology and nutritional sciences and a PhD candidate at the Harvard Chan School of Public Health. Joanna is in the lab of Dr. Sudha Bittinger in the endocrinology department at Boston Children's Hospital, studying the intersection of insulin resistance and non-alcoholic fatty liver disease and non-alcoholic steatohepatitis. Joanna's talk will focus on insulin regulates bile acid metabolism through zone-specific transcriptional programs. And I'll hand it over to Joanna.
Perfect. Thank you so much. Thank you for the kind introduction and for the opportunity to talk today. As mentioned, I'm Joanna Krawcyzk, and hopefully today I can convince you that protein localization can be more important than abundance with the talk of insulin regulation of bile acid metabolism. So just to start, bile acid synthesis and bile acid metabolism as a whole are really an important pathway in whole body regulation of lipid metabolism. And that's because bile acids are derived from cholesterol precursors within the liver to regulate nutrient absorption as well as signaling. Now, the bile acids are encompass two main classes, which differ in the presence of a hydroxylated site at the 12th carbon, making 12 hydroxylated bile acids and non-12 alpha hydroxylated bile acids. And the production of these bile acids differs solely in the expression of one enzyme, CYP8B1. So CYP8B1 is the sole enzyme that catalyzes the production of 12 alpha hydroxylated bile acids. And just important to note that all of the other enzymes are conserved across the production of these two subspecies of bile acids. So CYP8B1 is the true determinant of 12-HBA production. And what's known about insulin regulation of bile acids is that when we have periods of insulin resistance, perhaps in type 2 diabetes, we get robust induction of the production of 12-HBAs that really robustly correlates with metabolic dysfunction. This has been shown to lead with worse NAFLD and NASH outcomes, as well as hypercholesterolemia and hyperlipidemia. However, when we look in mice at the levels of CYP8B1 and 12-HBA with insulin manipulation, there's actually a fairly weak correlation of CYP8B1 and 12-HBA, which is surprising because we'd expect, as the sole enzymatic determinant of 12-HBA production, CYP8B1 levels would correlate almost linearly with 12-HBA levels. So we decided to do single cell sequencing using insulin manipulation to look at if perhaps CYP8B1 localization is changing in the liver. And we found that most bile acid synthesis genes are pericentrally zonated and that this does not change with insulin manipulation. However, CYP8B1 specifically is getting shifted out of pericentral hepatocytes in the presence of insulin signaling, separating it out from the other bile acid synthesis genes. And this was true at the transcript level in the single cell, as well as the protein level in situ. So we therefore wanted to ask the question of, is pericentral CYP8B1 sufficient to alter 12-HBA levels? In order to do this, we generated pericentral specific CYP8B1 knockout mice. This was done in collaboration with Dr. Nai-Yang Fu. So this is an LGR5 TET-dependent model where we get CYP8B1 recombination and deletion only in the hepatocytes surrounding the central vein, here outlined by the white dotted line. And when we got pericentral CYP1 deletion, this only hit about 5 to 10% of the total hepatocytes in the liver. So this did not correlate with whole liver levels of CYP1 protein or mRNA. However, this small change in total levels but big change in zonation was sufficient to correlate with a significant decrease in the gallbladder pools, decreasing the proportion of 12 HbAs. So this is suggesting that insulin can alter metabolism simply by changing where a gene is expressed, and hopefully highlighting the need to look at liver histology when thinking about some of these metabolic pathways in the liver. So thank you very much.
Thank you very much, Joanna, for that wonderful talk. That concludes session 1 for now. Thank you all to the attendees for sticking with us through the digital challenges this morning. I know we're running behind schedule, but I think everybody should have the right Zoom link now in their emails. We were just sent out to every attendee. So now we just want to take 5 minutes to stretch, maybe get some coffee and tea. We should be able to get right back on schedule in 5 minutes. If you have any burning questions that we couldn't get to live, I apologize, but feel free to send it in the Q&A box because the speakers are going to keep going through the questions and are able to type in replies to you. All right, so everyone can enjoy your break.
Okay, I think we've had 5 minutes. So shall we move on to session 2?
Yes.
Okay, perfect. So my name is Epithelial. I'm going to be chairing session 2. We have 3 half-hour talks and then 1 micro talk at the end. So hopefully we can catch up a little bit on time. So the first speaker for today will be Stacey Huppert from, well, currently Cincinnati's Children's Hospital. So she's trained at Indiana University and after that a postdoc at WashU. And for the last over 20 years, she has been working on hepatic development using, amongst other things, Alagille syndrome as a disease model. And today she's hopefully also going to talk on that and probably many other things. The title of her talk is Hepatic Epithelial Development and Plasticity, Identity and Commitment. So please go ahead, Dr. Huppert.
Thank you very much for that introduction. I just want to echo the other speakers thus far. This is such an honor to have the chance to go through our research on this platform. Thank you to the American Liver Foundation and also having this symposium in honor of Dr. Arias. And thank you to the organizers, Dr. Bhatia, Gosling, and Fleming. Our work, as already introduced to you, we've been interested in hepatic epithelial plasticity and also bile duct development, as this restoring the biliary architecture is really a clinical challenge. The standard of care is really focused on alleviating symptoms of cholestasis without steps towards bile duct augmentation. Our group stands on the shoulders of many leaders in the field of liver research and regeneration, just a few listed towards the left bottom of the slide. And now I'm sure everyone can appreciate here at this symposium how plasticity has really become an area of work, a broad area of work within the liver and regeneration. The idea being, I'm just summarizing very briefly in this slide of hepatocytes and cholangiocytes, the epithelial cells of the liver, being able to switch identities or give rise to each other. And this is in the phase of this elusive true stem cell within the liver. So the idea that hepatocytes can become cholangiocytes is work from our group and Holger Willenbring's group, where we show that there's very little proliferation as these identities change from hepatocytes to cholangiocytes. And the other group that kind of led the charge of cholangiocytes becoming hepatocytes was really led by Stuart Forbes. And it appears that this transition goes through a proliferative mechanism, as we know that cholangiocytes are not a very proliferative population, and then transition into hepatocytes. So what we know at this point is that there's really these dual reparative programs in the absence of a hepatic stem cell that's held in reserve from development. If the 2 epithelial cell types, hepatocytes or cholangiocytes, are unable to self-renew and proliferate in the face of injury, that there is this adaptive reprogramming mechanism. It's obviously dependent on the cellular compartment that's injured and the severity of the compartment. I do want to point out that there are many single cell papers now that have been published that have identified cell states with a hybrid signature that's in between hepatocyte and cholangiocyte in normal liver. And the question is now are those cells cycling in their transcriptional signature, or is part of this due to a signature due to dissociation techniques? So our group early on had been working, as you got it, from Alagille syndrome and this idea of paucity, but we really needed to build models that we could look at providing selectivity in this really ductopenic or truncated biliary system to understand mechanisms of repair and reprogramming. So our initial models we built with just looking at the Notch pathways. We know patients with Alagille syndrome, a majority have haploinsufficiency in the ligand JAG1, and a few families have mutation and are haplosufficient for Notch2. But you can see if we truncate or delete the system using RBP RBPJ, which is the common DNA binding partner for all 4 Notch receptors, you see that you get a truncated biliary system. However, these animals are not that sick overall. And if we do the opposite, where we activate Notch using a constitutive intracellular domain of the receptor, you can see that we get many connected and communicating bile duct system. So a review that was published a few years now, where we were looking at the different signaling pathways involved in bile duct development. And this really was the frame of our work, which was knocking out both RBPJ, as I show here, in addition to HNF6, which was studied by Frederick Lemegre's group being involved in bile duct development. Could we make a system that's very truncated and therefore selective? So I'm just highlighting here published work where we looked at, could show that you take a very truncated system in the background of the deletion of these 2 factors, and you can see that de novo generation of a bile duct system. What is interesting is that these cells have not escaped deletion, so they're still deficient for both RBP and for HNF6, and these bile ducts are stable once the cholestasis resolves in these animals. And we show that not only does the bile duct architecture resolve, but also the fibrosis and the serum chemistry. So what we did with Holger Willenbring's group was to understand where these cells are coming from, if they are not coming from that population of deleted cells, or they are coming from a population of deleted cells, which suggests that they're already coming from an albumin-crete lineage, right, to provide. So at postnatal day 17, we lineage labeled the hepatocytes using a flipase, and then we analyzed the liver later with the hypothesis being that if they're coming from hepatocytes, they're building these new biliary branches, that they will be dual labeled. And that you can see here where you have GFP lineage labeled hepatocytes that give rise to double labeled cholangiocytes that are labeled here with wide spectrum cytokeratin. What the liver does amazingly is that as these hepatocytes are transitioning into cholangiocytes, you see a clone over here that's hepatocyte labeled, and that they find each other in space and build a tube. And not only do they find each other in space, but they connect into these trunks that are the primary trunks of the biliary system in order to drain bile. So that is just the first part that I just wanted to give you an overview high level, which is that hepatocyte plasticity and transdifferentiation has extensive tissue repair capability and the ability to build a bile duct system. So next I want to go a little bit deeper, and these are just snippets and observations that we've had in directions that we're going. So one knowledge gap that we've had for a while to really understand these mechanisms that are guiding biliary development and hepatocyte plasticity is, why are some hepatocytes resistant to Notch activation? We know that Notch is a very strong driver in order for hepatocytes to become cholangiocytes. We just showed you in those earlier models that hepatocytes are able to do it in the absence of Notch. But if we drive Notch everywhere, you can see that you get this highly branched bile duct system. But you also can appreciate when we look at cytokeratin 19 that there are areas of hepatocytes that are resistant even though they're expressing activated Notch. So potential explanations for this is that there are temporal aspects of albumin-crete-mediated recombination, the idea being that it's just not early enough driving activation of Notch. But we know that the albumin-crete that we're using comes on about embryonic day 14.5. And that drives here, it's linked with nuclear GFP, and you can see that throughout the parenchyma and at postnatal day 3, and even those cells that are becoming morphologically in a duct. The other is that NICD, or the activated form, is not sufficient alone to induce all hepatocytes, that certain populations of hepatocytes need other factors and maybe don't have the potential. And the difference is there also could be in the ability of Notch1 and Notch2. We know that Notch2 is the fundamental receptor in the liver to drive biliary development. So to get at this, we looked again using models where we increase using NICD to increase the level of Notch activity. And then we took advantage of HNF6 again, as we had hypothesized that it is the loss of HNF6 that increases TGF-beta activity, and maybe perhaps can unlock the potential of those other hepatocytes that are resistant. And I can tell you HNF6 is a very interesting molecule. I think there's a lot that we still do not understand about it. It is not only localized in cholangiocytes, but also hepatocytes, although its role is primarily thought to be required for bile duct development. And in truth, we don't know that level where HNF6 really plays its role in signaling pathways in driving cell identity. But there have been publications from Mitch Lazar's group really showing that HNF6 has a role in metabolism in the liver. So just to orient you, since this is a liver group, I don't have to spend a lot of time, but we labeled glutamine synthetase positive hepatocytes, which are around the central vein, and we labeled SOX9 showing you the cholangiocytes. And when we activate Notch, which I showed you earlier looking at cytokeratin 19, you can see that many hepatocytes become SOX9 positive. What is surprising, it's really those portal vein hepatocytes that are resistant to that activity and Notch activation. But you notice that in the perizental zone 3 hepatocytes that they seem to be sensitive to that Notch activation. And remember that these portal vein hepatocytes and hepatoblasts are the ones that normally do see Notch activation as you're building the bile duct system. However, if we delete HNF6 in the context of this Notch activation, you can see that all hepatocytes across the parenchyma become SOX9 positive. And these animals are very sensitive and results in liver cancer early. And I think you can get some hints of this even at 60 days when we look at SOX9 and cholangiocytes and HNF4 and hepatocytes and control. If you activate Notch, you can see cells that fully commit into that SOX9 identity. You have a few cells that are a little bit confused. And these animals, a few of them do go on to develop liver cancer. You can see with HNF6 deficiency in that background that all of these hepatocytes are very confused and have varying levels of SOX9 and HNF4. So the question is there also this temporal aspect to this? And I just wanted to show you here that if we look at ENCODE DNA-seq public available data, that at birth, we're just showing you a large area, an 18 megabase area in the hepatocyte genome that is completely shut down and not active, but in adult is very active. So there seems to be these large regions that are becoming open and more accessible to transcription. So in these same animal models that I just showed you at postnatal day 60, at postnatal day 15, you can see that even just with HNF6 loss, that you have SOX9 positive cells and glutamine synthetase positive hepatocytes that are not positive. If you activate Notch, you can see things that are kind of in this periportal region that are becoming cholangiocytes fully. And if you deplete HNF6 in that background, you can see that you do have some cholangiocyte-like cells, but it's not throughout the parenchyma, and that looks very different from what I showed you at postnatal day 16. So it seems that there is this age dependency of those cells and hepatocytes to become at least sensitive to these genetic activations. So the conclusion from this part is that hepatocyte plasticity and identity and commitment required for these molecular changes are for reprogramming seem to be age and zonal dependent, and we have a lot more to learn in this area. So for the last part, I really want to get back to these niche requirements to drive the identity of hepatocyte and cholangiocyte, but obviously my group is mostly focused on cholangiocytes. And really there are three big knowledge gaps that have kind of drove our interest in this area. One is that we showed that hepatocytes have this capacity to become cholangiocytes. So why does this not happen in patients with Alagille syndrome? We know that in their case, they're haploinsufficient. It's not as much as what we showed where we completely decrease the ability of Notch to signal and activate into those hepatocytes. So you would think that they would be able to. And this is a very large study that was led by Benita Kamath, where studying algal patients with cholestasis, that less than 25% of them reach young adulthood with their native liver, suggests that this idea of hepatocyte to cholangiocyte is just not functioning in this patient population. We also know that when we do single cell sequencing in the animal model, which is like the patient is haploinsufficient for JAGED1, is that we see evidence in this red circle of this epithelial population that's observed in the jagged haploinsufficient animals that's not observed, or very few cells are observed in wild type animals. And if you notice and can see that this cell population sits in between hepatocyte zone 1 and zone 2 clusters and between the cholangiocyte cluster. So it's like they're entering into the cholangiocyte program, trying to correct the situation, but they're unable to fully implement the cholangiocyte program. So we wanted to understand why that is. And I think, as I showed you earlier, I think you can appreciate that this animal model, where we completely made the inability of hepatocytes to drive it or receive notch activity, they're still able to build de novo a bile duct system. But I want to point out that the portal vein mesenchyme is genetically, not genetically modified, so only the epithelium with wild type support cells. So we really wanted to get into this idea of, since we think that the mesenchyme is absolutely important to support the signal, and the patients are haploinsufficient in all cells in the liver, and based on Kathy Alum's work and a work from Luisa Arilla-Rispe's lab, testing both the endothelial compartment, the mesenchymal compartment, and the epithelial compartment, it looks like jagged is absolutely required in that mesenchymal compartment in order to drive bile duct development and specification. And I'm just showing you here in the jagged haploinsufficiency model, you can see that there are some tubes that are communicating, but they're not in the correct architecture. They really have this homogeneous architecture versus a hierarchical architecture. And even at 7 months of age, these bile ducts that are made are not remodeling properly. So we again took advantage of lineage-specific tools in the liver, using albumin-Cre again to drive human JAGED1, or SM22 to drive human JAGED1 in the mesenchyme. And this is just showing you the specificity of those drivers, both at an embryonic time point, showing that they have turned on, and then at a postnatal day 15 time point with albumin, you can see that it's both recombined, so that the hepatoblasts are giving rise to both cholangiocytes and hepatocytes. And you can see this, the mesenchyme that's surrounding both the bile duct and the artery, and part of the portal venous mesenchymal cells. And this is just showing you with RNA scope against a human jagged probe to show that both of these transgenes are really working and still have activation of jagged within the parenchyma. So I know these are going to be a lot of panels, but hopefully you can bear with me. You can see, if we look here out at the edge or periphery of the left lobe of the mouse liver, that you can see some nice hierarchical bile duct structures. If you overexpress human jagged in the albumin-Cre lineage, the epithelial lineage, you can also see some hierarchical structures, maybe potentially a few extra communicating fine network structures. And then also with SM22 in a driving jagged in a wild type background, you can see the hierarchical with also these small fine branch networks. So what happens if you're in a haploinsufficient jagged population? You can see again, as I showed you before, you have a very truncated system that doesn't make it out to the periphery, and you have this homogeneous bile duct network that doesn't resolve. If you drive jagged in an albumin background in the epithelium in a haploinsufficient, you can see that you get more of that homogeneous network. And this is potentially more severe than a jagged one, as we can tell by serum chemistry to look at liver function. And if you drive jagged with the SM22, you can see that you now are gaining back this hierarchical network. just at the cholangiocyte level, the panels are in the same order. So I just want to focus down here on the bottom, where you can see in jagged, you have these unremodeled, they're expressing these cholangiocyte markers that we use in the field, SOX9 and osteopontin. If you do the same thing driving with albumin-Cree in that background, you can see that you have cells that are entering into that cholangiocyte identity, but you also get the idea that just making more cholangiocytes is not beneficial. So the program of these cells don't fully implement the cholangiocyte program. And if you drive with SM22, you can see this remodeling going back to a more normal architecture. And just to kind of bring the idea and focus that we're driving jagged and the mesenchyme impacting the bile duct architecture, but we also impacted the mesenchyme itself. You can see that in jagged haploinsufficient animals that you have lots of fibroblasts expressing PDGF receptor alpha. You also seem to get more cells that are expressing actin smooth muscle, alpha smooth muscle actin. And when you have the mesenchyme compartment expressing jagged, you can see that you recover that back more towards a wild type pattern. So getting jagged expressed in the right compartment, and apparently it doesn't matter if you're expressing at really high levels. I'm not showing you that data today, but it's expressed at very high levels. You can impact both compartments. So this idea, conclusion is that human jagged 1 is sufficient to alleviate this jagged haploinsufficiency. And it suggests that really for some of these cholangiopathies, especially with Alagille syndrome, we need to think about reprogramming the niche as well as the epithelium. And just to end here, this last little bit, we're trying to, as we go further into this area, since we know the mesenchyme is important, we also need to build tools so that we can regulate it and understand the signaling mechanisms that are working towards bile duct specification and remodeling besides just the Notch pathway. So one paper from Robert Schwartz's group really highlighted that there are GLI1 positive cells, and I'm showing you here work from our lab in deep three-dimensional imaging. These GLI1 LAC-Z positive cells that you can see are tightly associated with the cholangiocyte marker osteopontin and a major tube, a bile duct tube running through the liver. So the first question should be, are these GLI-LAC-Z cells even there at a time that's important during development? And I could show you this is embryonic day 16, but just giving you an idea that as cholangiocytes are starting to be specified, you can start to see cells that are surrounding that portal vein to become positive for the nuclear LAC-Z. So as the bile ducts are being formed from the hilum out to the periphery, you also see the GLI cells starting to be. So does jagged 1 impact this population? And I'm showing you here in cross section what we're viewing in a deep cleared image to look at XSCAL, that these GLI1 LAC-Z positive cells shown here by the XSCAL staining or blue staining following the portal vein, and in cross section you can see that they're associated with the bile ducts marked with osteopontin. And if you look at a jagged haploinsufficient animal, less severe and severe, you can see that you have this truncated biliary system, but you still get some GLI cells that are in and surrounding there. But if you have a very severe phenotype where there really aren't any bile ducts formed or luminal spaces of bile ducts formed, you can see very few of these GLI positive cells. So there seems to be an impact with both of these populations. And if we look at human explants of Alagille syndrome, you can see basically a atretic duct that's still here in the patient, but you can see that PDGF receptor alpha is kind of missing in this population that's surrounding and should be around the bile ducts. And you can see these reactive ductules that are trying to form, and you can see that they're surrounded by this PDGF receptor alpha. So the question really is, what are the other subpopulations of mesenchymal cells and their properties that are required to get bile ducts to establish and maintain? And so I just want to, at a really high level, we're starting to work with spatial transcriptomics, not only multiomics, to really understand the cell states and their spatial associations, since this is a intercellular communicating developmental process. And we initially used the Resolve platform to look at 95 different genes. This is just showing you an example of that. And we're able to show that even with a limited subset from 2 to 6 markers in different cell types, that we can really identify 23 cell types using only 95 genes. And I think what's really important in our first set that we did comparing human and mouse at a high level, if you look at the cell types comparing the allogel versus healthy and the jagged heterozygous mouse versus the wild type, is that the populations I think we really need to be interested in, which is that different kind of cholangiocyte intermediate cell and the peribiliary fibroblasts are sitting right on that line of that they're changing in the patients and the humans. And so this really gives us a great path forward to really start to understand the human disease using our mouse models. So the last is that this mesenchyme is impacted by jagged haploinsufficiency, and this suggests that we really need to define these niche subpopulations that we're starting to uncover in other hepatobiliary diseases as well. And I just want to thank, I think I got everyone on the bottoms of the slides who was involved in this work, but now Ashley Castas and Jennifer Barwinkle and Carrie Huppert are really the leaders in this niche realm. And as we go forward, we're excited to share that work with you. And I have our collaborators who have worked in different areas, Holger Taco, as we're building different human organoids to try to study these concepts. Hamid Jafar has been a long-standing collaborator in the algal space with us. Greg Tiao and Alex Bondacht, I really couldn't do our human work without them, as there are liver transplant surgeons. Anna Peters has really been working with me as a hepatologist to help me get those samples. Rob Schwartz, Nathan Salamonis, and Andre Rendero has really been helping us with our bioinformatic work. And I'll stop there. Thank you so much.
Thank you very much. That was beautiful. Because of time constraints, I'm going to restrict it to 2 questions. We have already one in the chat from Aparajita Lari. So thank you, Dr. Huppert. I'm curious about the function of these SOX9 positive hepatocytes in the HNF6 flux NCD active mice. So this, I guess she's referring to those zone 3 cells. Are these functional hepatocytes by whatever albumin production, bile acid synthesis, etc.? And how does the bile caniculi network look in these mice? So to basically get an understanding of the polarity of these non-zone or like these cells you showed, these SOX9 positives.
Right. So yeah, so refinement during that remodeling is from a hepatocyte becoming a cholangiocyte. We're still working on, but obviously in the area where they're starting to do that transition, cells are making different connections. And so of course the canalicular surface as you normally see that is disrupted a bit, right? As they're trying to remodel and make connections with each other as they're changing identity. And what was the other question was about metabolism, right?
Well, how much have you tested these hepatocytes, these SOX9 expressing for hepatocyte functions? I think that's what she was getting at, I assume.
Yeah, so we haven't done anything as far as functional tests yet. We have done multi-omic analysis, and so as that data becomes available, we're happy to share that, but to really understand the transcriptional and both the chromatin level, what's changing in that time period, that temporal window, what happens with hepatocytes and their ability to be plastic.
Thank you. Are there any other questions? Maybe I can throw in one question. Have you tried in those SOX9 positive cells, if you then, for example, what Stuart Forbes did with the P21 overexpression and you do a partial hepatectomy, do they start forming little bile ducts sort of that are not connected to the main bile ducts, or?
So if you do SOX9 overexpression, we actually did that with Hamid, and it's published in a paper where we were looking at genetic modulation and sensitivity to SOX9 in the jagged haploinsufficient model. And actually they don't drive by itself. SOX9 is not able to drive SOX9 positive hepatocytes.
Oh, okay.
Yeah.
Thank you. So I think if there are no other questions, then we'll move on. So it's my pleasure to then, the next speaker will be Hans Clevers. So he received both his medical degree and his PhD at Utrecht University in the Netherlands. And then he did a postdoc, I guess, virtually here at the Dana-Farber with Cork Sterhorst. Moved back to Utrecht, and there he made many groundbreaking discoveries in the Wnt pathway, leading to the discovery of the LGR5 positive adult stem cells, and then the creation of tissue organoids. I think to list all Hans's achievements and prizes, I think is sort of a micro talk by itself. But I think these days I'm actually more impressed if there's an organ that you cannot grow as an organoid from than when you publish another organoid paper. So please take it away, Hans. I'm very much looking forward to this.
Yeah, thanks, Ipa. I hope this works. You see my screen? Yeah, so what I'll try to do in the next 25-30 minutes is give you a little bit of background as to how we sort of fortuitously developed organoid technology from this organ that you see here, the gut. And then I'll give you a few examples of how we have actually been able to transfer the technology to the various liver types and how we aim to build disease models from these. So these are the villi of the small intestinal epithelium, extending the surface to make uptake of nutrients more efficient. The crypts were known to harbor stem cells, and I indicate them here in multiple colors. We found indeed this LGR5 marker originally. We now know it's actually an active component of the stem cell. It's a receptor for our spondin, which allows stem cells to very strongly hyperactivate local Wnt signals. So if you have our LGR5, you're much more sensitive to Wnt if the ligand, our spondin, is around. There's biochemistry around it, we know how it works, but I don't want to discuss that here. So we made a GFP knock-in, also CRE-R. You see here these tiny little green cells. This is an LGR5 locus lighting up. And these cells we then by lineage trace could show that they are indeed the stem cells of the gut, extremely active. Every 5 days in the mouse, they entirely replace the inner lining of the gut. Also, they divide every day, which was against the common belief then that stem cells should be quiescent. So in the mouse, about every 23 hours, in human, probably longer. And based on that observation, Toshi Sato and me sat down and we came up with a very simple cocktail of growth factors. So our spondin, the Wnt amplifier that we later found out to be the ligand of our marker of LGR5, epithelial growth factor, and a BMP inhibitor noggin that we knew from an old transgenic mouse experiment to create what we called a crypt-permissive environment. The idea was take one stem cell, so sort a stem cell out of the gut of these mice, and then make many stem cells. We got these structures that we now know are good copies, phenocopies of the gut epithelium. They create the central lumen, so they're closed spaces, they're not a tube. Then these buds are the crypts, and all of the cell types that we recognize in the normal epithelium is present. In these organoids, you can actually play around with relative ratios by changing the growth factor conditions and essentially produce all of the different cell types at high numbers in these organoids, including the very rare ones. To test whether they were really normal after having been grown extensively, Mamoru Watanabe in Japan transplanted our organoids, and they actually, in an IBD mouse model, indeed can actually recognize the ulcers, and then with their basal site on the outside, their integrins on the outside, they bind to collagens and other subepithelial structures, and then cure these lesions. Let me see if yeah, there we go, sorry. And this is a more recent paper where we've played around with the conditions for the human small intestine, and CRISPR works very well, CRISPR-based knock-ins. Actually very easy to do this in various types of organoids, also in liver organoids. And here you see MUC2 for goblet cells, defensin A for Paneth cells here, and chromogranin A for the rare hormone-secreting cells, of which there are about 5 or 6 types in the gut. And they're all present, as you can see, in these organoids. Now, since then, indeed, we have managed, and other labs have joined in, to develop organoid platforms for other tissues. You see a whole list here. Typically, you don't start from a stem cell. Often in organoids, in organs, stem cells might not be defined, or professional stem cells might not even be present in such organs. It's always variations on a theme. So Wnt, tyrosine kinase receptor activation, and inhibition of BMP and TGF-beta. You can grow these organoids for very long term. Actually, the liver has been the hardest to grow for more than half a year or a year, and we actually have to reside on very young donors or on fetal human tissue to get lines. But for the gut, they've been growing many, many years. Telomeres stay long. We don't see oncogenic mutations, and the transplantation data just shows also confirms that this is normal tissue even after extensive expansion. No, I don't have to show this in this audience, but my view as a not really a liver specialist is that the liver is probably the champion of organ regeneration, and I'm convinced there is no professional stem cell in the liver. The cholangiocytes and the hepatocytes are the two cell types quite abundant, particularly the hepatocytes, that essentially endow the liver with a stem cell function, which means that damage is repaired, and I do not believe that there is a hardwired professional stem cell hidden somewhere in the liver, and it's probably not needed. The first protocols that we developed to grow human liver and mouse liver were done by Mary Hoog, who is now independent after a stint in Cambridge, UK, as a director of a Max Planck Institute in Dresden, and she actually continued on this. So we found that it actually is exceedingly easy to grow cholangiocytes, despite the fact that they're highly differentiated, beautiful polarized cells, as all of you realize. So here we sort them from a human donor liver, and much to our surprise, so these are fully differentiated cells. 1 in 2, 1 in 3 of these cells will actually grow an organoid, as you can see here. Hepatocytes have resisted long-term culture in many labs, and also in our lab, it's taken a long, long time for them to grow. And here we actually had not figured that out yet. A movie of what these cholangiocyte organoids look like. So they don't really look like bile ducts, but you can actually fuse them and then make longer ducts, particularly if you guide them in gels, in bioengineered gels. But it's very dynamic, as you can see here. Pretty fast growing. We can grow them for a number of months. When we transplant, particularly when we play around with the notch pathway, following up what Tracy, for instance, just said, what Stacey, sorry, just said, is that they can become hepatocyte-like, but in vitro they start expressing some albumin, some of the cytochromes, but they never become something that really looks like a hepatocyte. Upon transplantation, you can actually see that they can also make hepatocyte. Again, I think underscoring that these liver cells show a lot of plasticity. On the confocal, you can see they're very thin-walled. What they don't show here, they're highly polarized, so they have an apical domain and a basal domain, and you can actually mature them out, and they start looking like real cholangiocytes. A story that we've built on this particular organoid platform involves a tumor suppressor gene called BAP1. It's mutant in a number of human cancers, but of interest here is commonly mutated in cholangiocarcinoma. There are a lot of papers on what BAP1 would do as an oncogene, why it would lead to cancer if you lose it. However, there is a BAP1 homologue in the Drosophila genome called Calypso. All of these papers report that BAP1 would do something on apoptosis or on cytoplasmic function, cytoskeleton, etc., etc. However, the function of BAP1 on the fly is very different. It's a polycomb component, deubiquitinase, and specifically targets this particular mark on H2A. So very different from what was proposed for the human version of BAP1. And we decided to study BAP1 in cholangiocyte organoids. This is Benedetta Artigiani, who is now independent in Utrecht. This should also start playing. Yeah, so here you see again the same movie of the normal cholangiocyte organoids. And this is when we just knock out BAP1. We have multiple clones. They're quite easy to find because they're very dark, and you can see they're very dense, and they look more like blobs. And the first impression you get when you see these, they really are no longer very epithelial. And that turns out to be the case. When we look by EM, this is the luminal side and the outside of a normal cholangiocyte organoid. You can very nicely see these microvilli here, so the polarization of this thin-walled organoid. In the BAP1 mutants, we see no sign of polarization. We also see almost no sign of formation of mature junctions. There's a lot of space between the cells. So disorganized polarity is lost, and there's a lot more intercellular space in in the epithelium. That's confirmed by confocal. ZO1 marks tight junctions. You can see a beautiful network here in these highly polarized wild-type cholangiocyte organoid. This is a number of stacked sections, but here is a single section. You can nicely see where the ZO1 sits. If you do the same stain on the BAP1 mutant, only they're homozygous for BAP1, essentially there's almost no localized ZO1 expression. And you can see it's essentially, if you do see it, it's essentially everywhere. So again, saying something about the fact that BAP1 somehow controls an epithelial behavior. Now, when we now re-express BAP1 specifically in the cytoplasm by taking its nuclear localization sites out and we link it to GFP, this works well. You can see a nice induction, 24-hour movie here. It runs again. So we get it expressed in the cytoplasm, but there's essentially no morphological change. If we do the same thing in the nucleus by giving it a few extra NLSs, now we get a very strong reversion of this non-epithelial, this mesenchymal phenotype, implying that what BAP1 does, what it actually does is actually it has a nuclear function, which immediately says that the proposed functions in human cancer probably are not the correct ones, at least in this model. We then include a lot of genomics and similar analyses, and I won't take you deeply into this, but we find that there's much to my surprise. So it's an epigenetic regulator. It really is deubiquitinase in the nucleus of histones. We find that a very tight set of genes, about 300, are regulated by BAP1. So when we delete it and then re-express it, we actually see a very tight profile of genes. And when you look at the genes that one can recognize, they're all are involved in epithelial biology. So junctions, proteins that are either expressed in the polarized way or that set up the polarity, et cetera, et cetera. And chromatin accessibility, I just show you one slide here, is actually indeed controlled, because in the BAP1 mutants, you can see that a lot of the accessibility of some of these sites is Claudin-1, tight junction component, actually is, the access to this locus is controlled by BAP1. If we now try to build cancer models from these organoids, we've done this a lot in other tissues. So we start from wild-type tissue with CRISPR. It's quite easy to actually generate a whole set of mutants. So when we knock in the 4 most common tumor suppressor mutants in cholangiocarcinoma in the same organoid line sequentially, we also do it at the same time. So homozygous mutations for p53, SMAD4, NF1, and PTEN. These organoids actually look pretty normal. And when you transplant them, they don't really look too bad. If we now add BAP1 on top, we immediately get that very strange BAP1 phenotype again. And when these are transplanted, so this is the 4 mutations, so pretty vicious, you would think, with 4 potent tumor suppressors knocked out, but they give a very, very benign pathology when we transplant these orthotopically into mouse liver. So these are human organoids. And when we add BAP1 on top, our pathologist tells us this is prototypic cholangiocarcinoma with a fibrotic response around it. I'm not a pathologist, but you can see the huge difference between these 2 phenotypes. And we've actually used organoids also in other settings to find out what is the minimum requirement of oncogenic mutations to set up a benign or a malignant tumor, and also to find out what the functions of cancer genes of unknown function could be. And so for BAP1, I think this study strongly suggests that what its role really is, it's the proposed or the defined role in the fly, so a histone modifier, and therefore an epigenetic regulator. Back to the other cell type, the hepatocytes. So they resisted our attempts to culture them for a long time. We always start in mouse, and you can see we start from single hepatocytes, but nothing happened. Hu Li, now back in China, actually found a complicated cocktail. She published this 3 years ago or so. And you can see here, this is still mouse. So an hepatocyte marked by albumin Cree, large cell, 30, 40 micrometers, large nucleus. It starts growing, but you can see these are really growing hepatocytes. They don't differentiate much. They stay large. And everything we've learned since holds true. So here you can see a human hepatocyte organoid, very large nuclei, positive for HNF4 alpha. We stay in here for alpha-1 antitrypsin. You see massive amounts of alpha-1 antitrypsin, similar for albumin. Depending on the culture conditions, we can get all the cytochromes expressed at the levels that you'd see in the real liver. The one difference if you do single-cell sequencing with primary hepatocytes is the fact that some of them are in cycle. These are not the fastest growing organoids, 3-4-fold at most per week. But as I said, the younger the donors are, the longer we can carry them. And we now have 4 or 5 hepatocyte lines in the lab that come from human fetal livers. And these we now have had for several years, and we've done a lot of CRISPRing and subcloning, and they essentially do not change over time. You can see the large nucleoli here. They can be 4N, it can be even 8N in culture. And we actually know that we can start these organoids from single cells, single hepatocytes, and even from single 4N hepatocytes. Even more so, there was in one of the previous talks, bile canonically came up. It looks like these things have bile canonically, and on the blow up you can see it here. So it looks like in 3D that every hepatocyte, its apical domain helps build a bile canonicalist, and we mark here the MRP2, a bile acid transporter, and they feed into a central space. This is where we think that the bile duct should be hooked up, but these organoids don't have, under these culture conditions, don't have cholangiocytes. And we're currently trying to actually see if we can connect it to, and I know that Mary Hoog is doing similar work, and we indeed find that you can get very nice connections between these trees of bile canonically and the central space. Actually, in these organoids, this space will grow and grow. There's pressure buildup, and eventually it sort of explodes, and then it releases its bile acids, closes up, and then the same cycle starts again. And this is actually a knock-in for MRP2 for this bile duct marker. So this is a single hepatocyte, you don't see it, but once you have a few hepatocytes, the apical domains start forming. MRP2 is immediately put there. You can see there's a little lumen forming. It's actually now breaking up. These are sections. And then you get these very nice networks, and I believe this movie should play. Yeah, so we're actually now trying to take movies of this whole process and see if you can learn a little bit more about what the rules are, about how these exquisite branching systems are being set up. And then together with the chairperson of the session, Ipe de Jong, we performed a transplantation. This is not as easy as it is with primary hepatocytes, I learned from Ipe, but this was an experiment with one of these young donors. And you can actually see these larger hepatocyte islets that are developing from the transplanted hepatocyte organoids that have been expanded significantly in culture, and Ki67 marks the proliferating cells, and I believe this is done 2 or 3 months after transplantation. So you can see that the hepatocytes stay in the FAH mutant background that I'm sure all of you know. They stay proliferative, and they slowly appear to be filling up the liver of these mice. To build disease models, one that we're currently working on, I just show one slide, is an attempt to see if we can turn these hepatocytes into a platform to model chronic hepatitis B infections, which, as far as I know, has been very, very difficult in vitro. And it turns out to be quite easy on these organoids. We grow them in 2D. This is done by Daizong Wang from Shanghai, new postdoc in the lab. And here he shows 32 days, but he's now gone out 2 months, and we actually see persistence of the viral genome in this mini chromosome-like format, the cccDNA. We see production of the various antigens, hepatitis core, the circulating antigens, and we see that the viral particles remain to be produced and they're infectious actually in the culture medium of these hepatocytes. And we think this might be a platform maybe to better understand how the virus is maintained in its chronic state, and possibly also serve as finding cures for this currently essentially untreatable condition. And another model is and also very well known to all of you, Neville the Nash. Here, actually, this was done because we believe that ApoB, or some people in the lab believe ApoB is a cancer gene. No, ApoB mutants can get cancers, but it turns out that the moment you knock it out, so the liver cannot export the fat it makes. And actually, we have shown, I don't show it here, but this is de novo synthesis of triglycerides. You get a massive buildup of these droplets, and very surprisingly, they don't really care very much about this. They keep on growing. And actually, when you do RNA-seq, there's a lot of repair or resistance mechanisms that you see induced by the presence of the lipid, and we think that's why they apparently that they managed to live with this. You can do the same thing by taking normal organoids and give them high levels of free fatty acids. Normally, there's essentially no lipid in the media. We also add no serum. There's never serum in organoid media. And you can see you can load them up, but this is now not de novo synthesis, but this is basically uptake of free fatty acids that's then converted into triglycerides in these cells. And again, so you see the buildup here. And again, this is my last slide. We can create genetic models quite easily. So we've probably created something like 60 or 70 of these. And this is a well-known risk gene, probably known to many of you, where we can make, this is now using base editors or prime editing in this case, so you can make single base changes. And so we go from a wild type organoid to an organoid with a single SNP, risk SNP, and then a homozygous risk SNP, and then the knockout. And if you look carefully, you might see that essentially there's no lipid buildup here under normal conditions, tiny bit here. When you get homozygous, there are more, but when you knock out the gene, there's a lot of buildup, and which essentially tells us that PNPLA3 is essentially a protective gene in itself, and that the risk genes must be loss of function genes. And we have a number more, Dalila Hendricks and Benedetta Artigiani, who are trying to publish this study, actually have made a number of these risk genes and are currently trying to write a revised version of this paper. With that, I think I'll reach the end of my talk. I'll stop share. I think I mentioned the people who have been involved in these studies along the way, and I hope I left some time for Q&A. Thank you very much for your attention.
Thank you very much, Hans. That was beautiful. Are there any questions?
Hans, thank you very much for participating and presenting this very exciting new work. Do hepatocyte organoids show anything like the metabolic zonal distributions that we make so much about in the intact organ?
Yeah. So as Ipe mentioned, so my background is originally the Wnt pathway, and so our perspective on the zonation is essentially that there's a very potent source of Wnt in the central vein, around the central vein, and we can actually, so our organoids, if we grow them in high Wnt, they very strongly resemble the what is zone 3, I guess, hepatocytes, and when we block Wnt, we get zone 1 hepatocytes. And my prediction would be that if you would take a zone 1 hepatocyte and you put it in zone 3, given the local signals where Wnt is probably a very important player, it'll start looking like a zone 3 hepatocyte. So I don't think we've actually, over the 15 years or so that we've been looking at stem cell and stem cell function, I've learned that these systems are all extremely plastic and very tightly controlled by location and by niche. And I think the liver is a very good example of that.
Thank you.
Can I ask a question then, Hans? If you take the Wnt or you down modulate the Wnt concentrations, do the cells become more resistant to steatosis, either from DNL or from loading?
That Dalila should know. I
okay, I'll ask her.
Okay. Yeah, that would be a prediction. I think if you look at some of the enzymes that are part of the Wnt targeting program. Yeah.
And there's also another question online. Thank you, Dr. From Aparaita Larry, the same as the question from last time. Thank you, Dr. Clevers. How is the CLF-CFDA uptake in the liver organoids? Asking to understand if basal and apical transporter functions as they do in vivo.
Now I have to know what CLF and CLDA?
CLF and CFDA.
Yeah, what are they?
Basically bile salts analogs.
I know we have played around a lot with bile acids and derivatives, but again, Dalila could probably tell you, and I wouldn't be able to.
We'll table that one. Are there any other questions? Okay, with that, thank you very much, Hans. Then we're gonna move on. Have a good night.
Thank you.
Let me see. So our next speaker is Dr. Weigel. So Aubrey Weigel. So she trained in physics and in engineering, and she's currently a project scientist at the HSMI Genelia campus. Her lab studies ultrastructure of subcellular organelles using advanced imaging technologies, a little bit too advanced for my pay grade. So I hope today she'll undoubtedly enlighten us on some of these very advanced technologies, including, to start off with, what the V in VEM stands for. and how they provide insight in organelle biology. The title for talk is Towards Automatic Segmentation of Organelles in VEM. So the floor is all yours.
All right. Well, yes, thank you to the organizers for inviting me to share our work here and for organizing this excellent symposium. So the V in VEM is volume electron microscopy. So I'll be sharing with you some of the work that I've been doing with the CellMap project team that I'm leading here at Janelia towards doing automatic segmentation, so machine learning in volume electron microscopy. All right, so when we look at all the different organs and tissues in the body, and we zoom in a lot, what we see is a collection of cells. And within those cells are the organelles. All the cells have them. They're the building blocks, from highly specialized cells within tissue to plant cells to immortalized cancer cells, even simple unicellular organisms such as protozoa. At the foundational level, all the organelles within the cells have the same basic functions. DNA is packed in the nucleus, proteins are synthesized in the ER, and mitochondria generate ATP to fuel the cell. And of course, what we as a community would love to understand is how the stuff that's happening inside the cell propagates back to the whole cell, to the tissue, to the organ, and then to the animal itself. So historically, light microscopy has brought us a long way in understanding organelles and their functions. And the beauty of fluorescence microscopy is it gives us protein specificity. So it allows us to see organelle dynamics and how they interact with each other with global context. So we can see all the organelles in the entire cell. However, even with multispectral approaches, we're limited by the protein label. So we only see what we label, and we're limited by the resolution. So contrasting to that, electron microscopy offers more global contrast. We get higher resolution, but traditional methods usually limit the volume to which we can investigate to a very small area. So we lose the context. We don't know exactly where we are within the cell or the tissue. And a valuable tool that kind of selects the best of both of these worlds is focused ion beam scanning electron microscopy, which is a mouthful, so I'll be calling it FIB-SIM from now on. So for those unfamiliar, I'll briefly describe it. First, a scanning electron micrograph is taken of a resin-embedded sample that has been contrast enhanced. So if you imagine a block, you image the top layer of that block. Then a focused ion beam shaves off a very thin layer of the top of that block, about 2 nanometers, and another image is taken. And you do this ad nauseum, image, shave off a layer, image, shave off a layer, until you collect an entire volume. And the beauty of this volume is that it has near isotropic resolution. It has global contrast from the EM staining. And thanks to the ingenuity of the Hess Lab here at Janelia, we can do this across large volumes. So for instance, in our context, large means on the Janelia recently published a fruit fly brain connectome, which used this technology. So at the end of the day, not only do we gain resolution through this technique, but we also retain context, which is really important. So here's some examples of what these data sets look like. So there are lots of shades of gray, there's lots of texture, there's lots of information. And these are from 4 very different tissues. One is of a cancer cell, one is of a mouse brain, and a couple are from a Drosophila. So these data sets are just overflowing with information, and it's really just begging to be extracted. However, they're large in size. Oftentimes, they're terabytes in hard disk space, and extracting the information out of them really is a mountain of a task in and of itself. So to attack this, the COSIM project team, which was the cell organelle segmentation and electron microscopy, worked to develop a machine learning pipeline to automatically segment those organelles and structures within the cells. So we started with the beginning, a single cell, and we used existing data sets that the Hess Lab had created of conventional immortal cell lines, so just simple things to develop this proof of principle pipeline. So I'll walk you through what that pipeline looks like using a HeLa cell as an example. So we started with the aligned volume EM data that was collected by the Hess lab. Then Grace and Allison in the group manually classified every single voxel in a subblock with up to 37 different classes and substructures. So what did we care about? Well, we tried to be as agnostic as possible. So here's some examples. We included the popular things like the mitochondria, the plasma membrane. We included trickier organelles to annotate like the ER, the Golgi, lipid droplets, and the lysosomal system. We also included cytoskeletal elements like microtubules and nuclear components such as chromatin, the nucleolus, and nuclear pores. And then lastly, we included ribosomes as a classifier. So altogether, here are the 15 subblocks in this data set that we used for training. We just recently released a protocol on how to generate this kind of training data. So if you're interested, you can check that out on protocols.io. And I'll highlight here that we use different combinations of classes to feed into our machine learning pipeline, testing whether it was useful to train all 37 classes at once altogether, or a subset of those being the most popular ones, or just training classes individually, only the mitochondria, only the ER, etc. All right, you can also see in this bottom graph that the amount of training data acquired from each data set is pretty small. So even though, even from the cell that I showed you in that movie, which had arguably the most training data, it was only a fraction of a percent, so 0.18% of the entire cell volume that was annotated for this machine learning training. And on the right, what I'm just quickly depicting is giving you an idea of the hierarchy that we set up with the different organelles. And how we come to 37 classes, even though there's not necessarily 37 organelles. All right, so all of that training data that I just showed is used to train a machine learning classifier. We use a mostly standard setup. It has a 3D unit architecture, as you can see in the middle, and this is trained repeatedly feeding in blocks of raw data, and that produces some output for the innermost part of the block, so the red part's there, and then is compared to what it should look like according to the ground truth annotations, and the parameters of the networks are adjusted accordingly. So this process is repeated hundreds of thousands of times in our case, and we specifically train it to predict a signed hyperbolic tangent distance of the binary label for each organelle. However, those distance transforms can be transformed back to the binary segmentations that you would expect by just thresholding at 0. To find the optimal setup, so as the machine learning network trains, you go over many iterations and you are computing a loss at each part of that iteration. You're trying to optimize that point. However, we need a way to validate that and evaluate it. So one way of doing this is through by creating a manual annotation as a validation block. So here is one of those such blocks, and then an example of the predictions on that region. We then evaluated and used the F1 score or DICE score, which falls between 0 and 1 and is based on the measuring pixel overlap. So higher is better here. And the results I'm showing are from the validation and test performance for thresholded predictions on the holdout blocks from our 4 data sets. All right. And then if you remember from a few slides ago, we also looked at training at all classes versus many versus a few. And here are those results. So for each network, there's 4 points from the 4 data sets that we had. We had 2 HeLa cells, a macrophage, and a Jurkat cell. And rather than average over all 4, you can see the spread then by plotting all 4 of those. All right, and what's reported here are the test scores, the DICE scores, meaning that for each point, the iteration was optimized using the average of the remaining 3 blocks. So generally speaking, we didn't see a large effect, but overall there was a hint of a trend that having more classes together seemed to perform better. And I can say anecdotally, as we, since we've published this work and moved into more and more data, that this holds true. The more in general, the more context our systems have, so the more organelles we train on, the better it performs. All right, the other thing that we were really interested in, of course, is we didn't want to stay stuck in cells or just a silly HeLa cell all the time. Ultimately, what we want to be able to do is generalize across data sets so that we could blindly go into a new data set of something completely different and still be able to robustly predict what the organelles were. So to see how well our networks were generalizing, what we did was we had our base model, which was trained on all of the data sets that I'm showing here. And then in addition, we trained models using only training blocks from its respective data sets. Then to evaluate, what we did was pulled out one block for validation from each of the training subsets. And these are the results. So there's a bit to unpack here, but I'll walk through it. So each row of the matrix here shows which validation block was used in each column, the training data set. So the diagonal from the bottom left to the top right here is using a validation block. Sorry, the diagonal is using a validation block and training subset from the same data set. So that's the best. The last column is our attempt at a more generalist network, so being trained on all the available data. So with only a few exceptions, adding additional training data from the other data sets improved the performance. And like I said, anecdotally, we see this as well for into our other data sets that we have worked on. And some downsides to this type of evaluation is that the approach is that it's very biased towards the types of organelles that we're looking at. So for very thin structures like microtubules and the ER, this test statistic is much harsher than for large things like the nucleus and the mitochondria. So for that reason, we do rely pretty heavily on a manual evaluation approach. And in this way, what the expert annotators do is after the predictions are made, they are then offered two different prediction results, and it's randomly sampled throughout the data set. They're blind to which prediction they're looking at, and they just choose A or B, which is better. They do this 100 times, choosing one window versus the next to decide which prediction was better. And at the end of the process, based on a test statistic, we can decide which prediction is actually doing better for that specific class. All right, so from here, we can evaluate all of our results, we can combine them together, and then this is really the exciting part, right? So all of these segmentations that we have aren't quite perfect yet. There's always more work to be done, but some of the errors that occur are relatively straightforward to correct. So we can do things like size filtering for something like the nucleus, if you have little small bits, and we can also do things like the connected components. So for instance, the ER should be all connected, the plasma membrane is all connected. And then we do some slightly fancier things, like for the case of microtubules, we used a priori information about structural biases for microtubules to train a network to better improve those filaments. All right, so then all of these things can be combined together, the predictions and the refinements, and we're able to get pretty nice segmentations for a number of organelles. And this worked really well across the 4 data sets that were our main focus for the start of this work. What was really exciting at this point is that these predictions can really let us start to see the organelles in a way we've never really been able to do so before. So we can take the Golgi, for instance, which is one of my personal favorite organelles. And the textbook shape of the Golgi is this cute little stack. It has 5 to 7 neat cisternae and a clear cis and trans space. And when you look at it in its entirety, the whole Golgi in a mammalian cell, like what we have here, is you find that it's something that's much more complex, and I would argue is more beautiful. So these 7 cisternae typically seen in the textbook images are now actually weaving one into the other, and what was a cis face of one Golgi stack might be the trans face of another. You can also see the vesicles studded all around the edges. And furthermore, as I'm showing here, the ER is so intimately connected with the system that it can be hard to distinguish one from the other. So all these complexities are lost in textbook cartoons, and it's nearly impossible to resolve with light data alone. So this was a huge win for us. These segmentations also are more than just a pretty picture, so we can really start to quantify information from them as well. So this is what we sought to do from the beginning, right? To extract that information from these volume electron microscopy data sets. So for instance, the percent volume occupied by each organelle across different cell types. And I'll give you 3 examples here of what we've done in this cell as well. So first we have the contact sites between microtubules and other organelles in the cell. So you can see the microtubule here, and color-coded are the organelles that this single microtubule is touching. This particular guy is only about a micron in length and really quite busy. So we can quantify these contact sites for all the microtubules in the cell, and the results of which are shown in the graph on the bottom. The most contacts occur with the ER, which is maybe not so surprising. Next, I'll show you an analysis of membrane curvature. So for this, David took the ER predictions that we have and iteratively thinned them down to produce a medial surface. So it looks like a skeleton. He could then calculate the curvature on that surface and threshold between planar regions, which are in green, and tubular regions, which are in purple. And what we noticed are two things. So a highly secretory cell, like a macrophage, had more planar peripheral ER regions than a HeLa cell. And there seemed to be an affinity between the flat planar regions of the ER and the mitochondria contact sites in the HeLa cell. So as you can see here, when the movie pauses, the contact sites in blue seem to match up to the mitochondria in orange. And in fact, when we analyze those contact sites for the whole cell, we see that HeLa cells, in HeLa cells, the planar regions of the ER were almost always supported by the mitochondria, where that was not the case in the macrophage, where they had these stacked helicoidal sheets. The third analysis example is with ribosome classification. So we started with all the cytosolic ribosomes, and then we categorized them as being ER bound or not, and then further classified them as being on a planar or a curved or highly curved region, a tubular region. And what was interesting here is we found that polyribosome chains tended to happen on the more planar regions of the ER. So with an ambition to really democratize volume EM as much as we can to the broader community, we importantly make all this work publicly available through our open access data portal, Open Organelle, and with the data hosted by AWS. So here, together with the HES Lab, the FibSum Technology Group, and Scientific Computing at Janelia, we provide this browsable, queryable interface for folks to explore these EM data sets and the accompanying segmentations. So on the site, you can filter the data sets to more easily find what you're looking for. You can browse pre-made views, or you can customize your view based on what segmentation you want to see. You can then explore the data using NeuroGlancer. So this is an in-browser visualizer. We also have 3D visualization capabilities. So if you just double click on a segmentation, it'll make the mesh appear, and you can see that rendering. On the measurements page, you can query all of the measurements that have been done using the segmentations. This includes things that I showed in the previous slides, like the contact sites, volumes, surface area, and occurrence. We also have a glossary of sorts containing a definition of an example of each of the object classes that we have segmented, such as the centrosome that will pop up here in a moment. And then you can find instructions for navigating the website, other ways to visualize the data, including accessing the data through Fiji or Python, and other tools for anyone that wants to download any or all of the data. That includes the raw data, the training data, the segmentations, and the analysis. All right, so within the COSIN project team, we developed this workflow towards automatically segmenting and analyzing organelles and other subcellular structures within the cell. But ultimately, what we wanted to do is to apply this to cells in a more native environment, tissue. Also, a quick shout out to Philip, who made these beautiful renderings using a new API that he has up on GitHub if you're interested. So within CellMaps now, we are looking to expand what we're doing in the community. So we're looking to look at the same ultrastructure of cells and organelles within cells, but now within their native environment and tissue. And importantly, democratize this process by making the data sets, the tools, and all of the analyses openly available and accessible. So we began by scaling our work to a couple different tissues in a mouse, namely the kidney, and of interest to this group, the liver. We built our workflows with the scaling in mind, luckily. So this meant that we didn't have to change much to handle the increase in data set size. So I encourage you to explore these on Open Organelle, and you can browse through all of the data sets and the segmentations there. And just to sort of give a frame of reference for how much scaling was involved, you can see here the single cell that I've been referencing all along, and then the tissue, which is the same size for both of these data sets, but the tissue which is represented here as well. So a much bigger volume now. All right, but ultimately, if we want to drive technology development, it's really exciting and helpful if we have a scientific question. So towards this end, we teamed up with Daniel Feliciano here at Janelia. His lab focuses on the phenomenon known as metabolic liver zonation. So as you will know, the liver is structured as these hexagonal lobules with the central vein at the center and portal vein that marks the corners of each of the hexagonal lobules. The acinus is this territory from the central vein to the portal vein and is really quite interesting, especially to a non-liver biologist and a physicist turned cell biologist. So while the cells are in theory the same across this acinus, the oxygenation, therefore the environment that the cells are experiencing and all the stuff inside is really different. So Daniel's specifically interested in the mitochondria and the ER interactions across this acinus. So shown here is a fluorescence image that he collected of a lobule with the mitochondria and the actin labeled. And the central vein here for orientation is in the center and portal vein at the corner. And what's very exciting is we now have this beast of a fib-sim volume of this acinus. So for reference of size again, so that you can appreciate what kind of scaling is taking place here, I'm showing here again the single cell from the COSIM workflow that I've been describing up to now, the liver data set that I was showing that's now up on Open Organelle, and now this whole liver acinus that has been collected. And just to give an explanation of the volume size of this. So on hard disk, the single cell is around 100 gigs of space, and this whole liver acinus is about 70 terabytes. So this has required a lot of scaling, both in terms of just how we handle the data sets, but also in terms of how we predict into it. So we're very excited to start mining the data set with Daniel and applying all the tools that we've developed to date with these single cell data sets that are now scaled into tissue, and use them to help him answer exciting biological questions, such as how the organelles change across the metabolic gradient of the liver acinus. So stay tuned for more. We should have updates in the near future. All of this work was really made possible by this incredible collaborative team effort, and it really would not be possible without the larger support of the administration, of labs, and support teams here at Janelia. And lastly, a shameless plug that we are looking to expand our teams, and we're looking for excited individuals to come and work with us. So you can find a list of all the current job posts, or just really feel free to reach out to any one of us for more information. And with that, I will finish.
Well, thank you, Dr. Weigel. That was quite amazing. Are there any questions? I forgot to mention there can also be like offline questions later through the chat, but it's a bit late right now. Maybe can I ask a question? So you can actually see the ribosomes, no, with your technology. Is there a way to link it with the specific sequence?
Yeah, so this would require a multimodal approach, right? You would need some sort of, I'm guessing, some sort of fluorescence marker, and then do a correlative light and electron microscopy imaging. But yeah, this is definitely a realistic experiment. I don't know if anybody is necessarily working on it per se, but it could be done.
Okay, now that sounds, from a virology perspective, I'm always interested where are they just randomly the viral transcript versus the host transcripts? Could you distinguish those? Yeah, it's very beautiful. Sorry, Dr. Arias.
Aubrey, very elegant work. So now that you're introducing the liver world to the brave new world of, I guess it's cellular ultra structure, how might individuals who are doing studies involving, say, liver physiology or molecular biology, all the things that we've heard of, for example, today, how might they facilitate some collaboration with you and the folks at Janelia, aside from being able to access the information that you were accumulating and creating a library that's really going to change things enormously. But in a more immediate sense, is it possible for people to establish collaborations or to take advantage of this technology and your expertise?
Yeah, so this is a really great question. And the best answer that I can say is it is a work in progress. So we're a fairly new team. And the FibSim group as a resource, even here at Janelia, as kind of a building-wide resource, is very new as well. So we are working on entry points for both HHMI investigators, but also the community to have a way to access that technology. So one that's already available is through the AIC here at Janelia, the Advanced Imaging Center. So they have, you can go check it out on the website now, on the Janelia website. But through the AIC, you can submit a proposal to work with this specific technology. And we are open to collaborating, with folks through the AIC in that manner. For more direct collaborations and, potentially like bigger projects, there's also the Visiting Scientist program here at Janelia. But ultimately, what I would say is just reach out to any one of us and we will find a track for that to happen.
And I have one more question from Sammy Wang online. Could you annotate the different cell types in the liver based on these images?
Yeah, most definitely. So some of the things that we're very interested in is not only classifying the different cell types, but then using like generative AI to explainable AI to see if there are things that we beyond just what is obvious to us that could help us to identify things as well. If the networks can explain to us what it thought was important as sort of a hypothesis generation approach.
Well, thank you. If are there any other questions, either online or from the audience? Sangeeta, did you have a question or?
I did. Can you hear me?
Yeah, I hear you. Yeah.
Yeah, it's actually more of a plug than a question, Aubrey, just some things that we're all interested in seeing, which I think you now could maybe see would be the fenestrations in the sinusoidal endothelium and how those vary along that axis. We're all interested in those structures and they're really hard to study in culture. So you have a real ensemble view. And another thing that's been really interesting lately are these idea of condensates, cellular condensates, which are sort of membrane-free organelles. And I think many of us in the liver community are thinking about how that applies to hepatocyte biology. So it seems like you could see those too.
Yeah, with the liver fenestra, I think, yes, we're definitely interested in looking at that and trying to, make sure that we clearly annotate them across the data sets. And then with the phase condensates, absolutely, they're incredibly interesting. I think, even beyond just the liver, but it is an area I think that we're pretty interested in here at Janelia, but definitely requires some R&D in terms of knowing how to identify them in the EM data alone. But it is, I guess, another work in progress. Yeah.
Very exciting. Thanks.
Thanks.
Thank you. Thank you so much, Dr. Weigel. So now we're going to move on to the sole micro talk here. So it will be given by Dr. Schneider. He received his PhD in biochemistry at UMDNJ, studying the retroviral integration in Monica Roth's lab. And then after that, he joined Charlie Rice's lab at Rockefeller, where he focuses mostly on identifying novel interferon-regulated transcripts and a new project that he will talk about today. He will talk about an exciting new system to study hepatitis B virus. And the title of his talk is an RNA-based system to study hepatitis B virus replication and evaluate antivirals. So, Bill, go ahead.
Okay, thanks, Ipe. So before I start, I just want to take a second to thank Sangeeta, Heather, and Wolfram for the opportunity to present this, and thank all the people who organized this symposium today. So to get started, as many of you know, hepatitis B virus is a small DNA virus that chronically infects about 300 million people worldwide. And studying this virus in cell culture is challenging because unlike the situation in humans, it's not very infectious in our culture systems. What that means is that we typically have to add anywhere from 100 to 500 particles per cell to initiate an infection. And this ends up giving you a very high background of input material to start with, which requires additional purification and steps downstream. So in order to overcome this challenge, we asked the question whether or not we could initiate the life cycle of the virus, starting with RNA rather than with plasmid or with the virus itself. And the idea here is that we know that the DNA genome is reverse transcribed from an RNA template. So if we could make this RNA in vitro, transfect this RNA into the cells, could we actually get reverse transcription to occur? So what we did here was we made two versions, a wild type version and then a version which encodes a dead polymerase. And as you can see, without any purification steps or anything, we just lyse cells and take the total DNA. And by performing qPCR, we see that we get about a 5-fold signal to noise with almost no detectable background signal from the polymerase dead control. So this is working, and we wanted to ask then, what can we do uniquely with this system that other cell culture systems can't do? So the one application I wanted to show you today is what we've been working on for using this system in order to predict drug resistance in cell culture. And this is a bit of a challenge because without the ability to select a good infection or spreading system, it's very difficult to do this in cell culture. So we either have to use more complicated systems or most likely humans. And that's a little bit late in the pipeline. So what we do is we start not with just one RNA, but we start with a diverse library where we make this variant using a deep mutational scanning approach. So there's a lot of diversity inherent already in our RNA population. We transfect this into cells in the presence of drug. And then what the expectation is that only those variants, the rare variants in the population that are resistant to the drug, will then undergo reverse transcription. And then we can read those by Illumina sequencing. So I'm just going to show you the result from one of these here, which is a reverse transcriptase inhibitor. And the way I'm showing is in blue is an AlphaFold prediction of the hepatitis B virus reverse transcriptase domain. And here, just for orientation about the active site, I've included a double-stranded DNA primer. And everything that you see colored in these pink spots are all positions where variants were enriched in the presence of drug. And these include many of the known all of the known resistant mutations that occur in patients and several others as well. So this is one application. There are many others, but we're really excited about this. And I just thank you for your attention. And before I end, I just want to say very briefly, there's a lot of people involved. I don't have time to go through it all, but I just want to thank Charlie for supporting this work from the very beginning, and also Yingpu Yu, who I've been working with on this from the very beginning and really was the main driver in this project. So thank you.
Thank you, Bill. Are there any questions? Well, I ask you questions every week. You teach me every week, so I'm not gonna bother the group with my ignorance. Are there any other questions from people for Bill? Okay. Well, with that, then Sangeeta, I'm going to give it back to you for the announcements.
Great. Thank you, Bill. That was terrific. I think now, I'm not sure if I'm the transition person, but I'm happy to be.
I punted it, but I think there's a break now, no?
Yeah. So I think we're going to take a break now until 1 o'clock. Grab yourself some lunch, bring back, if you want to do cocktail for cocktail chatter, we won't tell anyone, and join us back. Oh, great. There's like 25-minute break. So see you at 1 o'clock, and thank you, Ipe.
I just wanted to say that I thought that the program this morning was absolutely incredible. One of the most exciting presentations I've ever heard. It really got, I'm sure got lots of people excited. That was the main goal. But you did a wonderful job in putting such a program together so far. Great.
Well, we agree. I think we're this is a public conversation, just so everyone knows. But I saw a couple mentions of cell polarity win, and I thought you were gonna. I saw AMPK and the cell polarity in Hans's talks. I thought that I thought of you. But yeah, it's been a wonderful morning. I agree.
Oh. How are we doing?
I believe we're waiting for Trump.
Yeah, maybe we can get started, and I'll send her the link and make sure she has it.
Sure. Hello, everybody, and welcome to this roundtable discussion. Actually, it's going to be, we're going to ask you to use your imagination a little bit. This is cocktail hour, virtual cocktail hour, where it's my absolute delight to be hosting. My name is Nicola Emma Mopus. I am the editor-in-chief of Med, Southwest's flagship medical journal. The focus of our journal is to build a strong bridge between the bench to the bedside. I have with me Joa, Sangeeta, Wynn, Wolfram, and soon to join us, Tram. And I'm going to let them introduce themselves very quickly. And the idea behind this is that we're just bumping into each other at the cocktail bar, and we are going to be talking about what's on our minds right now without necessarily having a specific idea how this is going to go. So thank you.
Great, so I can start, and then I can go next to Wolfram. So as you all heard this morning, I'm an MD PhD, a physician engineer based at MIT. So my perspective at this cocktail chatter is one of someone who's an educator who runs an interdisciplinary lab and center, and someone who's starting companies to help bring discoveries forward on the path to patients. Wolfram, you can go next.
Yeah, I'm Wolfram Gössling. I'm a physician scientist. I'm a liver doctor and a liver cancer doctor, and I'm also the chief of GI here at Mass General Hospital and run the physician scientist training program at Harvard Medical School. And so I come here with both our experience from trying to understand liver development, regeneration, and cancer, but also taking care of patients with chronic liver disease and often incurable liver cancer, and how we can bring new, badly needed new therapies to the bedside, as well as mentoring and training the next generation of scientists who need to help us with that.
I can go next. So I'm Ajua Nyaniyaboa. I am a gastroenterologist and a health services and implementation science researcher. All of my work is around health equity in gastroenterology, particularly around colorectal cancer screening and inflammatory bowel disease. So I'm here to bring the equity perspective.
And Wynn does not need introductions, I believe.
Indeed.
So I'm Wynn. I started out my career as a liver doctor and investigator, sort of morphed into a bit of a cell biologist. So the cell biologists think I'm a great liver doctor, and the liver doctors think I'm a great cell biologist. And so that's the whole purpose of the whole thing. We're building a bridge between these various disciplines, and that's what motivates my interests and goals.
So what's on your mind these days, Wynn, in terms of bridging the gap?
I'm sorry, was that a question to me? I didn't follow it.
I'm asking you, it's good to see you, Win. So what's on your mind these days in terms of bridging the gap?
Well, okay. The other symbol for this bridging the gap is sort of converting silos into bridges. And so my feeling is that so much of the science and the clinical aspect is sort of represented by silos, isolated structures. And as the science incredibly advances, as we received a very elegant sample of this morning, that gap gets bigger and bigger and bigger. And it's a huge challenge from the educational standpoint, the training, trying to attract young people into the field of excitement. And I think that is from my point of view, the single largest overriding issue is to get people to be able to communicate across disciplines. It's very easy these days to become sort of like the pizza pie, where you know your own little slice, but you can't talk the same language or even communicate sometimes with people of other disciplines. And that certainly is true in the gap between more basic research and more applied research. But you don't have to be an expert in any of these different things. But we should have mechanisms so that people can understand enough to realize that we're all asking the same questions at the end. And by communicating, I think we can make much greater progress than if we sustain this silo approach.
I totally agree. And Sangeeta, what's your perspective on this?
Yeah, I was going to say, I agree. And maybe for the trainees watching, I wanted to tell a little story. So I was starting a company based on the idea that we could support hepatocytes outside of the liver with a little transplant. And as I started to think about, what clinical transplant really looks like, I realized I hadn't been in medical school in 20 years. So I called up Wolfram and I just said, hey, can I come shadow you in transplant clinic? And he, very generously allowed me to spend the day, and it really, really shaped my thinking about how these technologies might be useful, because I didn't realize the ordeal that transplant patients have to go to get on the list, and all the reasons they might not be on the list. And it sort of fed back into how we think about the technology that we're developing. Wolfram, I don't know if you remember that day.
I do. I do remember that very well. And it was great to have you. You are welcome back anytime.
I think we need more of this. From my perspective as a journal editor is I find that language can often be a barrier. As part of the silos, we speak somewhat specialized jargony language. And something that I'm certainly trying to work at my journal is to improve communication. And we have a context and significance paragraph on our papers where we try and explain what the research is about in layman's terms. I think Joanna would certainly agree with us on this about.
Absolutely. I mean, I feel like the problem of silos is a big one, in that a lot of us are doing work that's, somewhat similar, a little bit different, but similar kind of overarching topic, but we're all working in our own little areas. And I think the question is, how do we connect, and how do we find people that we might not know? So we can't send that email saying, can I come and shadow you? But how do we find ways and bridges to connect people who don't already know each other, who are doing work that's similar, that can, help build that bridge? And so That question I actually don't have an answer to, but I think it would be interesting, if we looked into that in the future.
It's, when you know, but when you don't, and you don't really know how to start. But I think symposia like the one that we have here are an excellent starting point. And if anything, from the diversity of questions and conversations we've had ever since this morning, this shows the general interest and people being willing to reach out across the bridge.
Well, I think that, when you ask what the etiology of a disease is, it unless it's a specific bug, I guess, it's a very complicated business. And so the etiology of the silo is also complicated, and it involves education. You can say it even goes back to, high school education. Where did we ever come up with the idea that people have to be in their late teens before they're ever exposed to any education dealing with the human health and its mechanism? Why do we still do that? I mean, we have high school students who can put DNA together very simply, and they're very interested in learning, about human disease. And I think that's part of it. And that same educational thing gets arabesqued up into the medical school, certainly into the graduate programs. We've been trying to urge that there be graduate programs where PhDs have an opportunity to see patients, not in a big formal way, but to as one of our students said, it puts a human face on a disease. It makes sense if somebody's working on a certain disease as a basic scientist to have some idea as to what that disease is like in human beings, what they feel, what they know, what are the real problems. And that's the kind of bridging that is, I think, fundamental to, aside from linking technologies.
I think Wolfram would definitely agree with that.
Yeah, no, when I do agree, and I think the need for both to attract people and educate people as early as possible. And I don't have it here, but in my research office, I have a picture of the GI tract drawn by my youngest son when he was in 2nd grade. And so people do get exposed early on. And I look at this too, whenever I forget where the stomach is. But, I think what we see here in this symposium is also what I think is so badly needed. Is what we saw executed so well during the COVID pandemic, right? Where people from very different fields of both research, clinical, the clinic and operations, and all these different fields came together under pressure fast. And so the good thing about that is we can actually break down these silos really easily when the whole humankind is threatened. I think we just sometimes are complacent, and when the threat is not as imminent, right? We have now 80 or 90 million people in this country alone with steatosis of the liver. That is a silent and non-painful disease, and it may not be a disease at all. It's unless it progresses to NASH. And so maybe, right, this is not completely at the forefront of our thinking, both in the lab, in the clinic, and in Washington, D.C. at health insurance companies. And yet the long-term impact and outcomes are just as detrimental. And so, for us to both acknowledge that there are things that we may have not thought are urgent and worth our attention, yet are truly a sort of a long-term threat to our health is important. And we've shown it, we can do it. And so we should take that optimism and do it in other areas too.
Yeah, I Wolfram, my pressing cocktail question is why we have so many drugs for NASH, or that there's a lot of hope, and then they promptly fail. There's a lot of excitement around phase 2, and then once they get to phase 3, they just don't pan out. What do you think is going on here? What's your thought on it?
Yeah, no, I think that is an interesting observation and phenomenon, right? I mean, and I think it's multifold, and certainly not for lack of trying, for lack of big pharma doing it. But one big missing aspect is, I think, whether we are going after the right biomarkers or outcomes. All these studies are obviously expensive. Liver disease is slow, so by definition, if you wanted to see an outcome that is statistically significant or FDA approvable, you got to follow your patients for a long time, and that is often contrary to both what interests are in pharma, but also for us, right? We don't want to wait 5 or 10 years in the clinic to have a new drug. So figuring out what the best prospective biomarkers are, or endpoints that can be used as surrogate endpoints predictive of long-term outcomes, I think is key. I also think, and we've already seen some of their work this morning, and we clearly see this in the area. I think liver disease, especially metabolic liver disease, is an area on the move. It's dynamic. It will bring more results, and to really see how we can find new targets and new mechanisms that will have implications or are druggable, I think is definitely one of the next steps too.
Definitely, and equity is something that's really something that we all think about, especially in this room right now.
Absolutely. I feel like there's so many factors and so many areas in where we need to be thinking about equity. I think, one place we can be thinking about equity is in terms of who has access to the drugs, what drugs are being covered, who's not getting the medications because of coverage issues and uninsurance or underinsurance. I think another big factor around equity, especially in liver disease, is around liver transplant. And so we know that liver transplants are literally life and death. It's literally a life and death situation. And we see disparities actually across the continuum. So, Black patients are underrepresented on the wait list compared to their donor service area, compared to white patients are overrepresented compared to their donor service area. So I always wonder, how can we change the systems in which we're evaluating patients, in which we're listing patients, to make sure that everybody's getting this life-saving treatment? This is something that I think about and wonder a lot, and I would love to hear other people's perspectives.
Well, I'm just curious. This is something that I don't know a lot about, but at the university, for sort of faculty gender diversity, there were sort of best practices that were discovered, 20, 30 years ago, where you would sort of very systematically, objectively evaluate faculty recruitments. And that took a lot of the sort of unconscious bias out of the process. And so instead of sort of like a hallway whisper about oh, this is a star, we should get him on our team, there were, formal ways of assessing candidates. That's just one of the tools that's been put in place and really helped with that process. And I'm curious, are there attempts at taking unconscious bias out of listing folks on the transplant list?
Well, at the NIH, there's a huge emphasis in this area that's going on now for almost 2 years. It takes many different levels, including interviews, annual testing that everybody has to take, and so forth, to make an issue apparent and real, whereas before it was all buried. And it's also reflected in the increasing number initially of women, but now it's people of color, it's all sorts of backgrounds and so forth, who people applying for all kinds of positions at the NIH, from scientists to postdocs. So there is clearly a movement that is being encouraged by NIH policy. It has a long way to go, but there's an awareness which before certainly didn't exist.
I agree. It's also a time and a cost issue, right? We also have to factor this into question whenever we think about how to increase access.
And also, what does access mean, right? Access can mean so many different things. Access can mean being close to a transplant center. Access can mean coverage. Access can mean, not having to wait months for an appointment. And so I think it's also important to be like specific when we say, because a lot of times we say access broadly, but there's so many different barriers that people face.
It's teasing. I mean, what does access mean? And the transplant and who has access or who's eligible, who has the time.
Right.
So many issues.
So when it comes to patient-related access issues or transplant, as you brought up, Adjoa, I think the more objective criteria we have, because we used to do the eyeball test, right? I mean, a surgeon and a medical doctor, a transplant doctor would walk into the room, and that's what we would do. And we'd say, this patient looks sick, so we need to get them listed for transplant ASAP and go through all the process urgently. I think that we have more objective scales in the last few years on frailty and sarcopenia. And so these are predictors of outcomes that are more objective. And so the more that we can do that in every field and in the way that we assess faculty and the more objective criteria we have, the less there's going to be that inherent bias that we don't even realize that we're building into the system when we do this in a lot of different ways. So yeah, I take that.
I mean, just to follow up on what Adjoa said and Sangeeta, I think it's obviously not easy to increase diversity and equity and inclusion at a university setting, but it's a pretty monolithic and monodimensional area compared to, I think, what we're talking about, the healthcare system. And I think, as Adjoa said, access can be so different. And as Tran said, yeah, we can probably score and scale and measure some of the clinical and clinically objective criteria for transplant. But if these patients never see us because they can't get to us, if they can't pay for the $35 parking that it takes to be at the transplant clinic, if they can't take off time from their work to be waiting in a wait room and then be seen, I think for us to acknowledge that all these areas need to be highlighted and ultimately addressed, and that is painful, and that both to acknowledge and can very hard work to remedy. But I think the first step is, as we do here, to acknowledge that these bringing new therapies, bringing cure, bringing transplant to people is great, to the bedside is great. Bringing it to more people and more bedsides is really the goal.
I agree. And one more thing on this topic is that we need to also, I think, to know what the problems are, we have to track and follow the data. And so whether it's, faculty diversity, whether it's who we're transplanting, who's coming in and who's getting transplanted. And if certain people aren't coming in, how do we reach them? How do we meet them where they are? And same thing with diversity of faculty. Why are some people applying, some people not applying? Maybe it starts early on, maybe they're not getting in at all. We need to look higher up in the pipeline to figure out where we're losing people. And so I think having the data allows us to act on it.
I'm curious, do we have national databases that help us track this data?
So we do. Actually, our faculty member, Dr. Andrea Reed, did work on that, sadly, 15 years ago, and highlighted those things. And so, as Adjoa said, we have to track more data and better data, and apply to today's world. But we know this. And we so far we haven't addressed this all, and I think Adjoa, you can talk more and in more detail and more expertise about this, but we have obviously more databases that would allow us to track this or set it up.
Yeah, I mean, one thing that we talked a little bit about, we had a pre-call as a group yesterday, is sort of about the pace of change, and we talked about how long it's been since we had HCV medicines. And where we are today. And I think when we heard that Francis Collins was at the liver meeting, right, this week, did you want to relay what we think he said, or do we want to talk about that a little bit?
Well, I wasn't there, and I've been trying to find out from my colleagues what he said.
Sam, were you there?
They weren't there either, so
No, I
don't-
wasn't at his actual talk, but I know that we've been talking about Hep C global elimination, and I think now the U.S. is really focused on that because we came out with the societies came out with an elimination plan. We had a cure, right, for years now, and why wouldn't we be able to do this? And there's some states like Louisiana that had a deal where they made a deal with a pharmaceutical company to like have a one cost for all of the Hep C patients they could treat in their entire state. Like you don't have to pay per patient, you pay one fee and every single patient you could treat would be eliminated. So they really tried to do this elimination plan in the state of Louisiana very successfully. So that's kind of thinking out of the box instead of like patients having to apply for insurance and doing the Hep C testing and all of this stuff. It was really about test and treat as quickly as possible with reflex testing, not having the patient have to come back for a second appointment, like making the whole process as simplified as possible, and then working out sort of the reimbursement strategy so that it's maybe a more global strategy instead of one for one. And that seems to be part of the elimination strategy that has been discussed globally, but also now specifically for US is what I think is the focus for, I think, Dr. Collins and many others, stakeholders in Hep C to get to elimination. Because we had a goal of, I think, 2030 for Hep C elimination. And due to COVID and other things, and access, we're really not able to meet that goal of global elimination or US elimination by 2030. That was kind of the initial goal set.
I think one of the great things is having reliable ways of testing and diagnostics, and having things that are easy to implement, to access, and be able to basically diagnose and treat the disease based on that. And Sangeeta has done a lot of work in this area.
Yeah, I mean, I think it's interesting being in the diagnostics field. It's been a tough field actually, because it's a field where everybody wants a diagnostic and they want it, quick turnaround and low cost, and so that's great. But if you're developing a new diagnostic, then it's hard to bring forward, because if it's going to be reliable, you have to do large studies, and it takes a long time to do that work. So, I think in some ways COVID really shone a light on the power of diagnostics and diagnostic innovation. Like, what does it mean to have point of care testing and self-reported monitoring? And I think it forced all of us to think differently about that. And I'm curious, Trim, how do we do HCV testing today? Are there any innovations there?
Well, we're trying to do the test and treat, where you get a very rapid result from your Hep C test, and you don't have to do genotyping. We have pan-genotype cures. Why are there any rules on checking genotyping, which requires another expensive test, right? So it's just really about that one test, start the treatment, get the result, you don't have to wait and get the result. So it's really about simplifying that pathway for the patient. So I think Hep C is a pretty straightforward thing. I think for NASH, it's going to be more challenging. We're in the diagnostic NASH space, and we're really trying to do something. And sort of my topic that I was going to talk about today was this new NASH and NAFLD nomenclature. Things are going to change. We're not going to call it NAFLD or NASH anymore. So newsflash, we're going to probably be calling it steatotic liver disease as an umbrella. And then we're really going to try to define subgroups based on their metabolic profile or their alcohol use or other things, so that we can do better testing, have better diagnostics that are really going to be focused on the type of patient that's going to have the biggest sort of risk of progression. So I think in the diagnostic space, in NASH, we're really going to see this change with this sort of better characterization of subgroups and nomenclature counts in this. So this is being worked on right now.
So do we still have to rely on biopsies?
Oh gosh, well, that's a very big area. Well, I mean, regulatory agencies and, those are the endpoints that are being required. But we would really like, everybody in the field wants to get away from biopsies. If you've ever done or had a liver biopsy, it's really kind of scary to have that big needle. I know the risk is really low, but it's still not ideal. So I think everyone's trying to move into biomarkers away from liver biopsy, but we're not quite there yet.
So
And
I
To highlight that, just to highlight that conundrum a bit more, we obviously in the field historically think it is the gold standard. If you take 10 pathologists and show them slides of NASH livers, they only 7 will always agree on what they see. And this is, I don't think, a statement of the quality of the pathologist, but really about the trickiness and the variation that you see in the tissue. On the other hand, many of the tests we use every day in the clinic, such as the immunotransferase levels, ALT and AST, that have been in literally ubiquitous use for decades, for 55 years plus, measure injury, but in a very imperfect and non-specific way. And in between, we don't have much that is, there's currently, as Sangeeta highlighted, validated and in ubiquitous use to be a factor, and as opposed to other diseases and other studies where ultimately endpoints such as hospitalization or death or transplant are used. This is obviously something that would be prohibitive here if you wanted to introduce new treatments faster.
That's
so I think that, I'm sorry.
Go ahead, Wayne.
Although our emphasis today in the program and everything else is on really the incredible advances in basic biological and engineering sciences, from the standpoint of liver and the patients, we shouldn't lose track of the fact that the vast numbers of people with liver diseases are the results of largely lifestyle activities. They're related to diet, they're related to exercise, to alcohol. I mean, these are major public health areas which we don't deal with in our laboratories, but unfortunately our society doesn't deal with very effectively either. And so somewhere, that's one of the bridges that has to be made too. We can't remain in our own bigger silo to the exclusion of the fact that most people with liver disease have it related to these lifestyle issues. And there are some, predisposing things that make some people more susceptible, PLP3 perhaps being the major one that we know of. But our focus therapeutically should really, I think, be on detection of impending problems and dealing with that, rather than, at least intellectually, focusing most of or all of our efforts on the more advanced forms of liver disease that we...
Prevention is obviously better than cure. And I think this is going to be a perfect time for us to wrap up so that we can move on to the rest of the very, very exciting program. I do wish we had more time, but I hope to the trainees and all the attendees to give you the sort of discussion that goes on in person at the bar. And to be fair, this sort of conversation does tend to go on for many, many more hours. But thank you all. Thank you for your insight. And back to you.
Thank you, Nicola. We're going to transition now to the session chair, Letitia Kawasula, to take us into the afternoon sessions.
Hi, good afternoon. I'm Letitia Kawasulu. I will be the chair for this next session. So we will have 2 talks and 3 micro talks. And it is my pleasure to first introduce our first speaker, Dr. Elizabeth Speliotis. Dr. Speliotis is a professor of gastroenterology and computational medicine and bioinformatics at the University of Michigan. She's the director of precision medicine for internal medicines, where she's building biobank, analytic, and training resources to use genomic data for subtying human diseases and matching them to specific treatments. She's an internationally known leader in the genetics of human obesity and NAFLD. And she has identified and characterized hundreds of common genetic variants that associate with obesity or NAFLD using genome-wide association study, meta-analysis. And her talk is entitled Genetic Subtyping of NAFLD. So thank you, Dr. Speliotis, for being with us today, and please go ahead.
If you could please unmute.
Can you guys see the screen?
We can. Everything looks great, and you sound great too.
All right, sounds good. Well, I'd like to thank the ALF and Sangeeta and Wolfram for the invitation today to tell you a little bit about how our work. So as many of you know, non-alcoholic fatty liver disease has increased with the rise of obesity, and it's caused by fat deposition in the liver that can lead to inflammation, scarring, and possibly liver cancer in the liver. Its global prevalence is about 24%, and it's measured either by histology, which has a greater sensitivity and specificity, but is hard to get, non-invasive imaging, and liver function tests. And its prevalence is higher in individuals of Asian ancestry, European ancestry, as well than African ancestry. And it has become the #1 cause of liver disease in some areas of the world, and will become that very soon throughout the world. And as you all know, we have no effective medical treatments for this, making it one of the biggest unmet medical needs of our time. Interestingly, even as people become morbidly obese, many but not all deposit fat in their liver, and some will go on to get advanced liver disease, whereas others will develop metabolic syndrome and cardiovascular disease. And currently, we really have no ability to differentiate who's going to deposit fat and go on to get liver disease versus metabolic disease. And so one of the things we're interested in is trying to better understand who's going to deposit fat in the liver and go in which direction. And to do this, we've taken a human genetic approach. And non-alcoholic fatty liver disease, as we and others have shown, is a complex genetic trait. And what that means is that there's genetic variants that predispose to getting the disease that, when combined with the right environment, as you heard Dr. Arias say, will ultimately determine whether or not you get the disease. And the heritability of steatosis that we've shown is about 26 to 35%, suggesting that it can be studied at the population level. And one question that you may be asking yourself is, why use genetics to study fatty liver disease? And I will tell you that the reason why you want to use genetics is that if you can identify genetic variants that predispose to any human trait, by definition, they're causal. And the reason for that is that your genome came before any phenotype you could have developed. And therefore, if you can identify that variant and figure out how it works, it immediately tells you something about the biology of that trait, and you can understand susceptibility to getting that disease. You can actually use it directly to define personal risk. In fact, that's the whole basis of a lot of precision medicine that we're now thinking about. And you can use it to develop tailored therapeutics for that disease. And really the workhorse of this field has been something called a genome-wide association study. And the way that works is that you have a phenotype, and then you genotype people, and then you look for associations of variants with any trait of interest. And the traits that have been used in this field, include histology, but as I told you, this is hard to get. We pioneered the use of CT, and others have used MRI to measure fat in the liver, and you can use that as a phenotypic outcome. And this is has a correlation with histology of 0.92 or above for CT and MRI. And you can use more recently liver function tests, but I can tell you the sensitivity and specificity of these for really focusing on fatty liver disease are not great. They're about 0.5 or 0.6. So usually there needs to be some other sort of follow-up to hits from here to really show that they're fatty liver disease. And the other thing that we and the community have done over the years is that we've been able to scale the number of individuals that are in these studies over the years. And as you can see, we've been able to identify more and more variants that I would say reproducibly associate with non-alcoholic fatty liver disease, defined by showing at least steatosis, if not histology associations. And we in 2021 did a large-scale GWAS analysis of liver function tests in the form of ALT, AST, and ALK Phos. And what we did is we took 390,000 individuals from the UK Biobank and combined them with 160,000 individuals from Biobank Japan. That we did association studies, and then we meta-analyzed them to find, over 200 variants on average for each trait that were genome-wide significant, which is the threshold we use for reproducibility in these studies. And then we used other means to try and understand the biology of the genes and the pathways involved. And specifically, one of the issues with genome-wide association studies is that once you get an association, shown here are SNPs on the x-axis with any trait, what you see is a signal coming off, but this signal may or may not land in the gene. And furthermore, even if it lands near a gene, you don't know which gene it's affecting. Furthermore, most of the genes in the genome we don't really understand the function of. So how do you get from variant to gene to function? So we helped develop some of the tools to be able to map these variants to the genes and the functions. And I'll tell you about one shown here, which is called Depict. And basically the idea behind this is that you can take gene expression in cells and tissues, and then you can draw principal components in those, and then you can map genes to these principal components, and you can map pathways to these components. So now you can place genes that you don't know the function of into pathways, and this greatly increases your power to identify genes and the biology behind complex genetic traits. And so then you could, drop anchor around your GWAS as shown here and look at the genes. And then you could look to see if there's particular tissues that are enriched for expression of those genes. You can look for gene sets that are enriched across the hits that you have in the genome. And you can find particular genes that are part of similar pathways across the different associated loci. And you can see that when we did this for, let's say, BMI, which is another phenotype that we studied, you get the brain coming out. When we did this for abdominal obesity, you get abdominal fat coming out as the key tissue. And shown here, when we did this for ALT, you get the liver, intestine, pancreas, and adrenal, again, showing you that ALT by itself is not totally specific to liver, but really this does allow you now to have the power to really find the disease tissues that are relevant for the disease pathology. When you do look at pathways that are associated with these traits, you can see that for overall obesity, as shown with BMI, you get overeating, energy metabolism, neuronal biology. When you do it for abdominal obesity, you can get mesoderm development, cell growth and survival, and glucocorticoid receptor signaling. And when you do it across actually all the LFTs, which mostly reflect NAFLD and alcoholic liver disease in the general population, you get lipoprotein metabolism, triglyceride metabolism, and arachidonic acid metabolism, as well as some other pathways. So again, it allows us to use bioinformatics and large-scale population studies to really hone down on the key pathways that may be influencing disease development in you and me. It also allows us to hone in on particular genes, and really the top hit for fatty liver disease for many years has been PNPLA3. And in a very elegant work done by Helen Hobbs, she's shown that this might be a gain of function, which takes away a cofactor from ATGL, which prevents breakdown of lipids from lipid droplets, and thereby accumulating excess lipids in the tissues. And this is exciting because now we have a real target that we can go after. As you can see, there are companies now that are going directly towards targeting PNPLA3 for treating fatty liver disease. So really the human genetics is leading us to treatments for these disease, and in particular, precision treatments. And then when we looked at what we got out of our associations for ALT, AST, and ALK Phos, we crossed those with other liver disease phenotypes, and certainly we got some genes, 2 genome-wide significant, and many others that are implicated for hepatic steatosis. But we also got a lot of genes that affect other types of liver disease, like primary sclerosing cholangitis, alcoholic cirrhosis, primary biliary cholangitis. So, you know, liver function tests are very useful in terms of getting us to liver disease, but there needs to be more work done to figure out the type of liver disease. So to increase the power to find specifically fatty liver disease, what we did is we assembled many cohorts that had CT measured hepatic steatosis, and that comes from 14 cohorts. We harmonized the phenotype across these cohorts. And then we also downloaded 43,000 MRI scans from the UK Biobank, and then we used deep learning methods to measure liver fat in terms of PDFF in scans where we knew the result, and then tested it against other scans where we knew the results, and we were able to get this neural network to read fatty liver with a Pearson correlation of over 0.97. We then applied that to all 43,000 scans to be able to measure this in a large number of individuals. We then did association studies with a CT measured fatty liver with 1,000 genomes, imputed SNPs, controlling for age, sex, number of alcoholic drinks, as well as principal components. We did a similar analysis in UK Biobank with HRC and UK 10K imputed SNPs. And then we combined that with ICD-based non-alcoholic fatty liver disease diagnoses versus controls in the UK Biobank from the eMERGE consortium and from the FinGen consortium. And then after meta-analysis, we found 17 genome-wide significant independent variants that specifically associate with non-alcoholic fatty liver disease. And we refined this, making sure that these SNPs had congruent effects across all of these traits. This was a cross-ancestry study, and we were very interesting to see whether we can identify ancestry-specific variants, but we actually did not identify any single ancestry-specific variant. All of the variants had effects across ancestries. And also we looked for heterogeneity of effect across the ancestries, and we really didn't see a lot of difference in heterogeneity of effect. But we did see a difference in the frequency of this effect allele frequencies of some of these genes. And in fact, when you look at the effect allele frequency, you can see that it's higher in individuals of Asian and Indian ancestry than European, than African ancestry, which correlates with the prevalence increase that we see of this disease in those subgroups. And in fact, more than 70% of the variance in prevalence is explained by the genetics. We also looked to see if there were sex-specific differences of these variants, and we didn't find them in the majority of our loci, but we did find that PMPLA3 had a stronger effect in women than men, and TM6SF2 had a stronger effect in men versus women. We then looked to see if we could show, you know, there's a lot of controversy about, you know, whether fatty liver causes cardiometabolic disease and other diseases versus vice versa. And so one way to test that is to see if genetic NAFLD can cause cardiometabolic disease versus liver disease. And we tested that using something called Mendelian randomization. And when we looked at whether genetic NAFLD can cause these diseases, the only significant effects that we found were on fibrosis and cirrhosis, as well as esophageal varices. When we did the reverse and looked to see whether cardiometabolic traits were causal for development of fatty liver disease, we only found that waist circumference and BMI were causal for non-alcoholic fatty liver disease using Mendelian randomization methods. We then looked to see whether or not we can take these 17 genome-wide hits and use them to predict or risk of having NAFLD, cirrhosis, and HCC in an independent cohort. Here we have the Michigan Genomics Initiative, which is a hospital-based biobank of over 50,000 individuals where we have genotypes as well as ICD diagnoses. And what we did is we took our 17 genome-wide significant SNPs, we weighted them by their effect size, and then looked to see if individuals by their decile, quintile, or percentile of risk associated with NAFLD cirrhosis and HCC and MGI. And what we found is that this polygenic risk score was able to identify 10% of individuals that had an odds ratio of greater than 2 for these diseases, 5% of individuals that had an odds ratio of greater than 3 for these diseases, and 1% of individuals that had an odds ratio of greater than 4 for these diseases. So this is very exciting because, as you know, it's very hard to identify risk in individuals. This isn't like, height and weight and BMI that we can see on the outside and measure. This is in your liver. And so having biomarkers or something to be able to identify who's at risk is very useful. And in fact, to be able to have a biomarker that can identify individuals with odds ratios of 3 or 4 is very exciting because those are clinically actionable. We also were interested in seeing if we could identify subgroups within our 17 genome-wide significant hits. So what we did is we took these single nucleotide polymorphisms and we looked to see what kind of effects they had on multiple cardiometabolic and other related traits in the UK Biobank. And their effect is in red if they have a positive effect, their effect is blue if they have a negative effect, and then we use k-means clustering. And you can see that these break down into 7 subgroups. We then took those 7 subgroups, we again made a polygenic risk score, and then we looked to see how they affected liver as well as cardiometabolic traits. And what you see here is that all of them increase the risk of steatosis, which is not surprising because that is our main outcome in our study. And then some of them increase cirrhosis, whereas others don't have as strong of an effect in cirrhosis. Some of them increase risk of LDL, myocardial infarction, serum triglycerides, and HDL, whereas others decrease that. And some increase diabetes and BMI, whereas others decrease it. And the reason why this is exciting is because, as I told you before, some people deposit fat in their liver and go on to get liver disease, whereas other people get metabolic disease. And previously, we had no way of identifying these. But now we can use these as biomarkers to identify subgroups of individuals that have very specific patterns of which direction they're likely to go. And this pattern differs from the epidemiological overgeneralization that we generally use, where fatty liver is associated with increases in most of these traits and a decrease in HDL. And not only can we use these to identify these subgroups, but now we can link them back to genes that can be targeted to treat people in these subgroups. And not only can we identify these targets to treat them, but we can also look to see what would happen if you sort of mismatch the treatment and the risk group. So for example, if you were to reverse the steatosis in this PMPLA3 group, you would increase your LDL, MI, triglyceride, and HDL. And if you gave this drug to somebody in the insulin group, you would double increase your risk of LDL, MI, and triglycerides. And so this can also be very useful for telling us a little bit about some of the side effects that some of these treatments will have if they're mismatched to the real cause of the disease. In addition to that, when we looked at these subgroups, they kind of fell into different biological subgroups. One group of these genes affected lipoprotein output. Another group of these genes affected recycling of lipoproteins back to the liver. So you can think of these variants as likely causing fatty liver by increasing lipoproteins and their substrates in the liver to cause disease. Some of them affected BMI, like FTO, which affects browning of adipose tissue to lead to obesity. Others affected lipodystrophy and insulin signaling, either outside of the liver in adipocytes or in the liver, like SRABP1 and GRB14, to cause fatty liver. And others affected, absorption of fatty acids from the gut, like MTTP. And overall, I would say that this subgroup falls into this obesity, diabetes, insulin resistance group. And we don't know all of the details of how this leads to fatty liver. Some ideas are that from the insulin resistance, there might be increased fatty acid delivery to the liver to lead to increased fatty liver. Some have shown that direct insulin action on the liver increases de novo lipogenesis, and others have shown that insulin has other effects, like affecting cholesterol metabolism, that can lead to liver damage. So it's not clear how this leads to fatty liver and advanced liver disease, but these are some of the possibilities for how this subgroup may cause fatty liver. There's another subgroup that affected glucose to triglyceride transitions, and this may lead to de novo lipogenesis, to increase fat in the liver. And there was a final group whose characteristics are to decrease serum triglyceride. And some of the members of this group have been shown to increase phospholipids and may increase other intermediate phosphoproteins to be able to cause some of the pathology in the liver. But really, this data helps us now to basically understand the pathophysiology of fatty liver in a much better way than we have previously at the population level. In addition to that, as has been mentioned previously, there's been a lot of challenges in this field about getting treatments to patients and having all of them respond. And that may be due to the heterogeneity of the disease. And with these new categories, potentially we may be able to better link treatments to their real cause. For example, the most obvious one would be targeting PNPLA3 for people that have liver disease based on PNPLA3. Others may be to use a thyroid agonist to decrease the lipoprotein sort of load in the liver and the excess lipids in the liver. Others would be to use some of the new diabetes treatments, such as SGLT2 inhibitors and GLP-1 agonists to treat individuals that have fatty liver more based on their obesity and insulin resistance. Other directed treatments could be the use of lamidopide for people that have disease based on MTTP biology. Others would be to use some of the de novo lipogenesis treatments for the categories of a disease that may be going through that pathway. But you can see how, with new knowledge of some of the subgroups, we can better match the treatments to the disease and hopefully increase effectiveness and decrease side effects. But we're also interested, as Dr. Arias says, I'm not only knowing the genetics, but really what are the environmental factors that are really causing these diseases to take off. And here in a study that we performed in 2019, what we did is we took genetic variants and then we looked to see what physical and serum traits multiplicatively interact with those to cause fatty liver and what we found was that PNPLA3, for example, interacted with serum insulin, glucose, triglyceride, and BMI to increase the risk of fatty liver at the population level. But most interestingly, when you do conditional analysis, it was really the insulin that was most causal for fatty liver. It was not glucose. And this was surprising. And the reason is, for a long time, we've known that people with diabetes have an increase of fatty liver. And most people think that diabetes is caused by increased glucose and glucose is the pathogenic agent. But this would suggest that glucose is actually not the most causal. It's probably something about the insulin resistance. And again, we don't know what that is. Is that increase of fatty acids? Is that direct insulin action on the liver? We don't know. But at least these studies would suggest that that is more causal and we should be focused on reversing that. The other thing that this was important about this study was that a lot of people say, well, GWAS, these variants, they don't have big effects. But really here we excluded diabetics. And so anybody in our study had pre-diabetes. And most of the people don't know that they have PNPLA3, but really 5% of the general European population is at risk of having PNPLA3 and pre-diabetes. And just PNPLA3 and pre-diabetes account for about 8% of the population variance in steatosis. So really, the genetics plus pre-diabetes does account for a reasonable amount of risk in the population. And right now, these patients are not identified. We are not doing anything for these patients, but really, they are slowly probably causing damage to their liver. And over time, these are going to show up as your cryptogenic cirrhosis individuals to our liver clinic. So this really suggests that we should be using some of these genetics and other biomarkers to identify these people and bring them to medical care like we do for colon cancer or other things. The other thing that this shows is what it means to have a gene-environment interaction, and that's shown here on the left. And what this means is that if you have the high-risk genotype and high insulin, your risk of getting NAFLD is greater than 90%. If you have the low-risk genotype, and low insulin, your risk is 20%. But if you decrease your insulin resistance, actually the people most at risk benefit more than the people less at risk. And this is exactly what precision therapies are all about, right? It's finding the high-risk individuals and then reversing their risk factors to be able to give them the most benefit. And so that's what these studies would suggest. And to try and test that, what we are doing right now is we are enrolling individuals from our biobank that have these genotypes. We are putting them on a keto diet and really testing whether or not we can use the reversal of some of these environmental factors to more benefit the people that are most at risk. So finally, it'd be great if we can identify not only variants and biomarkers that, tell us about prevalent disease, but really about incident disease. And one of the things that we've done here is we've gone back to our biobank for over 10 years, and we identified individuals that had elevated LFTs and looked to see what happened to them over time in terms of developing incident cirrhosis and decompensation. And what you can see is that over time, you can see that the individuals that are homozygous for the GG allele at PNPLA3 have a higher risk of incident cirrhosis and decompensation compared to the heterozygotes versus the homozygous wild type individuals. In addition to that, we look to see, could this add anything above and beyond sort of the clinical markers that we use today? And one non-invasive marker that's made it into the guidelines, at least for diabetes, is FIB4. This is a non-invasive marker, and individuals that have diabetes, if they have a high FIB4, basically they get referred to hepatology for further workup and possibly, screening for HCC at an earlier level. But individuals that have intermediate FIB4 or low FIB4 are not. And so what we looked at is whether or not, how PNPLA3 and diabetes affected people's risk of getting incident cirrhosis or decompensation. And what you can see is that in the red, the lowest risk group is the people that had intermediate FIB4s, diabetes, but no PNPLA3. But then if you take those intermediate risk FIB4 individuals with diabetes and identify the ones that have PNPLA3, you can see that their risk of developing cirrhosis or decompensation actually exceeds that of the individuals in the high FIB4 score. So this would suggest that just with knowing their diabetes status and PNPLA3 risk, that this would allow us to reclassify those people that right now we're not, again, doing anything for because they're in the intermediate FIB4 class, into a high risk group that really should be followed in a more intense way to help prevent some of the complications of their high risk genotype and environmental state. So basically, I hope I've shown you how common genetics can be used for precision health, and in particular, how genetics can help us to understand disease pathophysiology, define personal risk of disease, identify and risk stratify patients for further care, define subtypes and match them to treatment, and identify environmental factors that we can change to mitigate disease risk. So I'd like to thank our participants, the many consortia within which we collaborate. For the latest GWAS analysis, I'd like to thank again our participants, the UK Biobank and Michigan Genomics Initiative for allowing us to use their data, the eMERGE Network and FinGen collaborators. I'd like to thank the other PIs of the Genetics of Obesity-Related Liver Disease Consortium that I lead, and in particular, Nicole Allred that co-led this work with me. And I'd like to thank the members of our laboratory as well as our funding sources. So thank you for your attention, and I welcome any questions.
Thank you, Dr. Speliotis, for this amazing talk. Are there questions in the audience? Maybe I can go first. So I don't really have a question, but more a comment. I'm surprised that it's insulin resistance rather than high glucose levels that drive diabetes or or fatty liver disease. And so my question would be, how do you think we could target insulin resistance rather than glucose levels?
Yeah, I mean, I think something that we clinically just haven't been doing is following insulin levels. And certainly, at least in the population-based studies that we looked at, the insulin levels better correlate with fatty liver. And so that's one clinical thing that we could do sooner than later to be able to better follow insulin resistance rather than just glucose.
Thank you.
I have a question. Do we know whether people who have the PL3 and consume alcohol, are they at inherently greater risk for developing? Do we have data on that subject?
Yeah, there's absolutely very clear data. So PNPLA3, TM6, SF2, and many of the things that we have found for non-alcoholic fatty liver disease also predispose us to alcoholic fatty liver disease, which is, again, one of the reasons why we probably do need to change the name of this disease to steatotic liver disease, because that's more reflective of the biology. And then our work has very clearly shown that.
So there is a question in the Q&A section. So have you looked at these variants in alcohol?
Have we looked at these variants in?
Alcohol, yes.
Yeah, so like I said, certainly some of the genes definitely have already been associated with alcoholic fatty liver disease. Some of our newer hits, I don't know that we have looked at those in alcoholic liver disease, but certainly we should, and we'll see if all of them have an effect on that as well. I think one of the issues is going to be sample size. So, getting good, high quality alcohol liver disease data sets has not been as easy as the non-alcoholic.
Thank you. Thank you, Dr. Speliotis. So we will move to our next speaker. So our next speaker is Dr. Joseph Takahashi. He's an investigator in the Howard Hughes Medical Institute and chair of the Department of Neuroscience at the University of Texas Southwestern. His research interests are the molecular mechanism of circadian clocks and neuroscience and the genetic basis of behavior. He pioneered the use of genetics in mice as a tool for discovery of genes underlying neurobiology and behavior. And his discovery of the mouse and human clock genes led to a description of conserved circadian clock mechanism in animals. So he's the author of more than 300 scientific publications and the recipient of many awards, including the Gruber Neuroscience Prize in 2019. Today, his talk is entitled Circadian Clocks and Their Importance for Metabolism, Aging, and Longevity. So thank you, Dr. Takahashi, for being with us, and please go ahead.
Thank you very much, Letitia. It's a pleasure to be here. I want to thank Heather, Sangeeta, and Wolfram to be a part of this symposium honoring Dr. Arias. It's truly a privilege to be here. Thanks. So as all of you know, we live on a planet that has a 24-hour environmental cycle. And one way to think of our 24-hour day is it's really an energy cycle driven by the sun. If you're a plant, that would be obvious, but even for us, our bodies go through an energy cycle every day, and this has profound impacts on our overall metabolism. Now, to anticipate these environmental changes, all living organisms have evolved 24-hour endogenous clock systems to anticipate these changes. And the common motif for these clocks is a negative feedback transcriptional feedback loop that takes about 24 hours to complete. So about 20 years ago, we in the field put together the feedback mechanism. This is the core mechanism in mammals. It involves two key transcription factors, clock and Bile, that my lab discovered. And two sets of target genes, which are transcriptional repressors, the period proteins and the cryptochrome proteins. These are turned on each day. They interact with each other as well as other proteins. And in the evening, they translocate back into the nucleus where they repress their own transcription by physically interacting with their transactivators, clock and Bile. Now, the discovery of these genes was led to another surprise, and that was that the expression of the genes are ubiquitous, found in all cells in the body. And that eventually led to the discovery that essentially every cell in our body has the capability of having a 24-hour circadian clock. And so all our major organ systems, especially the liver, have their own circadian clocks that are driven by the cell-autonomous circadian oscillator. Now, about a decade ago, my lab used a number of next-generation sequencing methods to study the gene targets of the circadian clock transcriptional network in the mouse liver. And when we did this, we found that there's a very stereotyped transcriptional program that occurs every day in the mouse liver. So at dawn, the transcription factors bind to regulatory sites in target genes, but they're repressed by one of the repressors called Cry1. Once this repression is relieved, there's a major activation phase where you see occupancy of Bile and clock and recruitment of a number of co-activators such as P300-CBP. This is followed by recruitment of RNA polymerase II, a burst of nascent transcription, followed by the classic repression phase of the PERs and the cryptochromes, which turn over, and then the process starts again. One of the surprises here is at the RNA level, you can see 2,000 to 3,000 genes cycling in the mouse liver. But when we look at chromatin marks or polymerase occupancy, about half the genome or half of the expressed genes in the mouse liver are actually under circadian regulation. So this is a very pervasive change that happens. Sorry. To give you a feeling for what those targets are in the mouse liver, here's a metabolic chart of all pathways in the cell from KEGG. You can recognize the Krebs cycle in the middle, and overlaid in red are all the Bile target genes. And what you can see is every fundamental pathway in the cell has direct target genes regulated by the clock Bile transcriptional system. This picture is actually not unique to the liver. It's the same in every tissue that we and others have looked at so far, including pancreas, brain. And it's telling us something about what the clock is doing. The clock is fine-tuning metabolism in a direct cell-autonomous manner every day. So what I'd like to talk about today is the influence of time with respect to nutritional signals, and particularly in respect to metabolism. So I've already told you that the clock, controls metabolic gene targets, but metabolism also feeds back onto the clock. And so here are two examples. The deacetylase SIRT1 can deacetylate Bile, a core clock transcription factor, Another example is the nutrient-sensitive kinase AMPK, which can phosphorylate the cryptochrome protein. Sorry, my cursor keeps switching the slides. So the clock not only regulates metabolism, but metabolism can feed back on the clock and the core mechanism. Now, why did we get interested in this? There's a very simple experiment that my colleague at Northwestern did in 2009. If you take a black 6 mouse, you feed it high-fat diet, it becomes obese. But if you restrict access to the diet to only 12 hours in the day during either the light or the dark interval, the effect of that diet is completely different. The mice that eat in the daytime gain weight, but the mice that eat the high-fat diet at night are protected and do not gain weight. This finding has been replicated by many labs worldwide now. Here's an example from Sachin Pandit, the Salk. You can see down here, ad-lib fed mouse is getting fat, but if it's restricted to, in this case, an 8-hour interval at night, it does not gain weight. And this does not depend on just high fat, but all kinds of high fat, high carbohydrate diets are also protected as long as they are consumed in the evening for mice during their active portion. There's also emerging work in human clinical studies, many more papers now showing that time-restricted eating has many health benefits. So why did we get interested in this? So we are interested in aging. And when we looked at the classic aging experiments performed on rodents, the most effective intervention, of course, is restricting calories for extending lifespan. So these are classic experiments from Richard Weindruck's lab. The more you restrict calories in general, the more you can extend lifespan. But what we found is that the standard protocol was a little curious to us. So here it is. Each mouse was fed 4 feedings per week, 1 daily feeding on Monday and Wednesday mornings, and a double feeding on Friday morning. Now, of course, a mouse doesn't really want to eat 3 times a week. It normally eats every day. And it turns out that feeding in the daytime or the morning is not the normal time for a mouse. They eat most of their calories at night. And so we wanted to look into the effects of feeding time and the behavior of the mice. And in order to do this, we had to develop an automated feeding system. It delivers 300 milligram pellets, which are under computer control, and we monitor both the acquisition of the food and the delivery of the food. And this was really pioneered by a really talented postdoc in my lab, Victoria Acosta Rodriguez. And so this actogram shown here is for an ad-lib fed mouse. The dark lines, each line is a day of data. This is a 43-day experiment. You can see the mouse runs at night, and each pink circle is a pellet that has been taken. And in this case, an ad-lib mouse eats about 14 pellets a day, and it consumes 3/4 of its food at night and about 25% in the daytime. Now, when we put a mouse on caloric restriction, this is the typical protocol you feed in the morning. As the mouse became hungry, what you see is that it eats all of its food as soon as it can. And in our case, it's within a 2-hour window after access to the food. Even though we allow 24-hour access, the mouse eats its daily allotment immediately. And of course, that makes sense because the mouse doesn't know that the food's going to be around later. It better eat it. If we initiate the food at the beginning of the night, the same thing happens. The mouse consumes all the food within a 2-hour window. So What this means is when you reduce calories to a mouse, not only are you reducing calories, but the mouse is voluntarily going into a very severe time-restricted feeding pattern in which it eats in about 2 hours, and then it fasts for 22 hours every day. And we know that all three of these factors are beneficial to health. So which ones contribute to lifespan? So to study this, we set up the following experiment. So here are the three groups you just looked at: ad-lib fed mice, where food is available continuously without restraint. And then the two calorically restricted groups, 30% reduction in calories, where they consume it in 2 hours. And then to get around this binge feeding, we spread out the food over 12 hours in these two additional groups here. And then we have a final fifth calorically restricted group where the food is spread out over 24 hours. All 5 of these groups get exactly the same amount of food every 24 hours. The diet is identical. There's no other difference except the pattern of consumption of the food. So here are the some activity records from these 6 groups. Here's ad-lib. This is the running wheel activity over a 3-year period. You can see that they all remain nocturnal. And overlaid in red is the feeding pattern in the ad-lib group. It remains stable over their lifespan, as we saw before. These are the 2 calorically restricted groups. They also maintain this 2-hour feeding pattern for their lifespan. These are the 2 spread out feeding groups, night or day. And then finally, the group that is essentially fed continuously, a pellet with no day-night difference. Okay, so here's the daily food intake. At the beginning of the experiment, the caloric restriction was 30% relative to the ad-lib group. As time progressed, by the 2nd year, the ad-lib mice ate more. We did not increase the amount of food to the calorically restricted groups, so they became 40% restricted. And here on the right, you can see the average 24-hour running level, which declines with age over lifetime. This is the body weight. The ad-lib group gradually increases body weight until they're about 20 months of age, and then they spontaneously lose body weight after that. And all the calorically restricted groups maintain low body weight, and there are no significant differences among those 5 groups. So the critical measure, of course, is what is their survival. And so here is the spread group, the mice that ate continuously. And this was an extremely surprising result because the field of aging thinks that calories are the principal driver of lifespan. Yet these mice only lived 10% longer, even though they were calorically restricted by 30 to 40%. The mice that ate in the daytime, so they have a fasting interval, but they're eating at the wrong time of day for a mouse, they live 20% longer, so you get an extra 10%. But finally, the mice that ate at night, whether they fasted for 12 hours or 22 hours, they live 35% longer. So there's a huge effect of time of day of feeding. And the major contributor is actually alignment of feeding at night. So why do the mice die? As we know from many studies, cancer is the major cause of death in Black 6 mice, both lymphoma and liver cancer. And so here's the summary of the pathology in all the groups. The liver had the most lesions, as you can see here. And importantly, what caloric restriction does is not to change the spectrum of disease that leads to death, but rather just to delay the onset of all these pathologies, especially the cancers. So to try to understand better what is happening, we first looked at gene expression in the liver in the ADLIB group between 6 and 19 months of age. And this is just showing you the differential gene expression. And what we found is at 19 months, there's a huge increase in about 2,000 genes, and about 500 to 600 genes go down. Okay, so what are these genes? Well, it turns out all of those genes that go up are involved in inflammation and immune function, as you can see from this gene ontology, super significant here. The genes that go down are all in metabolic pathways. And we see this striking dichotomy of genes that are changed with aging. And so on the left here, you can see that about 14% of the expressed genes go up and about 4% go down in the ADLIB group. What about the calorically restricted groups? So here are the 5 caloric restriction groups. And what you see is that they have smaller changes in gene expression. And the groups that live the longest, the ones that ate at night, they have the smallest excursion in gene expression with age, only 2% up instead of 14%. And so caloric restriction, especially nighttime feeding, is rescuing these aberrant changes that we see in ADLIB fed mice. And so what are the contributions of, say, caloric restriction alone? So about 1,200 of these genes are restricted under any condition, are rescued under any condition by caloric restriction, shown here. These, of course, are all the same categories: immune system, inflammation, and then metabolism. If you then look at the group that has fasting, now we see an enrichment for thermogenesis pathways. And then finally, the groups that live the longest with circadian alignment, these go back to innate immune responses and inflammation, okay? So what about circadian? It turns out that, as I mentioned, there are many thousands of genes that are cycling in the liver, and we see a reduction in the number of genes with age, but primarily the reduction is in amplitude, as you can see in these graphs here. Not all genes go down. ARNT-L is BMAL, it doesn't change, but all the other ones do. And overall, when we look at fold change genome-wide, then there is a significant reduction in amplitude overall. The other thing that happens with daytime feeding is if we look at a few genes, say FOXO3, GLYS2, IGF-1, or NAMPT, these are all cycling under normal conditions, but when we reverse the feeding, when they're eating in the daytime, you can see that there's a disruption in the circadian pattern. It's either reversed or disrupted, but there's no change in level. So there are two features. One is an effect on level of gene expression, but the other is on the pattern of gene expression. And we think both of these are important. And so to summarize, we see that circadian interventions relating to caloric restriction give a very profound effect on the extent of lifespan extension, where circadian alignment at night leads to more than a threefold increase in lifespan over continuous feeding, shown in purple. And that with age, activity is declining, metabolism is declining, and inflammation is increasing. Circadian amplitude is also decreasing with age, and all of these can be rescued by caloric restriction, especially aligned at night. And so I'll stop there. This work was done by an incredibly talented postdoc, Vicky Acosta-Rodriguez, and this is a joint collaboration with the laboratory of Carla Green. And of course, we have a team of talented postdocs and collaborators who are involved in the set of experiments. Thank you very much.
Thank you very much, Dr. Takahashi, for this very interesting talk. Are there questions? So maybe I can go first. So I've noticed that you performed the experiment with male mice. And I know that the females can be protected against metabolic disease. Do you think it's because their circadian clocks is different?
Yeah, so that's a very important question. So this experiment already took 500 cages.
Yeah, I saw you use a big number of mice.
Yeah, in parallel. And so when we began this, we did not have enough capacity to do both males and females. But we are currently now doing the next set of experiments comparing both males and females. There aren't too many differences in circadian rhythms between males and females. There's, of course, the estrous cycle is different. But if you want to look at the overall average properties, they look pretty similar. So we don't know, what is going to happen with females, whether we'll get the same results with these time of day interventions or not, but we expect they should be similar.
Okay, thank you.
Joe, excuse me, Joe, that was amazing. Really enjoyed it. So I had a question. Can you do normal calorie feeding, but time restricted to figure out, the time restriction benefit in that setting?
Yeah, so in the experiments that I showed on longevity, those are normal food. Is that what you mean? Lab chow equivalent, not high fat?
No, but weren't most of the groups calorie restricted and you measured, but can you not calorie restrict and see how much of a benefit that has?
Yeah, very good question. So that's called just time restriction alone, no limit on calories. So we did do that in this experiment, but it turned out the experiment was underpowered. With 36 mice per group, we could not detect less than a 10% change in lifespan. And it turned out the time restricted groups maybe lived 8% longer. So we're actually redoing those experiments now as we speak to really see if time by itself without calorie restriction can extend lifespan. That's a really important question.
I have two questions. One, in the mouse models, is there any information about whether alcohol alters the circadian rhythm? And the other question is, I wonder in your thinking, I seem to recall people like old Professor McCants, who was one of the first to say that if you markedly reduced your caloric intake, you'd live much longer. There were others who proposed, and he and others used to sleep for 20 minutes whenever they felt tired, but never did it on a regimen. So in your thinking about it and projection, do you see some relationships between these elegant studies in mice and implications for human physiology?
Yeah, if we look at just health measures, which is really what we can do with human studies right now in short-term experiments. We can't do a lifespan experiment, of course, like this. There are striking parallels in the benefits of time restriction, and also importantly, the phase that you eat. So, there's a fad diet called intermittent fasting where people eat only an 8-hour window, but they don't pay much attention to the phasing of that window. Do they start at breakfast, or they start at lunch, or they eat at night? And we would say from our work that the phase is actually the major contributor to the benefit. We can already see that with the daytime versus nighttime feeding in mice, on high fat. But so I think we hope that this will translate to humans, and there are many studies that are being conducted now on health outcomes, okay, not lifespan. Now, an important point from our work, which I didn't emphasize, is there were no changes in body weight across those 5 groups of calorically restricted mice. And the aging field would say that body weight is a very strong predictor of lifespan. But in this experiment, there was no correlation whatsoever. So we can uncouple body weight from lifespan under these conditions. I don't know if I answered all your questions.
We have several questions in the Q&A section. So the first one is from Lou Quantley. So did you attempt to measure serum insulin in the mice? So one might expect that serum insulin to be lower if the mice eat at night while they are more active.
Yes, we did measure insulin. I didn't put it in my talk. It's you can find it in the supplement of our paper if you look. There is a funny thing that happens with mice as they age. Their glucose levels actually, when they're very old, go down. They don't go up. It's a little bit counterintuitive. But without question, the caloric restriction was beneficial to both glucose regulation and insulin sensitivity.
Thank you. And one more question from Laura Frank. So love Dr. Takahashi's talk. Translationally, do you have suggestive messaging for patients who are seeking therapeutic diets and strategies for obesity?
Yeah, of course, we're projecting from mouse experiments, but I think it's clear that caloric restriction or reduction, is important. That's definitely true for humans. And I think that there's going to be evidence that the timing of eating and also having a fasting interval is going to be important. And so already there are studies showing that time-restricted eating, beginning in the morning, they call it early time-restricted eating, beginning at breakfast, as opposed to beginning at lunch, may be better. Both of those are better than not restricting, of course. So.
Thanks. And maybe the last one, is there thought that clock-related variations may affect liver regeneration or success of transplantation as well?
Yes, so that's not, we haven't worked on it ourselves, but a lab in Japan published a very interesting paper over a decade ago. It's a paper in Science magazine by Okamura, where they looked at liver regeneration in mice that were mutant for clock genes. and they found a clear effect of clock genes on the capacity of the liver to regenerate. And so I think it's, first of all, clock genes are important in some way for that process, but that would also suggest that time of day might be important also.
Okay. Thank you again, Dr. Takahashi, for your talk. So there are other questions in this section, so you will be invited to reply to them online.
Sure.
Thank you.
Bye-bye. Thanks.
So now we will have 3 microtalks, and the first one is from Scott Freeberg. Scott is a graduate student in the Biological and Biomedical Sciences program at Harvard Medical School in the Gosling Lab. He's interested in the molecular mechanism of liver formation and the functions of vitamin D signaling in endoderm development. Please go ahead. Oh, we can hear you. Okay, I think you touched a button and it worked for a few seconds. No.
How about now?
Oh, now it's good. Yes, perfect.
Okay, apologies for that. Right. Yeah, my name is Scott Freeberg. I'm excited to speak today and appreciate the invitation to do so. I'm going to share my work on how vitamin D receptor regulates energy metabolism. This is a project that we're interested in because vitamin D deficiency is associated with fatty liver disease and with obesity. And we're actually using genetic modeling in zebrafish to try to understand the evolutionary functions of vitamin D receptor in vertebrates. So a key tool that we're using for these experiments is a transgenic zebrafish line in which we're overexpressing a dominant negative vitamin D receptor under control of a hepatocyte-specific promoter. And these fish have a dramatic increase in hepatic steatosis compared to their wild-type counterparts. And they also exhibit a decrease in liver growth, quantified here. And these phenotypes are actually reminiscent of a starvation state in larval zebrafish. And in fact, we can see that nutrient repletion in the form of egg yolk treatment prevents the induction of hepatic steatosis by the loss of VDR function. So what we think is happening in these animals is that the loss of hepatic VDR signaling causes a perceived starvation state, which is marked by an increase in energy storage in the form of fat and a decrease in energetically costly developmental liver growth. We're also interested in how hepatocyte vitamin D receptor regulates organism-wide metabolism. We've investigated this using adult zebrafish studies. And you can see that wild-type fish fed a high-fat diet exhibit dramatic organism-wide tissue growth compared to control diet counterparts. And we can quantify this simply using weight of the fish as a readout. And you can see that the loss of hepatocyte VDR signaling attenuates that diet-induced growth induced by high-fat diet feeding. And in addition, the loss of hepatocyte-specific VDR also causes expansion of visceral adipocytes, shown here, compared to wild-type fish. So together, these data indicate that hepatocyte vitamin D receptor signaling is essential for adult zebrafish to fully adapt to fluctuations in environmental nutrient supply. And to put this in an ecological and evolutionary context, we know that in the environment, vitamin D is present in phytoplankton and in zooplankton, and that these are the core constituents of aquatic food chains. So therefore, vitamin D is a ligand that is sourced from the environment that has the capacity to antagonistically regulate energy storage and organismal growth in fish through its activation of vitamin D receptor. So on this basis, we're proposing that vitamin D receptor has an evolutionary role as a sensor and effector of environmental nutrient supply, with the idea that high levels of environmental nutrient supply are sensed in the form of high vitamin D, which reduces the storage of energy in favor of hepatic and organismal growth. Thank you.
Thank you, Scott. So we didn't plan time for live questions for the microtalk, but we encourage questions to the Q&A. Thanks again. And we will move to our next microtalk speaker, Dr. Shuang Wang, who is currently an instructor in Dr. Scott Friedman lab at Mount Sinai, New York, working on liver fibrosis. And his talk is about an endocrine signaling circuit in hepatic stellate cells that underlies advanced fibrosis.
Okay, so thank you. So my name is Shuang Wang, and I am from Mount Sinai, New York, in the Friedman lab. So as you know, there's currently no approved antifibrotic. So the goal of this study was to try to uncover some novel drug targets that could serve as antifibrotics. What we did then is to carry out single nuclear RNA-seq from a NASH model that we recently published in the lab called the fat NASH mice. And from here, we identified two different populations of stellate cells, which we call HSC1, HSC2. And as you know, stellate cells are the main drivers of liver fibrosis. What we see is that HSC2 is also coming from the NASH condition, and it expresses markers that are in line with stellate cell activation, and the pathways that are involved in activation are very active in these cells. What was really interesting was when we predicted cell-cell communication using cell-from-BB based on the expression of ligand receptors on different cell types. What we see is that the non-parenchymal cells, which are the endothelial cells, stellate cells, and cholangiocytes, were extremely active in carrying out cell-cell communication in the NASH liver, and especially HSC2, which are the activated stellate cells, seems to undergo this autocrine interaction. So it's expressing both ligands and receptors that could promote its activity in NASH. And we carried out similar studies in NASH patients using the same strategy, and we identified the same autocrine loop in stellate cells. This was really interesting because it lined up with some previous mathematical modeling that suggests autocrine activity in myofibroblasts in general, in different tissues and different fibrosis seems to drive disease. We took a further look at these autocrine interactions from HSC2, and what was really interesting was that most of these interactions were short-range interactions. So this is defined as ligands and receptors that need to be brought into physical proximity in order for the interaction to occur. How could this be achieved? So this led to the second major finding from this study, where I used a tissue clearance to try to image the morphology of the stellate cells. And as you can see here, I imaged the staining for stellate cells is desmo, which is in the purple here. We can capture the protrusion and projections that are coming out of the stellate cells in 3D space using tissue clearance. And what we uncovered was that the stellate cells seems to undergo a very dramatic morphological change in the NASH condition in our mouse model. So this is shown here. So in the control liver, we can see stellate cells with its various projections. But in the NASH condition, the stellate cell projections are much more elongated and that much more numerous, such that these different stellate cells are actually beginning to fuse together into a single physically connected network through their projections. And we quantify this through different times on the fat NASH model. We see this tendency to form physical contact increases with increasing disease severity. And in the rest of the study, we focusing on one pair of autocrine interactions, NTF3 with NTRK3, to show that NTRK3 expression is found on the projections coming from stellate cells, suggesting that the stellate cells, through their physical connected network, actually provides a physical basis to facilitate this autocrine signaling. And when we antagonize NTRK3, both in cell culture and also in vivo, we're able to reduce stellate cell activation and also reverse very advanced fibrosis in our NASH model. So with that, I'd like to thank my mentor, Dr. Scott Freitman, who has supported not only this project, but also my transition to independence. And this essentially is the graphical summary of what we've uncovered in this study, and the paper has now just been accepted for publication. So thank you.
Thank you, Shang. So again, we won't have time for live questions, but you can reach out the Q&A. So our next speaker is Dr. Sujin Park. So she's a postdoctoral fellow at Biozentrum at the University of Basel in Switzerland. And her work is focused on the transcription factors TEA, D2, and E2A that repress acetyl-CoA synthesis to promote tumorigenesis.
Thank you for this opportunity today. I'm Sujin Park, and I'm today talk about reprogrammed acetyl-CoA metabolism in liver cancer. Activated oncogenic signaling and reprogrammed metabolism help cancer cell escape from immune attack and keep the proliferation and prepare the EMT for the better situation. Among the metabolic pathways, I'm particularly interested in acetyl-CoA metabolism. Acetyl-CoA is a fundamental metabolite and also key energy source. It needs for TCA cycle, de novo fatty synthesis, and also ketone body generation. But more importantly, acetyl-CoA is important for modification of proteins and metabolites. Recently, it is reported that branched-chain amino acid catabolism fed oxidation, acetaldehyde to acetate conversion reduced in liver cancer. However, all the papers focus on individual pathway. We don't know acetyl-CoA levels and underlying mechanisms. So we thought to investigate global regulation of acetyl-CoA metabolism in liver cancer. So to begin with, we checked the acetyl-CoA levels in tumors of the human patient and also our mouse model. Compared to non-tumor parts, the acetyl-CoA levels dramatically reduced in tumor, and acetyl-CoA level is important for the protein acylation status. So we check protein acylation by Western blot and also tissue microarray. And as you can see here, compared to the non-tumor part from the same patient, protein acylation is really reduced. To get the overview of acetyl-CoA metabolism, we took advantage of multi-omics analysis using our mouse model, also core needle biopsy from human patient. It covered all different etiology. And we found that all 6 biosynthetic pathway of acetyl-CoA is reduced, and this one is transcriptional suppression. So we narrowed down our target and found 2 transcription factors. One is TD2 and the other one is E2A. Both of them are upregulated in liver cancer, and it suppresses expression of acetyl-CoA synthetic enzymes, more than 40% of whole acetyl-CoA metabolic enzymes, and in turn, acetyl-CoA level reduced in tumor and protein acylation reduced, including many metabolic enzymes and pluripotency markers. And pluripotency gene expression and stemness marker expression increase in liver cancer upon expression of E2A and TD2, and it promotes dedifferentiation and proliferation. And we validated in mouse model as well. So when we knocked down TD2 and E2A, it failed to form subcutaneous tumor, and it nicely correlates with the patient survivor. Thank you for your attention.
Thank you, Sujin. And so I would like to thank again all the speakers from this session. So we will now have a short break and we will resume with session 4 at 3 p.m. Thank you.
Hi, everyone. Welcome back to session 4. So my name is Jesse Kirkpatrick. I'm a postdoc in Sangeeta Bhatia's lab. And I'm delighted now to introduce the first speaker of session 4, Dr. Giovanni Traverso, who has done some really fantastic and innovative work at the intersection of gastroenterology and engineering. Dr. Traverso is an assistant professor of mechanical engineering at MIT, as well as a gastroenterologist at Brigham and Women's Hospital. He completed his medical training at the University of Cambridge, his PhD at Johns Hopkins with Bert Vogelstein, and his postdoc at MIT with Bob Langer. And his lab now focuses on developing biomedical devices to enable new modes of drug administration and to sense a broad array of physiologic and pathophysiologic signals. Dr. Traverso's talk today is entitled Explant Liver Model for Drug Development. Thank you for joining us, Dr. Traverso.
Thank you so much, Jesse, and thank you, Sangeeta, Wolfram, and Heather for that kind invitation. So what I'd like to do in the next 20 minutes or so is share with you some of our recent work with respect to modeling the GI tract, different parts of the GI tract, including the liver, and really with a focus on drug development. A lot of the work that we do focuses on how to help individuals receive medication, anything from a biologic orally to having a capsule reside in the body for a week or even a month or even longer, to help essentially make it easier and ensure that patients can receive therapy. And so just before I dive in, these are my disclosures. We work with several companies as well as help start them to help translate the technologies from the lab to help patients. And the work that I'm going to share with you is work that started about 7 years ago. There have been several critical members over the years, and these are some of them. Thomas Van Erlach, who's now the CSO at a local biotech, Yunhua Shi, who has transitioned to consulting, Melody Lin, who's in Taiwan, and Daniel Riker, who's now an assistant professor at Duke. But let me just briefly go over some work outside of the liver, which is how this body of work really got started. So a few years ago, we were doing some work with the Gates Foundation, and there are several drugs that they're interested in delivering in sub-Saharan Africa and Southeast Asia that are very challenging to formulate. Some might call them bricks because they're difficult to solubilize and absorb. And so we wondered if we might be able to help by developing better models of the GI tract to really help optimize the formulations. And what we recognized was that the industry standard, even to this day, is a colon cancer cell line that happens to polarize and look like the intestine after being in culture for about 14 to 21 days. That cell line is called KCO2. And even so, and with approximately a predictive capacity of about 50% with respect to human absorption, it, as I said, remains the industry standard. And so what we wondered was, could we try and develop a system that perhaps better approximates this environment? And we do a lot of work in our group with large animal models, very specifically pigs. And the reason we do that is because of the dimensions that in general are closest to adult humans, and which helps de-risk a lot of the engineering that we're doing in our group. But we wondered if we might be able to actually take tissue from the pig and interface with it in a high throughput way. And so that's exactly what we did. And essentially, we developed techniques to take tissue from local slaughterhouses from New England, and then essentially interface with it in a high throughput way. So we developed plates that can directly interface through high throughput drug screening facilities to now ask the question, are there formulations, and by that I mean additives or excipients, enhancers, that can enhance the absorption of a drug? And so we were able to accomplish this. I'll just walk through fairly quickly just to give you a flavor of what this involved. We screened several media compositions to try and identify media that could maintain whole tissue segments viable outside of the body for several days and even weeks. And so what you see in the green and orange essentially is a live dead assays, and we identified multiple media conditions. We also characterized the villi and demonstrated that the villi actually remained stable, so the architecture remained stable for several weeks. We also found a correlation with the age of the animal and its ability to remain viable. We characterized the expression pattern of several markers, including drug transporters, which is an area that we're very focused on given a lot of the drug development that we're doing. And noticed some changes, certainly, and perhaps not surprisingly, given that we were taking tissue out of an animal and placing it on a petri dish. But using the system, we then developed a mechanical interface that allows high throughput interrogation of this mucosa, thereby allowing us to ask the question, can we identify additives, for example, that enhance transport across this tissue? And these are the initial designs that you see, but then the actual laser drilled acrylic. We did a lot of modeling and optimization with respect to the amount of force that was sufficient to create distinct wells, while still retaining viability and not inducing pressure necrosis in the tissue. And then we demonstrated initially the capacity to predict a human absorption by taking a panel of over 60 approved drugs that had been evaluated in CAECO2, for which there is human absorption data, and then asking the question, how well does our system predict the absorption in humans? And what we found was, as you can see the bottom of this slide, correlation coefficient of 0.9. So this was very reassuring with respect to the performance. But then we actually took a peptide that generally is not absorbed through the GI tract. We took oxytocin, and we took and created a library of excipients. These are ingredients that generally fill capsules, and agnostically really mix and match these and ask the question, are there mixtures that enhance transport across the mucosa? And indeed, what you see here in this heat map is essentially a representation of that enhanced transport. So the bright green meaning that there was more drug that was going across the tissue. And then we gave those formulations to pigs and demonstrated that we could enhance the absorption orally of oxytocin by about an order of magnitude. We characterized the mechanism to demonstrate that this was through a transcellular route, so we weren't completely disrupting the mucosa, but rather had identified mechanisms or enhancers, I should say, that are transcellularly delivering the drug. So this we had done, and as we were doing this work, we of course wondered, could this be applied to the liver, recognizing drug-induced liver injury, represents a significant fraction of acute liver failure cases. And of course, a significant fraction are attributed to acetaminophen, but also we recognize the incredible challenges that the drug development process faces with significant drug attrition as a consequence of liver injury. With respect to acetaminophen, we recognize that it is responsible or associated with very high numbers of liver transplantation events and also deaths to this day. The toxicokinetics have been characterized to a fairly large degree, although there's certainly several areas that are unknown, but we recognize how acetaminophen is being metabolized, and also in the overdose settings, the toxic elements essentially that can be identified and where the injury, I should say, is attributed to. And so with respect to the liver, we wanted to ensure that as we started to look at the enzymes that are responsible for the metabolism, that actually we were at least, that the homology was high enough or close enough to human for this to be meaningful. And overall, we found the homology across this panel of cytochrome P450 enzymes to be higher than 80%, as you can see. And with the mouse homologs also fairly near to this. And so with this, we started to consider different ways of taking the whole tissue. And this is liver from pigs initially. We had identified some examples in the literature, some limited essentially efforts in trying to do similar things. And so we started to investigate different sectioning techniques, different processes on how to generate a consistent essentially slice of this explant so that we could then immobilize it in a high throughput system similar to the one I just described to you that we had developed initially for the small intestine. And so we honed in on using a dermatome. A dermatome is the surgical tool that's used for skin grafting and used for harvesting, and is a tool that can generate very thin slices of tissue. And so here, we eventually settled on a liver slice of approximately 250 microns. And we use and develop plates that could help immobilize this segment. And then by using essentially a live dead assay with some slight modifications, we're able to then interrogate whether we were having or inducing toxicity. What we found, at least, unlike the intestine that we were able to keep viable for many weeks, actually, we found that the optimal period for interrogation and working with the liver was approximately 24 hours. As part of our initial evaluation, we took several drugs, and I'm showing 3 here, to understand whether we could start to see that toxicity sort of readout. And as you can see here, with increasing doses of known drugs known to be toxic to the liver, we could start to see a dose response with respect to toxicity. So this was, all reassuring. What we also wanted to see is whether we could take known inhibitors of the cytochrome P450s for these specific drugs and see if we might be able to rescue that readout. And so actually, as you can see here, for the 3 drugs, we were able to reverse that toxicity, and we could actually identify those readouts. And this is all ex vivo in these explanted high throughput systems. So as far as acetaminophen is concerned, we looked at inhibition of the P450 enzymes that are known to metabolize the drug. And what we found, for example, was for CYP2E1, that actual inhibition could rescue fully that toxicity. But for some of the other CYPs that are involved in its metabolism, specifically 1A2 and 3A4, there was a partial rescue. And so we also looked at combinations of these inhibitors and found that we could fully rescue when combining those inhibitors. Now, the question that we asked ourselves is, could we identify new inhibitors and eventually potentially inhibitors that can be derived from inactive ingredients and the FDA is generally regarded as safe materials that could support that inhibition potentially. And so we applied some machine learning based techniques where we took known data from known inhibitors of these CYPs as well as non-inhibitors, and based on the different structures, we're able to identify 2 molecules. So as far as the 2E1 inhibitor, we identified rutin, and then we looked for a combined 1A2, 3A4 inhibitor, given the prior data with respect to the synergy that was required to help rescue that toxicity, and that was eugenol. And so rutin is a flavonoid that's derived, or found, I should say, in citrus and other foods. And eugenol is an aromatic oil often found in cloves and cinnamon, as well as basil and bay leaf. And so we took these, and initially in our explanted systems, we demonstrated that we could indeed still rescue that toxicity. And we started to investigate the timing of exposure, at least ex vivo, to see, to try and sort of map the kinetics or the needs or the opportunity for that inhibition and associated rescue. And but we saw that we could rescue, and as you can hopefully appreciate, at least ex vivo initially, it appeared that we had to predose the rutin in order to have a meaningful rescue. With respect to eugenol, we saw also, I would say, partial rescue, but again, perhaps what we started to see is that there was also a potential time dependency. Now, what we did next is we actually administered these to mice to investigate this reversal. And so we used a dose of 350 mg per kg administered intraperitoneally of acetaminophen, and then administered the rutin and eugenol. And I'm just showing you here the ALT data to give you a sense on the level of protection. But we found in vivo, actually, co-administration of these was sufficient to rescue the toxicity. And so just with respect to rutin, the doses that seem to actually support or mitigate that toxicity were the 20 to 40 mg per kg of rutin, and then for eugenol, approximately the 40 and 80 mg per kg that were co-administered. Additionally, we wanted to understand that whether we were interfering with the acetaminophen levels or the toxic metabolites more specifically. And our hypothesis was that we would hopefully be interfering more specifically with the toxic metabolites. And so what I'm showing you here is actually essentially a time course where we administer a dose of acetaminophen along with these newly identified inhibitors, and then collect samples over the course of 4 hours, and then calculate the area under the curve essentially for different metabolites. And in orange here at the bottom, we have the toxic metabolites. And what I hope you can appreciate is that we were able to essentially suppress the toxic metabolites while retaining the levels of acetaminophen. And so the actual therapeutic dose is still retained, but we have selective depression or lowering of the toxic metabolites. And then finally, with respect to the dosage form, we actually created a tablet and sort of started to map out its release kinetics here. And this is some initial work that's been done in vitro in order to scale up from the rodent dose towards human translation. And so with that, I just want to thank our incredible team. This is our team this past summer over in Cape Cod. I also want to thank Wolfram, who kindly collaborated with us, and Bao-Liang are also who collaborated in this project and has been a wonderful collaborator and mentor over the years. And I'm happy to take any questions, but if I don't get to them, here are my emails. And also would like to thank incredible support of our sponsors. Thank you so much.
Thank you, Dr. Traverso. That was fantastic. So yeah, please do put questions in the Q&A. And the first question is from Dong Fu. Dr. Traverso, I wonder if the ex vivo liver slides can respond to idiosyncratic DILI. Do you notice any differences in DILI response using liver slides obtained from different liver lobes?
That's a great question. We haven't investigated that.
Maybe another question from me. So I think that this is an incredibly powerful system, this ex vivo culture system. And I know that you're currently working with relatively short time windows of 24 hours or so, but I wonder if you've thought about other like treatment paradigms that you can think about for other liver diseases.
Absolutely. I mean, I think this is we took this acetaminophen case really as a use case. I think that's well known to many, and also because we have positive controls to characterize. And so, we think that this is something that we can apply across many use cases. So yes, I would say it's a short answer.
Great. Yeah, are there any other questions? And if not, you can feel free to put more questions in the Q&A, and Dr. Traverso hopefully will be able to get to them during the next talk.
Absolutely. Well, thank you so much. And like I said, anybody feel free to email me if there are any more questions. Thank you.
Great. Yeah, thank you so much. So I am excited to move on to our next speaker, who is Dr. Sonja Schreckfer. Dr. Schreckfer received her MD and PhD from the universities of Würzburg and Hamburg, and is now scientific founder and head of the hypoimmune platform at Sana Biotechnology, as well as a professor at UCSF. Dr. Schreckfer has made important contributions in the field of stem cell immunobiology, which promises to improve the efficacy of regenerative technologies through immunomodulation. And her talk today is entitled Protecting Transplanted Cells from Immune Rejection to Unlock the Potential of Regenerative Medicine. Thank you so much for being here, Dr. Schreckfer.
Thank you very much, Jesse. And I'm very excited to be here today. Thank you to Sangeeta and Wolfram for inviting me. And we go into my presentation mode. Yeah, so I would like to show you today our work that is not published, because I figured all the papers, of course, you can read by yourself. So this is my disclosure slide for today. And to start with the topic, whenever we do cell transplantation, but that's also true for tissue transplantation or solid organ transplantation, from one person to another, we face the issue with immune rejection, because our immune systems can distinguish between foreign and self. And whenever we transplant something foreign, tissue cells and organs do get rejected. There are different possibilities how to overcome this allo rejection. One is, of course, immunosuppression. We're using that for solid organ transplantation. However, we see a lot of toxicity, mortality, and this is really not the path we should use moving forward with regenerative medicine. Another strategy would be that we use HLA banking. And the reason for that is the HLA molecules are on the surface of each cell. And I'm very often comparing them to the fingerprint of a cell. So an HLA is very specific for a person. And that's how the recipient's immune system can recognize the foreign graft. So the idea here, or the concept, is that we would create HLA banks and try to transplant HLA matched cells or tissues. The problem with those HLAs is that they're not only sitting on the surface, they also present allogenic peptides. And these would always get recognized, even if we would be able to have a huge bank and do HLA matched transplantations. So most likely this pathway moving forward would also require immunosuppression, because we would get rejection through the allopeptide presentation. A third path, and that's what I want to talk today about, is the genetic hypoengineering. And the idea is that we have the cells in our hands before we transplant those cells into the patients. We could use engineering approaches to modify the cells that they don't get rejected after transplantation. And the concept I want to introduce today is that we have a healthy iPSC donor. We use this gene editing, and then this iPSC, what I call hypoimmune cell product, could be differentiated in basically any cell type. I show you today a couple of cell types we did. Unfortunately, we didn't do iPSC-derived hepatocytes yet. So that's something I would be personally very interested in, but we haven't done that work. And then we could transplant those cells off the shelf into any patient. And the goal is really anytime, anywhere. It shouldn't depend on the zip code you are living. You should be able to get those off-the-shelf therapies. And the advantages from those kind of strategies are that we would have one well-characterized master cell bank. And we wouldn't have to have that time-consuming and cost-consuming issue with like autologous approaches, where we generate the iPSCs in the cell product for each patient, patient-specific. The issue we faced, and we started working on that already in 2005. The first issue we faced was to overcome adaptive immune responses. And the adaptive immune responses, those are T cells and B cells. And they do see the foreign cell, and they do that by recognizing the foreign HLA molecules, so that fingerprint on the cell. So it became quite obvious quite early that we have to remove those HLA molecules on the surface of those cells to overcome this adaptive immune rejection. But what happened is, by removing those HLAs here on the right side, now we created a cell that is called missing self, because it doesn't have that own fingerprint anymore. And we have immune cells like NK cells and macrophages. They belong to the innate immunity. And those cells are actually in our body to recognize missing self cells and to kill them immediately. And the biological reason for that is, if we have, for example, virus infections, HLA molecules get downregulated, and the innate immune cells, they sense that missing self and they eliminate virus-loaded cells. So it is a defense mechanism, but in our case, it became a struggle, because we realized we have to do an HLA knockout to overcome the adaptive immunity, which is the main barrier in allogenic transplantation. But at the same time now, we created an issue that the innate immune cells are seeing our cells as missing self and would start attack and kill those cells quite fast. So the early work we did is we created an iPSC line. We knocked out the HLA molecules. We have HLA class 1 and class 2. We knocked them out with the CRISPR-Cas9 technology, and then we re-engineered in certain molecules in the hope we could control T cell killing and the innate cell killing. I show you here the example for the NK cells. And what happened is here you see on the top, so the first example is an HLA class 1, class 2 knockout iPSC. The T cells are not killing those cells because they don't see any HLA. But whenever we try to re-engineer so-called protective HLAs, like HLA-C is known in the literature to be protective for HLA-E, HLA-G. So whenever we re-engineered back an HLA molecule, we got issues and the T cells were getting activated and killed the iPSCs. When we re-engineered back other molecules, you might have heard of like PD-L1 or CD47, and the HLAs were knocked out. We didn't see any T cell activation. What I want to show you now on the bottom is the innate immune cells, because they are only killing when we don't have HLA molecules. So in green, you see our target iPSCs. And then we added NK cells, and what you see in the videos, our PD-L1 video doesn't want to work. Yeah, no. When you look at the videos over time, these are 4 hours for our videos, is that a cell that is HLA class 1, class 2 knockout, like in the first video, does get killed quite fast by NK cells. Now, re-engineering back certain molecules didn't inhibit the killing by the whole NK cell population. To be a little bit more granular here, those molecules are effective when we sorted NK cells with certain receptors, but with the overall NK cell population, how we find it later in the patient, only CD47 was able to protect from NK cell killing and also macrophage killing, what I don't show today. So the data I want to share with you today are all following now the same approach. We have an iPSC donor, a healthy donor. We disrupt the HLA or MHC class 1 and class 2 molecules, and then we overexpress the CD47 to avoid the killing by the innate immune cells. And then the goal is that we differentiate those into different cell types and transplant into allogenic recipients without any immunosuppression. You see here the published work. This published work only used mouse and humanized mouse models. These are not the ones I want to share today because we were looking for a more translational model and we choose the non-human primate model. So all the data I show you today are in non-human primates. And the reason for that is NHPs have a really highly activated immune system. They are a high bar study. So we transplanted NHP iPSCs and iPSC-derived cells without immunosuppression into NHP recipients. The first cohort we did was really using the platform. The platform is an iPSC line. And we transplanted into 4 NHPs, the unmodified wild-type iPSCs, and into another cohort of 4 NHPs, we transplanted the hypoimmune, or we call them HIP for hypoimmune platform, the HIP iPSCs. We had labeled the iPSCs that we could do in vivo imaging of our monkeys and that we could follow the cell transplant over time. So we overexpressed luciferase and we use bioluminescence imaging to track the stem cells. These are the first 4 monkeys where we transplanted the wild-type unmodified iPSCs. We transplant into the left leg muscle. I personally like the muscle location because it provides you fast revascularization for the graft. So you don't have to struggle like with subcutaneous applications where maybe the graft doesn't get vascularization and is dying for non-allogenic reasons. So we transplant it into the muscle location. You see that the grafts are there at day 0, but within 3 weeks, you see that all the signals decline. The signals are ATP dependent. So whenever the cell is dying, you don't see the signal anymore. And on the right side, you see the summary we measured every week, and you see that the blue grafts disappear into the background. The grafts were rejected. After 6 weeks, we transplanted into the very same NHPs hypoimmune iPSCs. So those didn't have the MHC molecules, and we overexpressed CD47. On day 0, you see the graft is there, and then you see that the graft survives without immunosuppression. What is interesting here is that those NHPs were sensitized. They had rejected the unmodified iPSCs before, so they know the antigen. And that is the concept of immune evasion. It's not inducing tolerance, it's not education of the immune system. the cells we put into those NHPs, they have the features of immune evasion, meaning that the immune system doesn't recognize that we transplanted them because the immune system does know the antigens. What I don't like so much about those graphs, and we can discuss it later more, that they go a little bit up and down, up and down. That's not something we usually see in mice. The reason for that is our iPSCs, unfortunately, tended to form bone. And when we do the bioluminescence imaging, the signal can't go through bone structures. So I think by looking later at the histopathology, that most likely, depending how that leg of that monkey was, that we might have hit bone structures and didn't get this full signal. That's why you see the curves not completely nicely like they should be. The other set of 4 animals had received hypoimmune iPSCs into the left leg, and you see that the signal is there, and the signal stays up, what you see on the right side, the red graphs. Here again, we transplanted after 6 weeks the other cell types, so in that case, the unmodified wild type iPSCs, and you see the blue hits the background very fast, so they do get rejected. What we learned here is that although we have after 6 weeks an immune activation and the graft gets rejected, the concept of immune evasion seems to work because the hypoimmune cells, they don't get killed or attacked at all. And what we also learned here is that we are not modifying the immune system, we are not immunosuppressing the immune system, because you see that the NHPs are capable of rejecting a wild type graft. We did a lot of immune analysis, and we will publish all in-depth analysis soon, but I pulled here the most important data, I feel, and that is the T cell activation, because T cells are it's the main cell type in allogenic rejection that attacks the graft in the early phase. So we looked for T cell activation using ELISpot assays. You see on top the 4 monkeys that had received the wild type unmodified iPSCs. Before the transplantation, pre-TX, there is not a lot of T cell activation, but 1 week after transplanting the graft, you see a high blue graph, and then it declines over time. This is expected from solid organ transplantation, that the acute cellular rejection declines over time when the graft is gone. What you see in red is those are the hypoimmune iPSCs we transplanted after 6 weeks, and you see that there is no red T cell activation graph coming up. The antibodies, here I show you the IgM antibody production, gave us a peak for the IgMs at 1 week. And then you see that switch to the IgG antibodies here around 2 weeks. So this is expected. That's what we would think would happen in a human. And that shows you how similar the NHP immune system reacts to the transplant. And what you see is that IgM antibodies here, there is no de novo increase, no de novo antibody production after we transplant the hypoimmune iPSCs. And what you see here in the IgG compartment is when we transplanted after 6 weeks, the monkey had a lot of IgG antibodies. So that monkey is pre-sensitized, those monkeys. But again, no de novo antibody production. And when we measured donor-specific antibody binding, what we found is that those IgG antibodies are not binding to the hypoimmune iPSCs, and therefore there is no CDC or ADCC killing. On the bottom, you see the hypoimmune NHPs, the 4. So you see in the beginning, there is no T cell activation. We don't see here antibody production. After 6 weeks, when we transplant the wild type unmodified iPSCs, you see the blue graphs come up. You see IgM antibodies switch to IgG. So we learned from that that we can most likely redose hypoimmune cells because of that immune evasion concept, that we could think about recipients who are sensitized, and that the hypoimmune transplant doesn't educate the immune system, but also doesn't immunosuppress the immune system. With those data about the platform, those are only the pluripotent stem cells, we thought we really want to reproduce that with differentiated cell types. So what we did first is, together with Chuck Murray at UW, where those transplants were performed, we used iPSC-derived cardiomyocytes, either wild type or hypoimmune, and they were transplanted into the heart muscle. The monkeys themselves, they were healthy, so there was no myocardial infarction. We had 4 NHPs that had received hypoimmune iPSC-derived cardiomyocytes, and 2 of the NHPs had received the unmodified iPSCs. And I love to start with this one picture because this is one of those NHPs who received an hypoimmune iPSC-derived cardiac graft. And you see in pink the transplanted cardiomyocytes without any immunosuppression. This is 8 weeks after the transplantation. And you see how beautiful the pink transplanted cardiomyocytes are within the host myocardium in green. So there is no local reaction. They are beautifully aligned. And I love that picture just to look at it and really to see how powerful it can be when we use it in the right setting. So I want to dig a little bit deeper into those cardiomyocyte monkeys. On the left side, you see survival of the transplanted graft in immunofluorescence, so the green, the pink cells in the green host myocardium. In immunohistochemistry, we use the SSTN1, and you see in brown the graft, just to give you a different area of the same heart. When you compare that to the right side, this is an example of an NHP that had received the wild type cardiomyocytes. And what you see, this is also 8 weeks after transplantation. You do see where we transplanted the cells. So in blue, the nuclei are still there, but you see that there is no pink color, there is no brown color, the graft itself is rejected. And you see here in the H&E staining a lot of mononuclear cell infiltrates still sitting around. So you see the difference of those pictures. And when we look deeper into the local immune response, what we find in the wild type are a lot of T cells, which you see here stained in brown, macrophages, and then lots of plasma cells. So a lot of plasma cells and antibody reaction in those monkeys. When we now isolated those local immune cells out of the heart, we did single cell suspension, did a sort for CD45 positive immune cells, and we wanted to understand if those local immune cells can recognize the graft. So we put them on wild type cardiomyocytes in a killing assay, and you see how the curve goes down and the wild type cardiomyocytes get killed. That shows you that those local immune cells, they build the memory, they see the antigen again in our assay, and they are killing. We confirmed that with a live dead assay by flow here in blue. On the left side, you see hypocardiomyocytes. You do see a few of those brown stained plasma cells, macrophages, T cells, because each organ does have some immune cells, of course, there. And what we did is we did the same procedure, homogenized the area, isolated 45 positive immune cells and put those on the hypoimmune cardiomyocytes. And what you see is in the tissue, there are some immune cells, like in each tissue and in each organ, but there's absolutely no recognition here of our transplant, what we had transplanted into those NHPs. And that shows you that there is no, according to an allogenic graft rejection, there is no recognition. Those immune cells don't see the graft. What we did is we also confirmed in the blood, like I showed you before for the iPSCs, and we see similar results for the cardiomyocytes. So the wild type NHPs on the top, they gave us T cell activation. You see those high blue graphs, they decline over time. We do see IgM antibodies, IgGs, so nothing special. On the bottom, you see the hypoimmune cardiomyocytes, and you see that there was no T cell activation, no antibody production. When we then looked at the innate immune cells, and I show you here the example for the NK cells, you see that hypoimmune here on the bottom doesn't get killed by innate immune cells. What you also see is that the wild type cardiomyocytes don't get attacked. The reason for that is wild type cardiomyocytes do express the HLAs, and those inhibit innate immune cells. So the killing by the immune cells is really only triggered because we remove the HLAs and have a missing cell. And that's what you see here on the white bottom, because here we are blocking the CD47 molecule. So now we don't have the protection, and you see that our missing self triggers the killing by the NK cells. That shows you the mechanism itself, but it also, of course, offers now a safety strategy, because we could think about if we transplant hypoimmune cells into a recipient and we want to control them and remove them for whatever reason, that we could block that CD47 by anti-CD47 antibodies, and then the cells which have the genotype of an HLA knockout would get killed or eliminated by the innate immune cells. In this example, we had NK cells. We then moved on, and we were interested in studying RPEs, retina epithelial cells, and for two reasons. One, because these cells are in the eye, and the eye is supposed to be immunoprivileged, so we wanted to understand that. And the second is the innate immune cells in the eye are not macrophages, they are the microglia. So we wanted to understand if CD47 also would work with microglia, how you would find it not only in the eye, but also in the CNS system. So Sally Temple from New York, the Neural Stem Cell Institute, they differentiated for us the NHP iPSCs into the RPEs. And we had 3 monkeys receiving the wild type and 3 monkeys receiving the hypoimmune RPEs. First, I want to show you that the eye itself is not immunoprivileged. And it's not immunoprivileged, I think, at least for one reason, because when you do a cell transplantation, you always break the barrier. The blood-brain barrier, you always go in with your device. So there is no protection. There is no graft protection after the transplantation. So what we have seen is when those RPEs, wild-type RPEs, were grafted into the eye, you see them here after 1 week in white. We find some of them after a week in green, but we see a lot of microglia and T-cell activation. So the eye seems to be not immunoprivileged by preventing this allograft rejection. When we look at the monkeys with the hypoimmune, you see in white here the transplanted hypoimmune RPEs. And you see in the immunofluorescence, we didn't identify microglia or T cells. So the red here in the immunofluorescent, that's the host RPE layer. And you see the green transplanted hypoimmune RPEs in the eye. And the study is not completely finalized, but hopefully next time when we see each other, I can share then the final data of those RPEs. What was interesting for us was to understand that the microglia really act similar like macrophages. So first, the microglia here on the bottom can recognize an MHC class 1, class 2 knockout graft, so is sensing the missing self like macrophages do. And CD47 overexpression can protect from microglia killing, like we have seen it for the macrophages before. So that was a learning effect we didn't know before doing these experiments. The blood looked similar, so I don't want to bore you with that. We saw T-cell activation, antibody production, didn't see that in hypoimmune. And then I apologize. Yeah, I want to show this first. Another experiment we did was not using iPSC-derived derivatives, but we wanted to investigate if a primary cell type can be modified to become hypoimmune and then transplanted. So we used the concept of primary islets. We used an NHP pancreas in collaboration with OHSU. And then the islets got isolated, and we created our hypoimmune edits and transplanted those islets into the muscle location of allogenic NHPs. Again, no immunosuppression, and the monkey is still alive, but we are now at 7 months after transplantation, and you see here the transplanted islet cells surviving without any immunosuppression. So the technology is not only for iPSC-derived technologies or ESC-derived technologies. We could also think about primary islets and other cell types. So I want to finish with the concept, what I just showed you, because to overcome the allogenic barrier, there are really different strategies. And I showed you today the number 4. This review by the Rice University summarized, in my opinion, very nicely the different options we have to overcome an allogenic barrier. And what I showed you today is creating a universal cell by immune evasions. So it's not like number 3, tolerance induction or education of the immune system. And with that, I think for regenerative medicine, when we can break that allogenic barrier, I think there are really big changes ahead. And I hope that the future for regenerative medicine looks like that for whatever tissue we are working on. So I hope that we can overcome allogenic barriers for so many more cell approaches in regenerative medicine. And with that, I want to thank you.
Thank you so much, Dr. Scheffer. That was really, really fascinating talk. So we have a question and a Q&A. Really love the presentation. Oh, this is from Konstantin Zuanas. Really love presentation and set of results. Would you have thoughts on other cell types that would work particularly well or poorly through this approach based on how immunosuppressive versus active the tissue microenvironment is at baseline?
It's a great question. And I don't want to advocate that what I showed you can work for any cell type in any organ, not at all. I can only speak to the tissues and the cells we tested. So, but we tested much more than I showed you. I just didn't put it in because it was only in mice. So I'll give you an example. We used, for example, endothelial cells and as factories, and we transplanted them in hypoimmune products that they would produce, for example, anti-1 antitrypsin deficiency to overcome that. So we tried that approach. We tried allo CAR T. So for the oncology space, if you think about an off-the-shelf CAR T product, hypoimmune could help. So Sana is actually filing an IND as early as end of this year in the allo CAR T space. It will be the first time where we look for the hypoimmune platform in humans. So it would be the first indication. I would love this platform to really be explored for any other cell type, but we have to be careful. It might not work for every cell type we want to do or any organ. I would be, for example, very careful with when we think about the CNS and we put an immune evasive cell product into the brain, how we want to control that. The antibody technology I showed you with anti-CD47 is one option, but antibodies really don't break the blood-brain barrier when you need it. So I think we have to think carefully where this technology can add benefit and where it might need modifications.
Great, thank you. We have another question from one of the panelists, Dr. Zhu. Have cancer cells naturally identified ways of evading immunity that can be adapted for your purposes?
It's a great question because we looked into two concepts where grafts survive, that is fetal-maternal tolerance during pregnancy, because half of the fetus, the proteins are from the father. Mother's immune system is not recognizing them. We tested those and we tested from tumors. So CD47 is highly expressed on many tumors, not on all. And we looked more into the pregnancy model because the tumor model is not an allogenic model. It's always our own tumor cell. So the molecules we found in tumor biology did not necessarily help us that much for the allotransplant issue. The molecules we found in the syncytial trophoblasts when we homogenized and screened, those were really helpful. The CD47 was highly overexpressed, almost no MHC, really downregulated. So because the fetus is an allotransplant, we found that a little bit more helpful in basically stealing those molecules that nature is already using to have an allograft surviving during pregnancy.
It's fascinating.
Thank you.
Yeah, please do continue putting questions in the Q&A. I had one question. So I think that, yeah, I think that the huge promise of the technology that you're describing is that you could potentially develop like an off-the-shelf, regenerative medicine that doesn't require, waiting on like a long transplant list, getting matching. I'm wondering if you feel like it doesn't really matter which iPSC cell you use, or like the characteristics of the patient or of the individual who donates their iPSCs, or I guess the cells from which the iPSCs are derived. Do you have thoughts on that? Like, what would make the ideal donor of an iPSC cell for the purposes of developing an off-the-shelf therapy?
I think we could discuss it for days, but I share my personal view on that. So I think age matters. So the iPSC donor, like maybe below 40 might be more ideal than above 80. But what really from the transplant perspective matters, what I just showed you, are the ABOs. We have shown that in the past that when we do solid organ transplantation, we pay a lot of attention to have ABO compatible transplants. And that is, of course, the same for cell transplantation, tissue transplantation. And then what else? And then, of course, very well characterized when you have the iPSCs, the cell bank, that you don't have like oncogenic mutations, etc. But from a donor characteristic, I think age and ABO would be the two things I would mainly look at.
And it really seems as though, based on like the lack of a humoral response, that like minor antigenic differences between individuals probably don't matter that much, right? Like SNPs here and there are not going to be enough to
That's a
induce.
great question. We looked actually into that. Yes, we looked into SNPs in transplantation. We used the kidney and liver transplant in that paper. I can send that to you, Jesse. What we have seen is SNPs do matter, but they are HLA presented, so for hypoimmune. And that is actually a really important part, because if you think about HLA reduction or downregulation, that is not good enough. The SNPs, the minor histocompatibility antigens, the MIHAs, would get presented. You would get some graft recognition. So you have to go for that knockout, which then gives you the issue I showed you with the innate immune system. But because of those SNPs, that is actually a very important point. Yes.
Fantastic. Thank you once again, Dr. Schrepfer. And if anyone has any additional questions that come up over the course of the next talk, please do continue to put those into the Q&A, and Dr. Schrepfer hopefully will be able to get to those. And so with that, I need to pull up my speaker bios. Aha. So I'm now very excited. We are very fortunate to be joined now by Dr. Lou Cantley for the final talk of the session, which is entitled Altered Liver Metabolism Confounds the Effectiveness of PI3 kinase inhibitors for treating breast and endometrial cancers. And in case you were wondering, you can very much trust what Dr. Cantley has to say on the topic of PI3 kinase, because he's the one who discovered it around 20 years ago. And he and his group have since made key discoveries about the many roles that PI3 kinase and other kinases play in tumorigenesis and metabolic diseases, and I'm sure among many others. And Dr. Cantley has founded multiple biotech companies. He's a member of the American Academy of Arts and Sciences and the National Academy of Sciences. And he has recently, I hear, returned to Boston as professor of cell biology at Harvard Medical School. So Dr. Cantley, thank you so much for being here, and please take it away.
Okay, thank you very much. Let's see, remind me how I get my talk uploaded here.
Sure, you hit the share screen button down at the bottom.